Hello. Hello.
Good morning. Jai Jagannath. I welcome everybody to this vibrant temple city, Bhubaneswar. So we are hosting 6th annual Congress of I-OSICON. So we have already started the day at 8 a.m. in Hall B, where there is a oral presentations by the research scholars, and we'll start the main session on day 1 in this hall. All of you must have seen the beautifully crafted, although busy and back-to-back sessions. So I request everybody to earn knowledge, interact, and you have seen there are star attractions. The Nobel laureate of 2018, Dr. James Allison, along with Dr. Padmini Sharma, Dr. Betty Kims, Sangeeta Goswami, and few other stalwarts in the field of immuno-oncology. So respected seniors, friends, and colleagues from oncology fraternity, I once again welcome to this scientific feast. So when you are in the city for next 3 days, please reach any of us in any case, any difficulties. Siddhant, put the slide. So now I'll introduce 2 of my young colleagues. So young and budding medical oncologist. So I started my oncology career with Dr. Dinesh Pindarkar sir in year 2000 while I was in Delhi before I joined my DM curriculum. So Dr. Pindarkar sir is my first oncologist whom I started working with, and later on, so it's almost 20 years, January 2005 and January 2025 is almost more than 2 decades now here in state of Odisha, back to my roots. So Dr. Antara is from Dehradun. So she is my 3rd year DM student at some hospital. So besides academics, she is a wholesome lady, so who is a good anchor, I can say, so sings beautifully. And we have another, Dr. Ritu Singhal. So she is our 2nd year DM students from some hospital. So another charming lady. So I will hand over the mic to the master of ceremonies for next 3 days. And I hope that these 2 ladies will do their job nicely, and I'll request everybody to cooperate in the interest of time. Please don't feel bad because because we are already delayed, so we have a back-to-back program. So please feel comfortable, and I will request everybody to finish on time, and we can always interact at tea, at lunch, and always outside the hall. Thank you so much.
A very good morning to you all, and a very warm welcome to the land of Jagannath. And thank you so much, Ghanshyam sir, for this beautiful introduction. So we'll be starting our first session on cellular therapies. And for this, I would like to call upon the stage the chairpersons for this session, Dr. Sanghamitra Patti ma'am, scientist RMRC Bhubaneswar, and Dr. Dinesh Pendarkar sir, medical oncologist from Delhi. Please give a huge round of applause. You know, it really brings out the energy in the room.
Thank you. First of all, thanks Professor Bishwas for setting the agenda and setting the context so nicely. Once again, I, on behalf of ICMR, welcome each one of you to this beautiful city. And in fact, the time is so appropriate because recently we concluded Make in Odisha Conclave, that is Utkarsh Odisha Conclave. I am Sanghamitra Pati, and currently I am holding the position of Additional Director General of ICMR. And apart from that, I am also holding the position of Director of ICMR's Institute at Bhubaneswar. Basically, I am a physician turned researcher. So it's my pleasure to invite Dr. Albina Nizar, who will be presenting on off-the-shelf CAR T cells, challenges and opportunity in India. Welcome, ma'am. I think she needs a big round of warm applause.
Good morning, everyone. We're starting very early, so I'm sure many of you must have missed breakfast for this, but I'll try to keep it very simple. So where is my this now? यहाँ खोलूं कि वहाँ खोलूं? नहीं, मैं यहाँ से भी देख सकती हूँ. Okay. I would like to mention that I'm here trying to fill very difficult shoes of Dr. Naveen Khatri, who couldn't be here because of his official engagements at back in ATRAC, but I'll try to make best of whatever has been assigned to me. So I'm here to talk about the off-shelf CAR T cells, the challenges and opportunities, specifically about India. As we all know, that there's an evolving landscape of CGT in the last few years, and in 2024, as you can see, there are about more than 219 cell therapy and gene therapy products, which have come out just only in 2024, which should explain how the field is evolving. But despite the evolution and the development, there are exorbitant costs, which is obviously a major challenge in the field of all cellular therapies, and particularly in LMICs, where it's completely unaffordable. In the last 2-3 years, we have seen an upsurge in the field of allogeneic off-the-shelf CAR T cell therapies. As you can see on the right, the global landscape for allogeneic field describes that there is going to be 29.8% increase by 2032, and most of it is because of the challenges which are on the autologous field, to which I'll be coming in the subsequent slides. As you see, the current market trend shows a deviation from focus on autologous towards allogeneic, with around more than 50% now focusing commercial as well as academics focusing on allogeneic CAR T cell therapies. So this describes the global leading innovators in the field of allogeneic cell therapies. Allogene is the oldest one, which has 2 different products, one for multiple myeloma and another also for hematological malignancies, but for improved persistence. Then CLAD has for solid and hematological malignancies, Fate Therapeutics, Bluebird, and Posida. Posida is important to mention because it owns the Sleeping Beauty platform, and hence most of the allogeneic molecules, cell therapies are coming out from Posida. As we all know, India is now on the CAR-T map with around 7 clinical trials ongoing in India. And with the Okay, so we already know that India is already on the CAR-T map with now another CAR-T product also FDA CDSU approved just in January itself, with Make in India CAR-T cell therapy products taking the global market because of their cost effectiveness. But as far as the allogenic CAR-T landscape goes, there is still a lot more that we have to do in that direction, with only 2 commercial companies right now focusing on allogenic CARs, NCure, which is focusing on the solid tumor and more on the CAR-NK side, and I-OSICON Bio also focusing on CAR-NK. And a few academic centers like CMC Vellore and TMC and Jamia Hamdard who are focusing on the R&D development of allogenic CARs. So what is the need? And the need for shift for allogenic CAR-T therapies lies in the challenges of the autologous CAR-T cell products. Not just the high cost, but also the delays in the manufacturing and shipping, the long vein-to-vein time, the logistic challenges, the need for a tailored autologous patient-derived product with poor immune quality at times because of the patient characteristics, and the complex production process, and of course the long production time also, which delays the vein-to-vein time. So allogenic products have the capacity to potential to lead the CGT revolution, I would say, because it increases the access towards the patient. The cost is down by 1/10 to 1/20th in comparison of the autologous. The speed and the reliability and the quality of the product is also better because it's made from healthy patients. And there is also more room for innovation in terms of more product optimization, more engineering techniques, though it comes with its cons, but that's what we are going to come to later. So overall manufacturing process of CAR-T allogenics is very similar to the autologous one, the sample, but the difference here is the sample is taken from the cells are taken from a healthy donor, post-activation and CAR transduction. There is, of course, gene editing in order to make it allogenic, and I'll come to that later. And finally, there is expansion, formulation, purification, and then batch manufacturing, and the transfer to the patient. So as you can see here, forgive me for the font, so there's some issue with the font with transfer of the files. So as you can see, in comparison to allogenic, autologous, the allogenic, there is an extended phase of electroporation, post-purification, freezing, and batch freezing in terms of the that one batch which is taken from one healthy donor. But after that, this batch is ready to serve more than 100 patients. So the timeline is depending on the demand with HLA matching and the type of the patient it is required to, in comparison to autologous, which is only specific for one patient, and the lead time is around 4 weeks. So now the desired starting material for all off-the-shelf allogenic cell therapies can be different. We could be using iPSCs, the healthy donors, or cord blood. And we could also be targeting alpha-beta T cells, gamma-delta T cells, INT T cells, or for that matter, the NK cells. So this table reflects some of the allogenic CAR-T strategies by virtue of the clinical studies which are ongoing. As you can see, most of the focus is on T cells, and there are different targets ranging from BCMA to CD20, CD22, mesothelin in solid tumors, EBV-specific T cells, gamma-delta T cells, and iNKT T cells. And there are different genetic modifications which are being done for these various off-shelf CAR-T cell products. In order to avoid GVHD, the most important focus is on TRAC1, the gene knockout of TRAC1, and then also in order to mediate the host, avoid the host-mediated graft rejection, there is disruption of the HLA genes, restriction of MHC recognition, and there is also a use of CD52 knockout or using antibodies against CD52 for preventing fratricide also. And then allogenic cells off-the-shelf CAR-T cells also come with less persistence, because of which, so there is a lot of focus on PD-L1 also. And then, of course, the cytokine support. So this describes the different gene editing technologies. I'm not going to go in detail in it, but these are TALEN, CRISPR, zinc finger-based editing, which are the common immunoengineering gene editing techniques used for gene knockouts and molecular cloning. So on the non-gene editing front, there is one successful report of a non-gene edited allogenic CAR, which is developed by a company called CARSGen. And they have used, already this product is in clinical trial. Similar reports on allogenic CD19-based CAR, where they ensured that there is intracellular retention of TCR via a specifically developed CAR. This again describes the successful published clinical experience, but based on the different types of techniques. As you see, there is most focus on TALEN, and a lot of other gene editing technologies are also coming up, which are being used for different types of cells. You can see PBMC is also there, iPSC is also there. So these are few examples I would like to run by. This is an example of a UCART19, which has shown successful clinical responses for BLL with OR of 48%, and only 2 patients developed mild GVHD grade 1. Here, 2 modifications were done, TRAC1 and CD52 knockout both. This is again another example of an allogenic CAR which was TALEN-based, and this was done in 2 infants with CD19 positive BALL, and both of them achieved molecular remissions within 28 days, and there was successful persistence of UCAR19 cells. So this is another example of a pooled donor-derived 4th generation CAR-T cells with CD19 and CD123 target for CD19 negative relapse case, and 7 and 11 months disease-free survival was observed in 2 patients out of the 3 patients. This again is another example of an allogenic CAR for multiple myeloma, which is currently in clinical trials, and this is a trial from China. This is an example of an allogenic CAR for solid tumor, where they've used dual CAR system. One is for FAP CAR, and another is inducible mesothelin CAR. It's a logic gate-based engineering strategy, and this has been observed in mice models, and there's no clinical trial ongoing, but reduced tumor burden and increased survival has been seen in TNBC models. This is an example of using virus-specific T cells for CD30 positive lymphoma, where the CAR T cells where the viral-specific, EBV viral-specific T cells were made allogenic and then transduced and then made allogenic. And this is also in clinical trial, and the manufacturing process is unique because it's used for, it's using platelet lysate medium for improving the specificity. This is another example of an allogenic CAR, which is an iNKT-based CAR. So iNKT, allogenic iNKT is also, this was initially used for cancer, but they observed that it has anti-inflammatory properties. So they used for ARDS in SARS. And there was safety and tolerability which was established with this study. Again, a similar study for allogenic gamma delta CAR T cells, which was used for B-cell malignancies, CD-positive tumors. And beyond cancer, allogenic CARs are now being used for autoimmune disorders. This is an example of 3 patients who were given allogenic CARs and no adverse effects, no neurotoxicity, and improved long-term efficacy even after B-cell recovery was observed. So what are the challenges associated with allogenic CARs, GVHD and HVG? And there are now established gene editing technologies which can target to control and eliminate the MHC molecules, or even, and these are 2 classical examples of the commercial CARs where they've targeted, they have eliminated MHC molecules, and they've also downregulated the NK cell inhibitory ligands. I don't want to get into the details, just for the interest of time. Then another approach is the enhanced lymphodepletion at the clinical front, where CD52 antibodies can be used in order to further strategize the lymphodepletion techniques, which are the treatment which is given to the patient before the infusion. And then there are also dual targeting CAR T cells to control the fratricide issue associated with allogenic CARs. Now, opportunities in India, is India really ready for allogenic therapy? So, as we know that the major CAR T pricing is located in the manufacturing and the clinical development aspect of the whole pathway for cell therapy, CAR T cell pathway. So I would say that the pathway which has been developed over the last 4 years, especially now, not only in the already established centers, but also the upcoming centers, the timing is perfect because there is a cost-effective pathway already in place, there is a regulatory pathway already established, and the government policies are also more favorable towards the development of more products. So we could have a quality by design approach, which we have established specifically in our center, which could target large-scale production of allogenic CARs, similar to a CMO-CRO hybrid model, and using different manufacturing platforms, manual or semi-automated, who could keep the cost further low, and use the same platform which has been used for autologous development towards allogenic, which could also keep the cost further low. Design our own GMP academic facilities, point-of-care facilities, and many of the academic centers and commercial centers are more coming up with such facilities, which could be used as CDMOs or CRMOs also for international clients. And already established regionalized, centralized model, which was developed by Immuno-Oncology and is now being taken by many other centers across the country. So the challenges which would be specific for India would be, of course, the working capital, more investment in R&D, the timelines which is there for the regulatory and the legal pathway, how much is the interest from the commercial market, the international commercial market, the manufacturing and the quality control, because that's one of the most biggest bottlenecks of allogenic cell therapies, supply chain and distribution, transport from the POCs to the clinical centers, and of course, safety concerns.
Time is up.
Yeah, I'm done. This is my last slide. And of course, you would need more robust clinical trial designs. So the pathway ahead, I would say, more investment on R&D side, focus more on immunoengineering and product optimization, use of existing data, what to do and what not to do, collaborative studies with industry, more academia-industry collaborations, hybrid CDMO models, and POCs in academic centers. With that, I would like to thank everybody and the organizers for this opportunity, and would also like to invite you all for our cell therapy conference, which is scheduled for 6th to 9th March in Mumbai, organized by CAR T Cell Center, Tata Memorial Center, APTRAC. Thank you.
Thank you very much. And thanks, Dr. Vishwas, for giving us this opportunity. So our next presentation is cellular therapies other than CAR T and applicability in India by Dr. Bhausaheb Bagal from Medical Oncology, Mumbai. Please.
Thank you, sir, for the introduction, and thank you, organizing team, for giving me this opportunity to treat. And I've been asked to speak on the niche area, non-CAR T cellular therapies, and that too in the Indian context. I will try and do justice to the topic given to me. Can I have my slides, please? Oh, thank you. Yeah, so I've chosen two areas where I think that there is some work being done in India and which is applicable or doable in our setup. And the first one is NK cells, because they are an important component of innate immune system, and they are acting actively. We see immature cells mainly in the lymphocytes and the periphery, while when they are exposed to the cells, they become mature and CD56 goes down. So a basic biology about this, so not only they are able to kill on their own, but they are an important part of the immune system where they interact with both myeloid and lymphoid component, and also dendritic cell for antigen presentation. And how they kill is through the fast and fast ligand pathways by degranulation and releasing the perforin and granzymes, and also ADCC, which is can be exploited to combine them with antibody-based therapy. How NK cells are regulated is a bit complex phenomena. Inhibitory and activating signals interact, but ultimately this balance determines what happens, whether it is going to kill a cell or not. If this balance is in harmony, the NK cell remain inactive. But when it is activated, for example, missing cells, so tumor downregulate MSC, and when MSC is not present on a particular cell, the NK cell identified it as a foreign and it is killed. So that's one way. Second thing is, under stress, NK cell senses the imbalance on the stress cell, for example, after infection, and this is how it is killed. There are many advantages to NK cell as opposed to T cells. So they are cytotoxic without prior sensitization. They are able to infiltrate deep within the tumor and overcome the immunosuppressive environment. They don't secrete a lot of cytokines which make them safer, but at the same time they activate other immune cells, so making it a multi-pronged approach. But there are challenges like they are short-lived, fragile, numbers is an issue, and they require cytokine support because T cells are IL-2 secreting, so they proliferate on their own. The initial hint of their use in the clinical picture came from the very early studies by Ragheri et al., and where in KIR mismatched post-transplant setting, the AML patient with KIR mismatch did not relapse. So this is how it all started. And there was a clear difference between the people who had a KIR mismatch on the myeloid transplant program, while there was not much of a difference when you did it for lymphoid malignancies. And this has been aptly exploited. For example, this paper from shows use of NK cell post-transplant after doing conditioning with Flumel, post-transplant cyclophosphamide, and the infusion of the CAR T cell therapy. And this MD Anderson study nicely shows that not only you have a safer transplant period, you engraft on time, you don't have increased acute GVHD, but you also have control of infection. So for example, CME, BK virus infections are lesser in this setting, and obviously the relapses were less. So definitely this is a retrospective comparison with their old data, but this is an early hint. Similar thing has been done from CMC Vellore by Dr. Uday Kulkarni and his group. And what they did is they use NK cell for a refractory acute myeloid patient, where they treat them with fludarabine RSE, and then they gave NK cells. After that, this was followed by a transplant. But definitely some encouraging result can be seen in this very difficult to treat setting. On similar front at our institute, Dr. Chetan is doing some work under guidance of Banawali sir, and this is his team. And he is using the GMP facility which Dr. Albina just recently showed. And this has come to a preclinical result has been validated. So for example, purity, the functional assay for interferon secretion level, proliferation assay has been done. And this should soon be coming into clinical trial for our patients. The next cell which I have chosen for today is virus-specific T cell, because infection is our problem post-transplant, and we get CMV, adeno, all these infections. There are drugs to treat these people, these patients, but definitely this is insufficient. The drugs are costly, they are associated with side effects, and they are not very useful, and this is a need of time. And based on our data, roughly 80 to 90% of our patients develop post-transplant some or other infection which are difficult to treat, and this is need of our setting. And the typical period is also period 30 days to 100 days when the immunosuppression is maximum. Once you taper down the immunosuppression, infection goes down. So that's the time when we need something for our patients. The standard therapies, as I said earlier, are not feasible, and that's why we need something in this setup. We know that virus-specific T cells are there in the blood, but they are very in the small numbers, and obviously after immunosuppression of transplant, they go down, and that's the reason why we have this difficult to treat infection becoming reactivated. So how these virus-specific T cells are non-modified are done is a PBSC collection is done. Then these cells are exposed to the viral antigens, and there are different ways of doing that. Then we choose CD4, CD8 positive specific T cells, and they are infused back. And this cell can replicate in the host itself once the person has engrafted with these cells, and they can give a long-lasting and long-term immunity. So this looks very doable thing in our setup, and there are good evidence. I would not say good evidence because this is a rare complication, so you would only see case series which shows a good response rate. So for example, in this paper from Sweden, 7 out of 8 patients had a recovery from a refractory cytomegalovirus infections. This is again from a German people where 5 of the 6 patients with refractory infections had a recovery. So definitely you can expect a good amount of recovery in this difficult to treat population by using this non-engineered NKT cells. So this cell can come from the donor which you are used for your transplant, and this would be useful if the donor is seropositive for that particular infection. But this will require a long time to get these cells. And since you have same donor, there is no risk of having additional GVHD or this or rejection in this. But if you have third-party cells which are kind of off-shelf therapies available, so they would be associated with a small risk of being rejected or having a GVHD, etc. So there are pros and cons of different approaches in this setup. And I think this is one of the large paper which shows that roughly 65 to 75% of the patient can be treated with this off-the-shelf product, which is also a good thing for in this difficult to treat setting. We have some preclinical work presented from CMC Vellore, so they have shown that these cells can be manufactured, and they have done a testing in the experimental model as of now. We are also doing similar work at ACTREC. Our work is still in the preclinical phase. So this has been studied, funded by BIRAC, and we are trying to get VST cells for adeno, CMV, and EBV, the common culprits in our transplant setup. What we have done is we identified the common HLA so that we can manufacture the cells using these donors so that they would be useful for most of our patients. And the process is same. So you identify the donor, you expose their cells to the peptides which are in-house, and we also have compared them with the commercially available stimulating agents. And we have roughly synthesized more than 50 VST products, and they have been tested in the lab, so they seems to be active, and this should be coming to forward. So this is our HLA lab, our transplant team, our cell therapy team working on this project collectively. Lastly, I think this is not a big thing, but not to forget is granulocyte. And these are also cells which can be very useful for you when you have a difficult to treat situation, like you have some severe infection in our setup. And we, not recently, but quite some time, I looked at our experience of doing it this, and we did it quite commonly. So we did 55 patient infusion in roughly 1 year period with a GCSF and dexamethasone mobilization. Our yield was quite good. I think the crux is you should have a very good granulocyte yield, at least 100 million cell per kilo minimum dose as suggested by the ring study. And if you use this, you get a good yield rise in the ANC and WBC count after infusion. And also added advantage is platelet increase. You see the rapid jump in the platelet. So even platelet transfusion come down. So this comes very handy when you have a very sick patient to treat. You see, our usage was most commonly between day 8 to day 20, but within 2 to 6 days of granulocyte infection, patient became febrile. And also the culture negativity is also very rapidly achieved. So this is something doable which should be available to this. In this cohort of patient, we had 2 patients having TRALI, but touchwood we had no deaths as of now on this study, and all these patients did well. So probably timing, patient selection, a good granulocyte dose is crux to use these cell therapies effectively. This has been done with the help of a very active transfusion medicine team, our pediatric oncology colleagues in our adult medical oncology, our granulocyte donors, and our patient itself. So with this, I will stop here, and thank you for your attention.
Thank you. Thank you for giving us the extra time.
So, thank you very much. If there are any questions to both the speakers, welcome, please. Dr. Abhina and Vagal, can you come on the dais, please? Both of you, please come. Any questions? So, even if there are no questions, I have a question. What both of you feel, how fast would we go into especially allocars and maybe NK?
Sir, with NK CAR, there are advantages and disadvantages. It's going to be safe, off-the-shelf product available. But in terms of my understanding of its current activity, it looks kind of slightly lesser active. You know, T are very violent attack. You get a good remission, good. Second issue with NK cell product, probably they are very fragile because they are sensitive to temperatures and proliferation. So I think it's a bit of a technical issue, but we would be there soon.
What about allo?
Yeah, I think there's not really much of a difference when we are comparing autologous. Both are required, autologous as well as allogenic, because there is a gap and there is a challenge in production for both. It's going to be a clinician's discretion at the end which is the product which he wants to give to that patient, because it's ultimately customized. If he feels that the patient is really going to benefit, there should be more focus on allogenic CAR. And I feel there should be focus on development of both autologous and allogenic. And India is really ready because there is already a pathway set for both. And the challenges are very similar and they're all manageable. We've already proved that.
Yeah, so I can just add that there are 2 CARs which will get approved in 25.
Sorry?
There will be at least 2 CARs will be approved in 25. There will be myeloma and lymphoma. That's expected. There are 6 hospital applications with Government of India for approval of GMP facilities in the hospital. So there are a lot of hospitals moving towards indigenous, I'll say, local point of care facilities. So I think with these 6 point of care facilities, if we all scientists together can actually come up together and design something, I think we can have really a good future.
So we are designing something very similar. It's a biomanufacturing hub which we are designing at CAR T cells at TRAC. And I feel that the more focus should be on the R&D pipeline. 3 or 4 GMP facilities throughout the country are good enough, because the scale and demand is not going to be of that sort that every academic center needs investment in terms of GMP, but there should be more focus on the R&D pipeline, because If there is no seed funding for R&D pipeline, there is not going to be any novelty, and what are you ultimately going to do in those GMPs? So there should be more focus on R&Ds, and 4 or 5 GMPs in the country and hubs should be good enough to save the production.
Thank you. Any questions, please? If no questions,
Taking forward what you told and Dr. Pendekar told, very recently I had the privilege of attending the PM's TIAC meeting, which is held under the chairmanship of Principal Scientific Advisor. Therein a huge impetus is going to be on, both cell and gene therapy product. CGT is going to be the future for coming, at least for coming 5 years, and both ICMR and DM and DBT, both are in not only, as you rightly told, not only setting up new R&D pipelines, but also how to increase the production so that it becomes affordable at the end of the day. That is something, scale-up is very important, and both DBT and ICMR are working in harmony towards that. So I see. Congratulations to both of you for being a part of that journey.
The whole village to take this. Thank you.
Thank you for the wonderful session. Now moving on to the next session, I would like to call upon Dr. Subhashish Chattopadhyay, Dr. Saumen Chakravarti and Dr. Bharat Rekhi to chair the next session. Doctor. Yes, please, can you please have a seat. Actually, our 2 speakers, Dr. Girdhari Lal, Dr. Manas Kumar Santra, they are busy right now in the upstairs. So we would like to proceed for the first session, Dr. Shubha Chiplankar.
Good morning, everyone. I have the pleasure to introduce the speaker, Dr. Shubha Chiplankar, who is a scientist, IOC SI, past president, Mumbai, and her topic for her talk is gamma delta T cells for cancer immunotherapy, no longer innocent by bystanders.
Yeah. Thank you.
Welcome, ma'am.
At the onset, I would like to thank the organizers for this invitation. I don't think the audience can see me, but I'm here. The topic I have selected today on gamma delta T cells is where my lab has been working for the past 30 years. When we have a lot of preclinical data, and I feel it's time now to think about its taking it to the clinic. Yeah, slightly better. So I'll start with this, a movie which was called The Children of a Lesser God, and I'm sure some of you must have seen it. It was way back several years ago. The theme of the movie was the society discriminates a fraction of people, considering them to be less deserving, less talented, and so on. This was a movie about a woman who was hearing impaired and speech impaired, and her struggle in life. I draw this analogy, and of course, it's called Children of a Lesser God because the God of these people is said to be less powerful from the God of the ones who are more favorable. I draw this analogy to gamma delta T cells. And the gamma delta T cells was discovered by Susumu Tonegawa way back in 1984. And nobody knew about its function, what these cells are doing. So if you go through the old literature and the reviews, they are referred to as enigmatic. Unconventional cells, mysterious, riddle-wrapped in enigma, and largely ignored. So gamma deltas are children of a lesser god in immunology. So if you look at the chronology and the milestones of gamma delta T cell research, the discovery of gamma delta T cells was in '84, but their anti-tumor function, or rather their function, was known in 2001, and that too in mice. The anti-tumor functions in humans was reported in 2003, and in 2019 was the safety and efficacy of allogeneic V delta 2 T cells in cancer patients. And the real trials now, the phase 1 trials have been initiated, say, after 2021. So we're still awaiting data on these cells. A lot has happened in between, which has led to a better understanding of these cell types. To give a brief overview of gamma delta T cells, they are strikingly different from the classical alpha-beta TCR expressing lymphocytes, which are more than 90% present in the peripheral blood. Gamma deltas constitute a mere fraction of 5 to 10%, and they form a bridge between innate and adaptive immune responses. They're very restricted in their TCR repertoire. If you look at the alpha-beta, there are 70 variable region genes, but in gamma deltas, there are 14, and out of which 6 are pseudogenes. So the two major subtypes where the gamma and the delta chain is utilized is V gamma 9, V delta 2, more than 90% in periphery, and V gamma 9 delta 1, which infiltrate tumors or which are there in the epithelial tissues. The very striking feature of gamma deltas, which distinguishes them from alpha betas, is the MHC-independent antigen recognition. So they don't need the antigen to be presented by the class 1 MHC. And as you know, in most of the tumors, MHC is downregulated, and this is the cause of immune evasion in these tumors. So gamma deltas don't need MHC presentation of the antigen. They're more like immunoglobulins. They can bind the antigens directly. Now, even if you look at what the antigens gamma delta T cell, and that was not known for several years, but now there's data that they look at non-peptidic antigens. And I'll briefly describe what these are. Upon activation, they produce copious amounts of interferon and TNF, a cytokine which is very important for cytotoxic functions. And they're killer cells. They're cytotoxic effector cells. They release perforins and granzymes when they come in contact with tumor cells. They function themselves as antigen-presenting cells, and they play an important role in immune regulation, immune surveillance. Now, coming to what gamma deltas recognize. They recognize metabolites of the mevalonate pathway. This pathway is an obligate intracellular pathway, which is involved, and those who have, if you recollect your biochemistry, this pathway is involved in cholesterol metabolism, synthesis of vitamins, dolichol, so very obligate pathway, essential pathway rather for cells. This pathway is dysregulated in tumor cells. Now when you treat these cells, with aminobisphosphonate. Bisphosphonates are given to treat patients with osteoporosis or to prevent skeletal malignancies or bone metastasis. So the zoledronate, for example, which is a nitrogen-containing bisphosphonate, it blocks the downstream farnesyl pyrophosphate synthase in the mevalonate pathway. So what happens is there is an upregulation of isopentenyl pyrophosphate, and this intermediate, this metabolite, is recognized as an antigen by gamma delta T cells. Mind you, no presentation is needed. And how IPP is presented, I'll let you know. But it is this IPP molecules in cells which is recognized as the antigen. This pathway is also present in bacteria and fungi, and it is there it is called the HD map, which is also, it's a hydroxymethyl pyrophosphate, and this HD map is more potent. So in infections, gamma delta T cells also play a very important role. It's about 1,000 times more potent than IPP. Gamma deltas are now used as candidates, as I mentioned in immunotherapy, either as unmodified gamma delta T cells or engineered gamma delta T cells. I mentioned to you the two major subsets, V delta 1 and V delta 2, and the antigens which they recognize are stress antigens, which are elevated in tumors on stress, the MICA, MICB, ULBP, and of course the delta 2 recognizing the metabolites of the mevalonate pathway. Now, way back in 1999, when the functions of gamma deltas were not known, we published the first paper from India in gamma delta T cells recognized heat shock proteins on oral tumor cells. And heat shock proteins, and we found it was a heat shock protein 60, and these HSPs are constitutively expressed in tumor cells. Nobody paid much attention to this, what we said in those days, but we went on and showed that the recognition occurs through the binding of the gamma delta TCR directly to HSP, and there are other ligands which play a role like NKG2D and so on. So from the graph, what you see is just using antibodies. This is, mind you, this is inhibition of cytotoxicity. You give V delta 2, V delta 1 subset antibodies, there is inhibition. And so we looked at also giving anti-HSP 60 and anti-HSP 60, there is inhibition. So this was very clear. From then on, mind you, I've just cherry-picked data from the work we have done. I'm not presenting all the latest data, just to prove the point. So we have been profiling gamma delta T cells in head and neck cancers, in colorectal, in gallbladder, in breast, and the way these gamma deltas infiltrate tumors is very different in different cancer scenarios. In head and neck tumors, they're there in the peripheral blood, clonally expanded, high in numbers, and much higher in the tumor infiltrating lymphocytes. And as I said, the dominant population circulation is gamma 9 pairing with delta 2, and in tumors, it could be gamma 9 delta 2 or gamma 9 V delta 1. In gallbladder cancer, surprisingly, which develop on the background of cholelithiasis, on the background of inflammation, we do find gamma deltas in the peripheral blood, but they are almost the same in the peripheral blood and the tumor tissue. Now, the reason for this is there is a subset of gamma delta T cells which is pro-tumorigenic. That is, they are the IL-17 producing gamma delta T cells, and these are elevated in gallbladder cancer patients. In colorectal cancer, the scenario is very different. They're they're downregulated almost, or they're low presence of gamma delta T cells in the tumor infiltrating lymphocytes. Interestingly, we thought that if patients are given, and breast cancer patients are given zoledronate, and we took a set of patients who were postmenopausal, breast cancer patients treated with zoledronate, and what we found, interestingly, was there is increase in activated gamma delta T cells, the CD25 positive gamma delta T cells, and these were TEMRA positive, so that the ability to perform cytotoxic functions and proliferate. So gamma deltas expand clonally expand in response to zoledronate and acquire cytotoxic potential. The strategy we used was to take tumor cells and treat tumor cells with zoledronate for some time, and then incubate them with autologous, this is a cell line, so treat them with gamma delta T cells. We also did with fresh oral tumors, and you clearly see the tumors treated with pamidronate or zoledronate are aggressively lysed by gamma delta T cells. I'm not showing you the video, but take my word for it that they develop beautiful immune synapses with the tumor cells treated with zoledronate. And of course, we looked at what are the molecules which mediate this cytotoxicity, and we found that it is through NKG2D ligand, which is very important in this interaction. Now, gamma delta T cells have the ability to also mediate what is called as antibody-dependent cellular cytotoxicity. We made monoclonals in the lab, monoclonal against the oral squamous cell carcinomas. We used this, and it was really surprising to us that the monoclonal we made against the tumor lysate also recognized heat shock proteins. So we could use it as a bridge where the FC receptor docks on to the gamma deltas, and the FAB recognizes the heat shock proteins on the tumor cells. And doing this co-culture along with zoledronate-coated tumor cells. So we took uncoated tumor cells, zoledronate-coated tumor cells, adding the antibody and the gamma delta T cells, which brought the gamma deltas in the close proximity of the tumor cells. You see, you compare the blue to the blue across, okay? And what you see is tumor cells not treated with zoledronate, about 20 to 25% kill. Once you treat them with zoledronate, you got 7 to 80% kill. This was in cell lines and oral tumors. So this was that they can directly kill tumor cells, they can mediate ADCC. Now, we were interested in understanding what regulates the functions of gamma delta T cells. Now, there was no information on this, and the only clue we had is Notch plays an important role in lineage discrimination and self-aid decision making. So we asked the question, if not just playing the role in lineage, alpha, beta, or gamma, delta, would it be also playing a role in the cytotoxic functions of gamma delta T cells? And we published this, and we showed that gamma delta T cell have the Notch intracellular domain. You have to understand the pathway, because when Notch binds Notch ligands, there is a release of the Notch intracellular domain, which goes inside the nucleus to mediate a canonical or non-canonical Notch signaling. And we showed that we used siRNA approach to block the NICD, where the killing is decreased. So it was clear that it is the Notch which regulates cytotoxic effector functions of gamma delta T cells. Very recently, we had the Unit of Excellence project in collaboration with Dr. Sanjeev Galante from ICER, and we were interested in understanding, is there a crosstalk between the T cell receptor and Notch signaling, and whether the gamma delta T cells response to the IPP produced in mevalonate pathway and the HD map produced in the Romer pathway is different. And clearly we identified signatures for IPP, where there is a IPP-treated cells, the cell cycle IF and gamma response pathways are more abundant in IPP compared to HDMAP, while in HDMAP, cytokine production, lymphocyte migration is more pronounced. And then definitely we showed that there is a crosstalk, because if you inhibit Notch signaling, either by using siRNA or gamma secretase inhibitor, the response is inhibited for the gamma deltas to recognize the antigen. Later on, we were also interested in looking at this subtype, because it is really interesting to understand, is there a pro-tumorigenic subtype lying within the gamma delta T cells? And we were thinking of what model to use, and we thought it would be a good model to select would be gallbladder cancer, which develops on the background of inflammation. And we showed that IL-17 producing gamma delta, that pro-tumorigenic subtype is elevated in the GBC patients. How much time do you have? 1 minute?
We have 1 minute.
And we correlated this with survival analysis and showing that the delta 17s do not survive, while the classical interferon gamma producers have a better survival. We also looked at what interaction gamma delta T cells have on osteoclasts. Again here, no data on gamma delta T cell interaction. So we generated the human osteoclast and looked at activation. And this was a very important finding, because we showed that there is a difference if you take naive gamma delta T cells versus activated gamma delta T cells. Activated gamma delta T cells produce more of interferon gamma, while naive produce IL-6, which is pro-tumorigenic. So we could distinguish, and these are classical assays, the PIT assays, and those who are working will understand that these are PIT assays which are used to show the bioactivity. So while selecting, because why did we do this? Because metastasis is very common, and zoledronate is often used to treat patients, so we wanted to study this interaction with the osteoclast. Now, very recent papers looking at the landscape, and this is all the CyberSort data, deconvoluted data, which was published. Interesting. A paper came in 2015, and a very recent paper now in Cell, which talks about cancer gamma delta TCR, and both showed that gamma delta T cells emerge as prognostic markers. A very important paper in Nature by Carl June's group showed that they were looking at 10-year follow-up in patients, and they showed that it is gamma delta T cells which persist in one of the out of 5, I think it was one of the patients, where they had elevated gamma delta T cells. Again, another paper which CAR T cells using gamma delta T cells. So there are a lot of approaches. I'll just finish in a minute. There are a lot of approaches now which are used, which uses unmodified gamma delta T cells, gamma delta T cells as using bispecific molecules or by using agonistic antibodies against them. I'm not going through the trial data. Several trials are on. We're still awaiting the data, but definitely it looks very promising. And for 3 reasons, gamma delta T cells bridge the innate and adaptive immune responses. MHC recognition is not required, and they are potent killer cells. I've very briefly told you all the work we have done. There are much more. We have looked at epigenetic regulation. We have looked at hypoxia. We are looking at EMT transitions. And this is all because of my students who have done fantastic work and my clinical collaborators. Thank you.
Ma'am, thank you. We can take one question if you have a question. If anyone have any question, we can take one question.
Yeah, I can't see you, but you can ask the question.
Okay, okay, you have one question.
Madam, nice talk, and I would like to know regarding CAR gamma delta and CAR NK being, having non-specific effectiveness also, non-MHC restricted. Which one you would feel that, CAR gamma delta will be better or CAR NK?
CAR gamma delta will be better than CAR NK?
I'm just asking, you think, will it be equivalently
No, I cannot comment on that unless the data is out, but they have done that. What they're doing is takes what they're, this gamma delta TCR is transduced into alpha beta, and then generating these cells. And CAR gamma delta, I showed you the data on prostate cancer. Fantastic results in the preclinical setting with CAR. I cannot comment on the comparison between CAR NK and CAR gamma delta at this moment.
Okay, thank you.
Okay, thank you. Thank you very much, ma'am.
Thank you.
So we have the next speaker, Dr. Girdhari Lal. Dr. Girdhari Lal is from NCCS Pune, and the topic is personalized vaccines for cancer therapies. How promising is the bench to bedside data? So, sir, you have 14 minutes, so keep in time so that we can have 1 or 2 questions.
Thank you, Chairperson, for introduction, and let me start thanking the organizing committee for having me here to discuss. In fact, actually, I was given to talk about this topic rather than my interest. So I'll try to discuss about this, what is personalized vaccine in cancer therapy, does it really promise, or we have some challenges to go ahead with this. So before I go on to this area, just quickly remind you that we all are exposed to every time one or other type of carcinogen. It can be chemical, radiation, or whatever, but we are still safe. We are safe because our immune system is working. And it's working because if you look at here, the immune system is always trying to actually control any transformed cells which develop in our body from the healthy control cell. And whenever the immune cell fails, irrespective of whatever the equilibrium or the controlling of this, and that's where the tumor wins, and then because of this disbalance or uncontrolled, what you have immune system dysfunction or uncontrolled way of acting on it, you have a tumor. So elimination, equilibrium, escape, all these we study in textbook knowledge. And it's true that, yes, we are actually always fighting with cancer. And example, if you have any of these molecules which are listed here, if you are deficient, you end up with developing, at least the animal model, you end up with developing spontaneous tumor. Suggesting that, yes, these cells are acting in our body and they are always fighting for it. There are other lot of experiments to prove that these 3 events happen. I'm not going to discuss that part. Let's come back to see how immune system develop, especially anti-tumor immunity, and then how it modulates a different activity. We all know in our classwork that you have a tumor cells which antigen expose the antigen presenting cell. It goes to secondary lymphoid organ, triggers your T cells, adaptive immune system, and these cells comes out of the lymph node and then circulation come back to the tumor, and that's where we are. Now, if you look at the biology or molecular event in all these steps, the molecules which are listed here in the green, if you want to actually boost this molecule, they are all going to act as the anti-tumor effect. The molecule at each one of these steps, what you see as red color, all these molecules has been exploited or need to be inhibited in order to have anti-tumor immunity. Now, coming back to the vaccine, if you want to induce vaccine, this has to follow this pathway. That means all of these network molecules or cellular interaction at different places play very important role. Now, if we look at one of the event, for example, what I showed to you, that a tumor cells immune cell APC has to go to lymph node to trigger the immune response. Here is one of the example where if you do not have a lymph node, and try to treat with your immunotherapy, for example, PD-1, and just remove the lymph node, all this production of checkpoint blocker disappears, just because the cells cannot interact in the right place where they need to supposed to working. That means this is just one example to prove that each one of these contribute in mounting the effective immune response. And then if you can see that there are a lot of not only secondary lymphoid organ, there are tertiary lymphoid organ, lot of cellular and molecular interaction happen there. And if that also can be disturbed, you actually interfere one way or other in mounting the anti-tumor immunity. For example, that's what you see, some tumors are hot, some tumors are cold, some tumors are infiltrated more. And if they are not, then immune system, actually you see that the immune system or immunotherapy is not going to work that strong until unless you modulate the microenvironment that immune cells actually start infiltrating into the tissues. Now, so important question in tumor immunology is, can immune manipulation cause the regression of established immune cancer? If it is yes, how do we take advantage of it? What are the antigens involved in immune recognition of human cancer? That's another very interesting question to understand that how do we identify what we need to target. Can anti-tumor T cells be generated in patient by immunizing with cancer antigen? Is that simple what we see in vaccine? Or there's something more we need to learn about? What mechanism limits the cancer regression despite the in vivo generation of anti-tumor T cells? So it's not that you have peripheral anti-tumor T cell is going to give you anti-tumor immunity per se. So that means there is a lot more to learn from there. Now when we're talking about vaccine, you can broadly classify them: therapeutic cancer vaccine that you treat the existing disease, or prophylactic vaccine, you are healthy but you don't want to become sick, or that's where the prophylactic vaccine prevent the development of disease or cancer in healthy individual. And when we talk about antigen, you can also broadly classify them that it is tumor-specific antigen you're talking about or tumor-associated antigen. And each one has its own advantage and limitations. When we talk about vaccine, now because we are talking about vaccine, mostly I'm going to discuss therapeutic vaccine. And if you look at the history, goes back, long back history, that how much we have progressed to develop a vaccine, therapeutic vaccine for the cancer. And any of these, you now see the n number of these diseases, these methods including RNA vaccine, cell-based vaccine, to have this type of protection or provide anti-tumor immunity. Now, what is the limitation and scope of these therapeutic vaccines? Obviously, the first thing, positive point is if we know the target, what is the antigen we are going to target? Does having antigen is sufficient, or antigen need to be processed? And like I showed to you, they need to travel to different lymphoid organ in order to capture the induced immune response. That means antigen processing and presentation. What are molecular events required to actually boost those processes in order to have effective immunity? Now, T cell activation, obviously we're talking about adaptive immunity, B cell activation. Does innate immunity also potentiated in this vaccination process? How does these cells traffic between secondary lymphoid organ and the tumor? That also plays very important. And how do we increase this process in any of our vaccine modality? That also helps it. Overall, if you look at negative side of it, what we don't know and we need to work on more is the immune evasion. And also dysfunctional T cell generation if you inject something long term. So when we talk about memory response, it's long term protection. But these are the things which limits, like dysfunctional T cell in tumor, dysfunctional APC, cytokine dysregulation. That's what we see as a toxicity, that if any vaccine induced a lot of inflammatory cytokine, you end up with a toxicity. Here the model, which I don't want to discuss, but if we want to develop a therapeutic vaccine, there are certain specific requirements. What we need to learn, that why vaccine has to be good or bad. It should have direct immunity to a specific tumor antigen. If it is non-specific, you're not targeting the the molecule or the cell type or cancer cell in this case. They clinically have enough time to mount immune response to control the progression of cancer, and should work in early stage of cancer, and should stabilize at least cancer progression or contribute in disease reduction, and should have synergy with chemotherapy, because you may actually want to combine with the chemotherapy, radiation, or hormone therapy in your treatment protocol.
Now, if you look at personalized medicine, that's what we want to discuss today, that there are several of them. There are different ways actually personalized. It can be autologous antigen, subunit vaccines, or autologous whole tumor vaccine, genetically modified tumor cells, and autologous tumor cells are adjuvant. Because if you look at the cycle which I showed it to you, a different molecular event, each one people has tried to just to boost the immunity, and each one placed a significant contribution in mounting a better memory response, or what we call as T-cell response or anti-tumor response. Likewise, there are several cellular therapy has been also adopted. The madam just mentioned about gamma delta T cells, and like dendritic cells, because you can not only just give the antigen, you pulse the antigen of your choice to these cells and inject back into the patient to mount antigen-specific immune response. And these can be manipulated, that can be seeded for the neoantigen, autologous antigen, or you can also genetically modify it or treat with mRNA vaccines and then look for the immune response. Now, when it just comes to personalized medicine, where are the secret for the cancer in individual patient? Do we know? If not, there are a variety of ways to just unvision that where is the secret, how do we find that secret? Is it just population-based study? Or do we need to have a lot of clinical data to understand that? And how all of these, what molecular finding we have, they talk to each other in a body. And ultimately, we decide on something, okay, here is the thing which is specific to patient A, B, C. Or the secret is actually within the tumor itself, where actually we need to look at. It is the patient or in the tumor or together. All right, so just to brief you that when I discuss about the vaccine, there are a variety of ways people has tried vaccine. Each one have this advantage and disadvantage. We're not going to discuss all of these, but I wanted to put forward here that there are different ways people has tried to have these different types of vaccine, therapeutic vaccines for anti-tumor immunity. Obviously, you can go back to literature, you will find n number of clinical trials is also done. I don't want to read this, just wanted to point out that yes, there are a lot more study going on in this field, a lot of clinical trial is going on to have you personalized medicine using all these methods. Now, one example I just want to quote it here. This is the mRNA vaccine that everybody think that is very hot and very interesting. This is the Keynote 942 trial, which result is out. Using miRNA 4157. What they have done, they have used either 9 to 34 different epitope per person, which they have found that this person ABC has the variable or differential data. And now, if they combine all these epitope with or without a checkpoint inhibitor molecule for the melanoma, and then ask the question, what happened to them? And what they found is that yes, it gives better response if you combine it, but now when you test in time, then it turns out there are a lot more to learn actually to really use in practice. All right, so actually this is just one example I'm quoting that where people has tried personalized vaccine for one of the disease. There are n number of studies to that. But that means if we are not able to take right away into the clinic, where are the challenges? What we are missing? So the thing is that what is the best antigen we should use for this? And that's one of the example we need to know. And also, what is the adjuvant we need to incorporate, and what type of adjuvant in the vaccine response, and also what kind of therapy we can incorporate along with that. It is hormone, metabolism, or neuropsychological one. This I just showed this pop-up here. I'm going to skip it. I'll just take one example where, let's say, we're talking about hormone and also the neurotransmitter. One of the neurotransmitter, pain is strongly associated with most of the cancer. And one of the neurotransmitter is substance P or trachykinin receptor. If you block the trachykinin receptor or antagonize trachykinin or pain receptor, you actually see tumor growth is controlled. Here what you see, just by inhibiting one. So it's a hormone immune system crosstalk. And we know that when we have treated with the chemotherapy or immunotherapy, nausea and vomiting is one of them, and the trachykinin receptor is used, antagonist is used in the patient. The moment you give them, your immunotherapy goes down. That's what you see here in the bar graph. actually the protective effective of your chemotherapy is reduced. So that means we have to understand the biology, how each one of them talk. I'm not going to discuss this, but one of this interesting thing, one of the technical officer in the lab, she was interested in looking at NK cell, and what she found is that as we look under the microscope normal condition, or look into 5,000 magnification or 10,000 magnification or 50,000 magnification using scanning electron microscope, and what you see that NK cell, when they are functional, they actually acquire this raspberry morphology. Now, what is the advantage? Does each person have this personalized development or differentiation of this NK cell? If yes, how they are functional? That's where she came with the example here that NK cell, the first kiss of NK cell with the cancer cell, and when they have it, the tumor has actually gone back, and we'll be looking at in more detail how this is. And so far, I will conclude here that personalized cancer vaccine is an excellent strategy to treat the patient with less toxicity and better clinical output. But we need a deep scientific understanding of cellular and molecular interactions, inter-organ crosstalks, metabolic and neuropsychological status. I showed you one example of neurotransmitter, pain receptor. There are a lot more other to learn. They also impact the immune response. And then if we don't do this, then we actually, our therapy does not work. Then we end up with the blaming that, okay, it's an environmental factor which decide, or the genetics of individual which decide why therapy does not work. So I'll stop here, and I acknowledge all the people and the funding agency and other collaborators, what we do in the lab. Thank you very much.
Thank you very much. So we can take one question if someone has a question. No? Ma'am.
Very nice talk, Girdhari. I would like to ask you, there has a lot of debate or discussion on precancerous lesions, and Oliver Effin's group has worked on MUC1, which is a tandem repeat as an antigen expressed. So what is your take on that?
Yeah,
Look at precancerous rather than cancerous.
Right, so when...
As a sort of preventive...
No, the last one what you said?
No, as a therapeutic, more preventive than therapeutic. And my second question is, what about the tissue-resident memory cells, which play an important role there?
Right. So the first thing is, do you have good response against MAC1 in each individual to develop a memory response? So then question comes that if you have memory cells, do you have resident memory cells? So what drives this anti-tumor immunity against MAC1? What drives the generation of tissue-specific memory cell? Because antigen exposure and migrating the cells to the tissue is the key. So, so the challenge here is that how do we know that what drives actually the expression of chemokine receptor to mobilize those cells into the tissue where they need it? So that's the thing we need to understand, that how things actually move, molecular movement happen that drives this migration of the cells.
Okay, thank you. Thank you very much. So our next speaker is Dr. Manoj Kumar Shatra. He is a scientist again from NCCS Pune. The talk is about mechanisms employed by the tumor microbiome to mediate responses to immunotherapy.
Good morning to all of you. Thank you, Chairperson, for introducing me. And so today I'll little bit change my title of talk, and I'm telling this is totally unpublished data I'm sharing with you, and I would like to thank organizer for giving me this opportunity. So today I'll speak about that importance of AKT signaling pathway in immune evasion. So you know that there are several immune checkpoint pathways are responsible for T cells activation as well as the inactivation. So basically today my talk is basically inactivation pathway, the PD-1 which is the receptor present in the T cells, and its ligand in the PD-1 which is present in the cancer cells. So if you think about it, the PD-1 and PD-L1, basically in the initial stage, basically T cells will recognize the cancer cell and then inactivate them or kill them. And when that cancer progression happens in the stage developed, actually reverse happen, the cancer cells basically inactivate the T cells. And we know that immune checkpoint inhibition, immune checkpoint therapy is routinely used. And basically the response rate is very good for the specific type of cancer like lung cancer and melanoma. And other cancer, basically the response is less than 10%. So why it is so? Why only two types of cancer, there is only 30% response, and then other cases it is not. So that is our basically driving force for studying this pathway. And as you know that there are 7 the immunotherapy antibodies is approved by FDA for the PD-1, and then PD-L1, there are 2 these. But as I said, their response to majority of the cancer is less than 10%. So if you think about the PD-1, actually the PD-1 is heavily glycosylated. And once it is glycosylated, it is highly stable, and that stable glycosylated form that will be in the membrane, and that membrane form is actually binding to its receptor present on the T cells, and that way inactivate the T cell. And if it is not in the glycosylated form, the previous study, a recent study I'll say, that will actually be targeted by the ubiquitin ligase. Okay. So it's targeted by the non-glycosylated form of the PD-L1 will be recognized by ET ubiquitin ligase, and that will facilitate the polyubiquitination. Polyubiquitination means that this is one kind of post-transcription modification where ubiquitin will be covalently conjugated with the substrate and forms the poly-1. Okay. And these polyubiquitinated proteins, PD-L1, will be targeted for proteasomal degradation, and that way T cell activation happens. But and then this ubiquitination, just here I'll give you the brief that this is a type of post-transcription modification. The ubiquitin itself is a 76 amino acid proteins, and it conjugate to that substrate to either monoubiquitination, multi-monoubiquitination, or polyubiquitination, means that at least 4 ubiquitin chain will be there, and that will be ligated to the substrate. And ubiquitin has 7 lysine residue. So when the ubiquitin chain forms, there is a specific lysine, one of this lysine will be involved, and so that number is 6, the K6 means lysine 6. Similarly, the other lysines actually the polymer form. And till date, only we know that the functional consequence of the ubiquitination is that lysine 11, lysine 48, and lysine 63. So lysine, if it is the protein, is covalently modified by ubiquitin through lysine 11 or 48, those proteins generally go for proteasomal degradation, and then clearance happen. If it is case lysine 63, this protein will be stabilized. And there are several ubiquitin ligase, I will not talk about that. So they are basically involved in different type of ubiquitination process. So then we ask how glycosylated PD-L1 is regulated in cancer. Recently, there are, the paper came out that cancer research that's showing that if you inactivate AKT, then basically the glycosylated form of PD-L1 is going down. So basically what I'm saying that the AKT pathway activation leads to stabilization of PD-L1. And you know the breast cancer, almost 70% breast cancer patient, the AKT is highly activated, constitutively activated. So if you think about it, if AKT is activated, and then one important study we published already, 2018, we found that if you inactivate AKT, there is ubiquitin ligase, FBXO31, so that gets activated. So, and basically that AKT inactivation, activation leads to the FBXO31 downregulation. And we also know that from the literature, if AKT is activated, GSK3 beta pathway is inactivated. Okay. So then we ask that whether AKT pathway stabilizing P53, the PDL1 through inactivating FBXO31 or through inactivating GSK3 beta. Okay. And that way, AKT facilitating the immune evasion. Okay. So then we studied basically that inhibitor we have treated the cells, different cells, both breast cancer and lung cancer cells, and we saw that inactivation of AKT by pharmacological inhibitor or by genetic inhibition, that the PD-1 level goes down significantly. And if we overexpress that AKT, you see the stabilization of PDL1, and this is two different, this is the exogenous and endogenous. So AKT basically positively regulates PDL1. And this is immunofluorescence. We see that expression of AKT is increasing the stability of PDL1, and if you inactivate, the PDL1 stability goes down. Okay. And so then, how actually AKT controlling the expression of PDL1? So we do see, basically here we see that there is not much difference in that mRNA level of AKT, but if we inhibit the proteasome by MG132, we see the stability of PDL1. So that means AKT post-translationally regulating the PDL1, and it actually prevents the polyubiquitination. So then we find that whether the AKT is stabilizing PDL1 through inactivation of FBXO31. So we have simple experiment we have done. We have inhibited the AKT with and without inactivation of FBXO31. And we see that if we deplete that FBXO31, then AKT inactivation mediated downregulation of PDL1 is blocked. Okay, this is the quantification. So that tells us that FBXO31 is responsible for downregulation of PDL1 when if you inactivate AKT pathway. So we have done other experiment. Okay. Then we asked how FBXO31 regulate PDL1. We did not see any change at the mRNA level as previously, but we see that FBXO31 controls PDL1 level at the post-translation level, because when we add that MG132, which is the proteasome inhibitor, so it blocks the FBXO31 mediated downregulation of PDL1. And these are that basically another experiment we have done, cyclohexamide pulses experiment, and show that FBXO31 actually regulating PDL1 at that post-translation level. Okay. And this is basically any ubiquitin ligase so far established that if they attenuate the level of protein through proteasome, so then they will actually ubiquitinate the substrate. So, and as I mentioned earlier, there are two types of ubiquitin linkage happens. If it needs to be proteasomally degraded, that is lysine 48 link and lysine 11 linked. And we found that FBXO31 basically promoting K48 linked polyubiquitination of PDL1, and that's the way it is facilitating the degradation of PDL1. Then we ask, and as I said, the PDL1 more oncogenic effect is because of its glycosylation. So then we ask that whether FBXO31 targets that oncogenic form of PD-L1 or not. So to test that, what we have done, the glycosylation sites we have mutated to glutamine, and then we check the expression of that glycosylated form and the non-glycosylated form, because PD-L1 undergoes only in linked glycosylation process. So you see that the protein, the molecular weight, basically this is the non-glycosylated form which you get, and this is the glycosylated form which molecular weight increase. And when you express the FBXO31, only glycosylated form is getting down, but non-glycosylated protein level is unaffected. So that means FBXO31 specifically targets the oncogenic form of PD-L1. Then we have that, as you know, that in the patient, previous recent studies showing that there are truncated form of PD-L1 or splice form of PD-L1 is present in the lung cancer patient, and that is basically devoid of C-terminal domain. So we have deleted C-terminal domain. We just intracellular domain we have deleted here, and then here will the transmembrane domain deleted. And this is basically C2. So we found that all the deleted form are getting targeted by FBXO31, and here all the glycosylation sites are intact. So that means FBXO31 targeted the extracellular domain of PD-L1, and that way it is degrades. Then you ask, as I said that AKT basically, AKT basically inactivates GSK3 beta, whether GSK3 beta has any role or not. So we have treated the cells with the GSK3 beta inhibitor, and we see the PD-L1 stabilize. I'll go through that. So then we have generated mutation of the GSK phosphorylation site, and we said if you mutate the GSK phosphorylation site, so then it cannot degrade, and it cannot also promote the ubiquitination of PD-L1. So then we asked that whether if we express that FBXO31, whether T cells can kill that cancer cells. So we have done the MTT assay, and these are the morphology that we can see that if we express that FBXO31 in the presence of PD-L1, the activated T cells can kill the cancer cells more efficiently, and this is the conformation assay. So this is our proposed model, that if you inactivate AKT, then GSK3 beta will phosphorylate in the 4 sites, and that phosphorylation form will be recognized by FBXO31, and that FBXO31 then promotes that K48 linked polyubiquitination of PD-L1, that then PD-L1 will be degraded, and that way growth suppression of cancer cells happens. Okay. So this is basically my group. So entire work is done by Tanisha, and these are my collaborators, and the funding, definitely intermodal funding from NCCS, Department of Biotechnology, ICMR, and DST. Thank you.
Manas, thank you very much. So the question comes up is that PD-L1 you say is phosphorylated. So what dephosphorylates PD-L1?
What? This PP2.
PP2 is the one that dephosphorylates PD-L1. So if you activate PP2, wouldn't you get, or if you suppress PP2, wouldn't you get the same function?
That we have not yet.
Okay.
Okay.
Thank you. Any other questions? Anyone has any other questions?
Yeah, it's a very nice talk.
Thank you.
So as you mentioned that AKT GSK3 beta is controlling this shift, means glycosylated and phosphorylated. Have you checked using any metabolic shift and checked that that metabolic shift is changing to glycosylated or phosphorylated form?
Very excellent question. Actually, we are in the process. We are in the process of that what kind of metabolic reprogramming happening this one.
Okay, thank you.
Okay. Thank you.
Thank you. We are going for the next speaker. Okay. Our next speaker is Professor Bijay M. Patil. He is from TMH Mumbai, Department of Medical Oncology. Welcome, Professor Patil, and his topic is, or the title is, Cellular Therapy in Solid Tumors and Practical Applications. Stage is yours, sir. Thank you.
So thank you. I was in Tata. I am no more in Tata. So the next 15 minutes you would be seeing whatever my scientist colleagues have discussed, how things are applicable in clinics. And let me start with case. There were only 15 minutes for this talk, so I've taken only 4 cases. So this is a 26-year-old male, no comorbidities, PS1, gets unfortunately diagnosed with a frontal glioblastoma multiforme. It's a IDH wild type, that's why it's a glioblastoma. And NGS is done because the patient is young, and what we come across is has MLH1 and MLH3 mutations which are there. MSI is low and PDL1 is negative. He undergoes gross tumor total resections followed by radiation with temozolomide and receives adjuvant temozolomide. But unfortunately has a progressive disease in January of 2024, and this is very recent, so he is unresectable and re-radiation is not feasible. He receives the standard CCNU plus bevacizumab from Jan 2024 to September 2024, has a progressive disease in October 2024. Because he has had MLH1 and MLH3, he gets discussed in a molecular tumor board and he's been offered olaparib with pembrolizumab, and on which he has progression within 2 to 3 months. The performance status of this patient is PS1, he's 26-year-old, and now he's standing in front of you at what are the treatment options this patient has. Now, whenever we come across such cases, we go to the NCCN guideline or ESMO guidelines and see what are the options. And you could see that this patient has actually exhausted all the options which are there. Now, it was not a surprise that the tumor kept coming. When you looked at the median progression-free survival with all the lines of therapies which are there for GBM, it's between 2 to 3 months, and the overall survival is between 6 to 8 months in this situation. Now, what to do in such situations? Let's look at some of the options of CNS CAR-Ts which have got developed, and let me first discuss the option of EGFR variant 3, seen in 30% of newly diagnosed DBM, and in those in which it is there, it has poor prognosis. Now, as opposed to CAR-Ts which are done for other cancers, CAR-Ts for GBM are more or less which have got developed well, are those which are intracavitary, or either given by a Ommaya reservoir or by a Rickman catheter. Now, this was a peripheral EGFR variant 3 CAR-T. The interesting thing was the response was 90% plus in these patients, but the sustenance of the response was poor. In none of the patients, the response sustained beyond 6 months, and you got a median survival of 251 days, which still looks promising, considering that these patients will post 3 lines of therapy. We had this intraventricular CAR-T, which was developed by MGH, in which again the response was 100%. This was only 3-patient study, but the sustained response was again poor. In between 6 to 9 months, all the patients nearly had a progression. Now, every CAR-T is not the same. We had a paper from Dr. Goff's lab that suggested that in this CAR-T there was not even a single response which was seen. So what we would see with EGFR variant 3 is that the patients are responding, but it is not sustaining, and most of the patients within 6 to 9 months are progressing. Let me shift gears to something called interleukin-13 or alpha-2 CAR-T in this situation. This is a unique molecule which is expressed in nearly 90% plus of GBM. And initial studies from Dr. Christopher Brown showed that the response rates are around 60 to 70%. It is intracavitary, so it's a multi-dose CAR-T as opposed to traditionally a single-dose CAR-T which has been given. And the response rates in few of the patients were till 1.5 years also, and the response durations were continued beyond 9 to 10 months in this situation. Now, this year in 2024, we got a paper around 57 patients who were treated with intraventricular interleukin-13 R alpha-2 CAR-T. Interestingly, what we saw is nearly 20% of the patients of relapsed GBM, which were post 3 lines of therapy, going beyond 3 years. These are one of the most encouraging results for relapsed GBM with this CAR-T. Now, obviously, as scientists get greedy, so there was a thing that why not combine EGFR and interleukin to both CAR-Ts. One is giving good response rates, one is giving very good sustainability. Why not combine both of them? And that was tried. Unfortunately, what happened was in this situation, what we saw was there was more amount of neurotoxicity which was done, and the response rates were not there. Now, is there any other CAR-T which you can work? Now, in Indian databases, 15.4% of GBMs have high HER2/neu expressions. Now, you need to be clear that just HER2/neu 3+ does not mean significance. So high means really high expression has to be seen. And there is published literature to show that a dose of around 1 × 10⁶ to 10⁸ can give you a 50% response rate in a HER2-positive CAR-T, and around by 2 years, 30% patients are still continuing with the response. So again, a very good target. So what could be offered in that patient would be interleukin-13 or alpha-2 CAR-T, which would give around 60–70% response rate with sustainability of for 3 years of around 20%, or if it's HER2 positive, which he was not, then there was an option of doing an HER2/neu CAR-T in that patient. Let me go to a case 2. This is a 22-year-old male, has a diffuse midline glioma, H3K27 mutated. A VP shunt is done, biopsy has been done, and that's why you came to know about the mutation. Received IMRT radiation, concurrent temozolomide, and after that, he actually fails. So he receives the ONC201 therapy, which quite a few people in this hall may have used in midline glioma. The disease progresses on that. On progression, the patient receives proton therapy, and post-proton, he has issues with swelling in the pons region, so he's been managed with BEV, and now he has a progression. And the question is, this patient is perfectly PS1. His swellings have disappeared after the bevacizumab. Now, what are the options which are there for these patients? And let's look at the results of GD2 CAR-T in midline glioma. Now, midline glioma, in most of the Indian series, the average survival after diagnosis is below 1 year. Most of the time, it is between 8 to 11 months. Now, with that figure in mind, let's look at the results of intravenous and intracranial GD2 CAR-T in this situation. This is the outcome of these patients after a single infusion of GD2 CAR-T. We are looking at median survivals of 21 months in these patients, which is a very remarkable finding considering what are the results of ONC201 and what are the results in the upfront patient itself. What we see in this study is that once you give this CAR-T, a good volumetric decrement is seen by 50 to 100 days, which remains sustainable. Also, the GD2 CAR-T in this situation is both given intracranially and peripherally. It is sustained in the CSF for a long period of time, and you could find that this CAR-T is sustained in the CSF till 500 days. And even when you look at the interleukin and interferon markers, you could see that for 300 days these are active in the CSF. Interestingly, the response here is a bit deferred. You would see continuation of response has been shown in this figure in the middle, where you could see that pons lesion is decreasing till 16 months. So one of the things which you must understand is you might get a continuous response for a long period of time. There is a good sustainability with this, and median survivals in the range of around 20 months can be achieved with this. Was this one-off paper? No. 7th Jan in the Nature, we got another paper of B7H3 targeting CAR-T in diffuse pontine gliomas. This was a UK study, and what was interestingly seen is that similar results were recapitulated. We saw again median survivals of 18 months or more in these patients. Another interesting thing which we saw, and I would want you to focus on the figure C. What they did in this study was they gave the option to the patient to undergo the CAR-T either after the standard therapy when they progress or before progression itself. What they saw was those patients who underwent CAR-T before progression, that is, radiation is given, temozolomide for 6 months, and even if there is no progression, the patient was taken on the CAR-T, their 3-year survival was 40% in this situation, which is a remarkable number for H3K27 mutated midline gliomas. Both these data have gone for FDA approvals, and maybe in this year we might be seeing some approvals or actual accelerated approvals coming for this. Again, the same pattern has been seen here. We could see that delayed responses to 6 to 8 months are seen in this situation in these patients. Let me go to another case, and I hope I have time left. Okay, there are 5 minutes left. So this is a 72-year-old male. He is a salivary duct carcinoma, and it is HER2/neu positive. He undergoes surgery followed by radiation in January of 2023, and then he has a repeated episodes of loco-regional recurrences with skin nodules coming and lung mets coming. Receives trastuzumab, has a progression in 3 months. Receives TDM1, has a progression in 3.5 months. Received capecitabine and lapatinib, has a progression in 4 months. Receives TDX and has a progression in 2 months. And what do you do for this patient? This patient, we actually did a HER2/neu CAR-T for this patient. It was a 2nd generation CAR-T done after lymphodepletion for FC, and an infusion was given on 3rd December. He's awaiting his response assessment, but this was the actual morphological response which was seen within day 10 before decrement. At present, morphologically, the patient is in CR, and we are awaiting his response assessment at day 60 to confirm that the patient is completely in CR. Let me go to another case. Another difficult to treat tumor, pancreatic adenocarcinoma. Gets diagnosed in August of 2024 with a lot of peritoneal deposits. Receives 4 cycles of FOLFIRINOX, has a progression. Receives nab-paclitaxel gem, has progression. Has a KRAS G12D mutation. Discussions were going for sending him to a clinical trial for KRAS G12D TCR therapy. And at that time, a claudin 18.2 was done for this patient, which came 99% positive in cells, which was 3+. And hence, what is the role of claudin 18.2 in GI cancers in which it comes positive? We have data which came in ASCO of 2024, which shows that post multiple lines treated patients, the response rate overall is 39%. For gastric and GE junction tumor, it's around 55% in this situation. Now, it doesn't seem very interesting, but when we looked at what are the data for FDA-approved therapies in third-line therapy, let's take an example of colon cancer, for friquintinib, for regorafenib or TAS, you would see the response rates are in the tune between 1% to 10%. And in the light of this, when you look at this response rate between 39 to 55%, that seems to be more interesting in these patients. We got this recent GI ASCO, we saw a large trial of 134 patients in which 98 patients were treated. These patients had a claudin 18.2 expression of 70% or more. 63% of the patients were third line or beyond. 80% had peritoneal metastasis. And they were smart enough to exclude patients which had massive liver metastasis. What we saw was a response rate of 39%, which in gastric tumors was 55%, and in pancreatic ductal adenocarcinoma cancer was 20%. What was interesting, and I want you to focus on the median PFS graph, if you had selected the patient which had high expression of claudin 18.2, then around 20% of the patients went beyond 24 months in this situation. It's very important that not only your patient has to be fit, your tumor marker neoantigen selected has to be perfect, and a high amount of expression has to be seen in this tumor. Now, up till now, everything data of CAR-Ts has had to be about phase 1 data or phase 2 data until 30th of December. And this is for Certecil, Carcigen announced that its first randomized trial for CAR-T, which was done for solid tumors. This was specifically done in gastric and gastroesophageal junction tumors, which were claudin 18.2 positive, in patients who were third line or beyond, randomized to either CAR-T, single infusion, or for irinotecan, docetaxel, or apatinib. And what we saw was there was a PFS and a survival benefit for this patient. This is a press release, so I can't share more data for you. I even don't have access to this. But it seems that this patient, we gave claudin 18.2 on 21st of December 2014. He is awaiting a response assessment, but his pain in back, his breathlessness, everything has disappeared. What I'm showed you around is also its graph of day 30 and day 50, in which we could see how nicely the CAR-T is proliferating in the blood of this patient. Now, to conclude, this is newer forms of therapies. Here, the new data are going to come. Patient selection is always going to be the key. These are patients who are post multiple lines of treatment. So please select patients who are fit for the therapies. Selection of neoantigen is key. Selection of those antigens which are tumor specific, you are bound to get more success. Timing is also the key. If you wait for more lines of therapies to get exhausted, then the lymphocytes get very depleted, and in this, the quality of product is not good. And more important is, which are not spoken about, management of CRS and ICANN in this patient is of prime importance, because these patients are post multiple lines of therapies, and they are fragile. With that, I thank you for your patient listening.
Any question? Yeah, please.
Excellent talk, sir. Just one question. Is there a difference in the type and incidence of the side effects that we see in solid CAR-T versus hematological CAR-T?
So there are, you see a difference in CAR-T, CRS and ICANN, which differs according to different CAR-Ts. And I'll give you 3 examples. You do a GPC3 CAR for hepatocellular cancers, you hardly see CRS or ICANN happening. You do a TCR for KRAS G12D, you are definitely going to find a CRS and ICANN happening. You do a claudin 18.2 and go for the, if you want a good response, you want to go for higher doses like 600 to the power 10 raised to 6, you will find CRS and ICANN happening. expect in solid tumors when the CRA is going to happen happens within first 2 to 3 days itself, and ICANS also happens within first 2 to 3 days itself. It does not happen a bit delayed. It happens within, so we have patients in which within first 12 hours we have started seeing high-grade fevers in these patients.
Thank you.
Thank you. Any other question? Okay. If there is no question, then thank you, Professor Patel. We invite our next speaker, Dr. Prashant Kumar Agarwal, Chief Scientific Officer, Datar Cancer Genetics. And his title or topic is Dressing Up Viruses to Fool Cancer, Personalized Oncolytic Vaccine for Solid Tumors. Welcome, Dr. Agarwal.
Thank you. Thank you, Dr. Girdhari, for the invitation. I think this is a cellular therapy meeting. Part of the meeting has been put to discuss about the cellular therapy, and the title which I have been told to speak is Dressing Up the Viruses to Fool the Cancer. So, for the cancer treatment over the year, the different therapy which is used, and if you dissect it and understand that over the time we evolved, and surgery, radiation, chemotherapy, combination chemotherapy, targeted therapy, and the most advanced, what we are using practically is immunotherapy. But any such cellular therapy when we discuss, starting from TILs, CAR-T or oncolytic viruses or gamma delta, what eventually we are trying to do is that we are advancing the immunotherapy. And this advancing is happening two ways. Either you stimulate your immune system, or the other way is that you do some kind of manufacturing of those cells, tweaking the cells, and the goal is ultimately that how can you stimulate the immune cells or the immune system. Now, if you see the type of immunotherapy in cancer, the way we learned the different kind of small molecular therapy, and we discussed about the PD-1, PD-L1, CXCRs. Then we developed and moved further about the adoptive therapy or adoptive cell therapy when we discussed in detail about the CAR-T, TCR, TILs, NK. Oncolytic virus, which I will detail a little bit in much detail. Of course, the different kind of oncolytic virus have been used. Very interestingly, that we are also in the era where we are using monoclonal antibodies therapy, and these are some bispecific monoclonal antibody and checkpoint monoclonal antibodies. And last is the cancer vaccines, which have been discussed and detailed previous speaker. So we kept discussing about the hot cancer, cold cancer. And in a very simple way, any cancer which are more immunogenic versus non-immunogenic. And this is what you could see, the cold cancer, which is non-T-cell inflamed tumors. While what actually this oncolytic viruses or any such immune trigger does, it actually triggers from cold cancer to hot cancer. And this is where we call T-cell mediated tumor killing. And oncolytic virus, which I will discuss, what it does, it makes actually hot cancer or hot tumor to more cold tumor to more hot tumor. And this is a very suitable for immunotherapies. So what we do, oncolytic virus, where you could see the virus, we can do two things. We can insert immune stimulating genes, or we can remove disease causing gene, which could be selective targeting of the tumor. Now, what would be the different mechanism of oncolytic adenovirus in cancer immunotherapy? You see very clearly oncolytic viruses which attack. Now, attacking on the normal cells, perfectly fine. Interferon gamma pathway is very activated, and this is completely unaffected. But once it goes and attacks to the tumor cells, what it does, it divides. And over the time when it divides, there's a lysis of the cell, and cancer cells lysis happen. And these viruses coming out and re-attacking on the other tumor cells. And this is what you could see, the release of tumor and virus antigen, uptake of antigen by disease, and generation of tumor and virus-specific CD8 cells, T-cell migration, immune infiltration, and this leads to the immune-mediated lysis of tumor cells. So when we talk about the oncolytic viruses, there are multiple set of viruses which have been used, and some of the examples you could see. the one which we used enveloped viruses, and the another set of the viruses which are non-enveloped or unenveloped. And then the enveloped virus, there are multiple examples. But what is very interesting that over the time, if you see the kind of clinical trials have been done and executed using the adenovirus is on the top, followed by many other viruses, herpes, Rio, pox viruses. So milestones, not going much detail. What is very interesting is that I would like to bring your attention that 2015, this is the only FDA-approved oncolytic virus treatment which was used for melanoma, and this has got FDA approval. And this is something very interesting. We know the virus could in fact and acquired natural resistance, but over the time we understood that how can we use these viruses for oncolysis. Now, the oncolytic viruses and cancer vaccine, which actually enhance the anti-tumor response. And there are different ways. What I have shown to the left and right side, the left side where you could see there are 5 different ways it can attack. The oncolytic virus can express tumor antigen, and this is where you could see that dendritic cells loaded with the tumor antigen and attack to the tumor cells. Other ways could be the oncolytic virus coated with tumor antigen. So there is a coating, and this is where the virus with the capsid where it expresses those proteins, and this is again getting attacked the tumor cells. Oncolytic viruses expressing bites. I will detail you further. Oncolytic viruses, immunostimulatory molecules, and could be interleukins, interleukin 2. Oncolytic viruses expressing checkpoint inhibitors, and the other ways where you can combine these oncolytic viruses with the checkpoint inhibitors, or you can use the dendritic cell vaccines or CAR-T. This is one such extraordinary trial, I would say, and this is called Telemogene. TVEC, we call, with granulocyte macrophage colony stimulating proposed dual mechanism of action. The first where the virus gets attacked to the cancer cells, and it divides, it kills the cancer cells. At the same time, there is a systemic effect, what I told you, post-infection, it stimulates your immune system, and this is where you can see GM-CSF, and both combined way it is killing the cancer cells. The example given below, where the long-term complete remission of acral melanoma at diagnosis after 6 months and after 12 months, and the patient goes into complete remission. Now you compare the benefit risk between both the trials. N is 436, so 436 patients has been opted for the trial, and you could see that median overall survival, durable response by 18 months, overall response, and again treatment failure free more than 18 months, and the differences are quite high, 24%, 38%, 26%, no therapy post 18-month treatment. PeptiCard, again a novel cancer vaccine platform. So this is another lab-based. So any of this method when we develop about the TILs, CAR-T or oncolytic virus, the lab works, and what you do in the lab, and this is where you can purify HLA peptide complex. This is what it is showed. Once you purify those peptides, you can put it into the mass spectrometry, which is called LC-MS. And the goal is that you would identify the peptides, and this is what the peptide has been identified. HEX is a software which was used. 2 ways you can do. You can actually identify against those peptides using the RNA-seq data, or you have the algorithm where you can identify which of the peptides which are actually pathogenic. And based on that, validation of the candidate peptide immunogenicity, followed by validation of candidate peptides to virus binding, and evaluating the tumor reactivating use PeptiCard. And the result of the PeptiCard beautifully shown that it actually post engrafting in the mice, there is a tremendous reduction of the tumor size. Bites, we keep listening about the bites, and again it came in the focus, and it's a very simple concept. Again, when we talk about the bites, there are 2 different, you could see the receptors, the T cells which is binding for the CD3, and the other you can actually design for any anti-specific tumor cells. It could be EGFR, HER2, or any other markers, and this is where the bites have been developed. what it does simultaneously, it activates T cells where the cytokine get released, and also it binds to the tumor cells. And the beautiful example again, where it has been compared with the chemotherapy for advanced acute lymphoblastic leukemia, and you could see the survival benefit using the BiTEs. Claudin 18.2 bispecific T cell engager, and you can see the structure where this linker having the anti-CD3 and anti-Claudin 18.2. Again, the combined bispecific shows much higher survival, and this is, you could see overall survival also, which has given a very good prognostic impact in the pancreatic cancer. So oncolytic adenovirus armed with CD44 V6 BiTEs, where 3 different markers, cytokine, checkpoint inhibitor, which enhance CAR T cell activity. You could see that CAD trio, we call it CAD trio, where it actually binds to TCR, CAR T, and PDL1. And post-binding, there is a co-stimulatory signals, and you could see the multiple signaling, and this actually stimulates the immune system, and it's showing a beautiful results. What you would see here that this is the data on the mice where the trio has been given. And you could see day 0, day 10, day 24, day 45. And most of the mice were alive compared to any other combination where CART or CART-DO compared to the trio. And again, it's showing a very good survival days post-treatment to CAD. TILS is one such area which I have been working for a long time, and TILS is something that's tumor infiltrating lymphocyte. So you're not doing any manipulation to the tumor cells. You are just taking the patient tumor and expanding it in IL-2. And this TILS has been also combined with oncolytic virus mediated tumor reprogramming. And again, post-treatment, you could see day 0, day 2, day 4, 8, 12, and 16. And this combination with the TILS and oncolytic virus giving the reduction of the tumor volume compared to any other combinations. So combination of oncolytic virus and adoptive cell clinical trials for cancer, there are multiple trials are going. I just named few of them. Some of them are in the phase 1, some of them are in the phase 2. And I think much more effort has been put even in the adoptive cell therapy, including the oncolytic viruses. So this is a very good example, and people keep asking that where you are going to put the oncolytic virus. The ideal situation would be that if you directly do the injection to the tumor cells, because the solid tumor having the thick extracellular matrix. So this is giving an example that if oncolytic viruses could be inserted directly to the tumor or intravenous injections. So precision engineered oncolytic virus therapy, that's the future I would call it. And many such data when we are presenting about the CAR-T or TILS or oncolytic viruses, the time will come, the future will come when we would combine many of the therapies. The goal is that we can enhance 2 things, the survival and least impact to the patient. So what you would see in this particular image that there could be targeted therapy which we use or cytotoxic chemotherapy, but if we combine with many such therapy with either oncolytic or CAR-T cells or TILS, what would be would going to happen that you would give and kill the tumor cells more effectively and efficiently. And this is where the immune checkpoint inhibitors or any such therapy where the multiple trials are going on, where we are combining an effort on both adoptive as well as the therapy which we use routinely. And that's the future. So thank you very much for the attention, and this picture clearly shows that how you are going to kill the tumor. The clinician is putting an injection, and injection is filled with oncolytic viruses. Thank you very much.
Thank you, Dr. Agarwal. Any question? Yeah.
So I have a question here.
Yes, please.
So you said that the cells, the cancer cell burst and the oncolytic virus comes out, right? So what happens to the cytokines that are present in the cell? They all come out, right? And
yes,
there will be inflammation there, right? So how is it taken care of then?
Yeah, so this is one of the bigger challenges when we call that there's a lot of cytokines. So this happens not only with oncolytic viruses, even when we talk about the CAR-T. This is one such drawback, we call it, that there will be so many cytokines and so much cytokines will come out in the system. And for the time being, we cannot control this, because this is one of, like when you call about the immunotherapy, there are circumstances where HPD, we call hyper-progressive disease, also happens. So this is something which need to be dealt over the time.
And the second one is, you said that it's directly in the tumor, but when you put in intravenous, right?
Yes.
So how do you direct that oncolytic virus towards the tumor? Do you coat them with something?
So there are different ways you can coat that, but we use the term homing. So these cells can reach to the organ, and where especially the tumor cells, because the healthy cells, the viruses will come out. But in the tumor cells, what happens, there's interferon gamma pathway, which is getting down-regulated or it gets dysregulated. And this is where the virus survive, it grows, and it attacks to the tumor cells, specifically tumor cells, absolutely.
Okay, thank you. Any other questions?
Yeah, I have a question.
Yes, ma'am.
Why are you changing, why is the choice of the virus? Like people are shifting from adeno to Newcastle and different viruses. What is the reason? And what about development of neutralizing autoantibodies through the virus?
Yeah. See, there's no straightforward answer to this question. The way I understand, we are exploring, and there's explosion that how can you train your immune system, and how can you actually manipulate your immune system. Virus could be another ways, it's another source. Somebody can ask why CAR-T, and there's always debate that why not TILs, why not macrophages, why we need to not train the neutrophils. So there are different ways, there are different domains where researcher or the clinician are exploring and finding that where we'll get the better survival. So for me, that why viruses is very difficult to answer. The only, in the nutshell, that there are different opportunities where we can deal with the immune system, and viruses are one such tool which we use routinely, or may not be using routinely, but it may be in the future could be used based on the data what we will achieve. What was your second question, ma'am? Why we are not? I just, what exactly?
What about neutralizing antibodies to the virus being generated?
Okay, so you're telling that why not we use the neutralizing antibodies?
Are neutralizing antibodies generated in a patient after virotherapy?
I'm not getting, yeah, I'll talk to you. I'm not getting your question. Yes, ma'am.
Hi, great talk. Just wanted to, two parts to my question basically. One, I would like to share that we are also working at our center, CAR T-Cell Center on Oncolytic Virus, and very happy to share that we are very close to clinical trials. It's in collaboration with NIRH, and probably early 27, we hope to go to clinical trials for safety, first one. So my question is, when you're looking at development of oncolytic viruses in India, where do you see the choice of virus, and then where do you see the IP going? Because it's all, you have to have a clear pathway for the translational and the commercialization. So the choice of virus is obviously very important when you're talking about IP and commercialization. So do you prefer to take a product which is already developed outside and then immuno-engineer it, or are you starting like us from scratch using an indigenous strain? How are you doing it?
Very interesting question. See, the choice of viruses, even I was brainstorming that what kind of viruses could be used. Adenovirus, people keep debating that why not retrovirus, and that answer you may be well aware that why adenovirus has been given more preference compared to retroviruses. The part of the reason is any such viruses, strains are used, we would like to find a system, the beautiful way the viruses we are using because of the transduction. The system where virus can go attack, but we don't want them to integrate their genome. And this is where oncolytic viruses, the beauty of the adenovirus compared to retrovirus, that it may not go to integrate in the genome, where retrovirus can go and integrate. So adenovirus can divide multiple copies, and then they can express what you want to be. Now your question is that would you like to get it manipulated if you want to add some of the gene, or you just want the viruses to be making the multiple copies?
So my question is, are you going to use the same strains which are outside and purchase it from outside, or are you going to look at because we have a lot of pool of viruses which are underexplored in India,
yes,
which are have less pathogenicity, could also be used, like the one that we are working on. There are a couple of other R&D studies also ongoing. So my question is, what the source of the virus is basically the IP related thing. So that should be really clear from the beginning.
Agree. The ideally would be if we can develop some virus strain which is based in India. It is same like when we talk about the genomic profiling and we keep discussing that why the Caucasian population, why not we identify a variant based on Indian genome, EGFR, why not Indian variants identify and develop the therapy against that? So ideal answer would be that, of course, if any viral strain which is more effective on Indian patient, we would opt for that. But currently what people are doing, something is developed, you purchase, you buy it, do IP, sell it and develop and learn from it. So what I believe that we are learning, what has been developed in the Western world, at the same time you start doing your R&D and develop something which is very Indianized.
I believe both of them would be,
Can we have this discussion
Yes,
on the
ma'am.
tea? Can we have a discussion on the tea?
We'll take it offline. Thank
Okay.
you.
Sure, sir. Thank you very much.
Thank you very much. So,
Sir, I would like to request all the chairperson and speakers to come on stage for a group photograph.
Okay.
So now we have the panel discussion on cellular therapies, the other side of the story. And I have the pleasure to invite the moderator for this session, Dr. Hari Menon from Department of Hematology, Bengaluru. Dr. Menon, the stage is all yours.
Thank you and good morning. After this debate, which I could not be part of because I just arrived, we go back to, we know that cellular therapy is one of the emergent treatments and shows a lot of promise, but it has got a lot of other flip sides also to it. So this panel discussion that we put together, which was originally supposed to be done by my colleague and student, Dr. Hasmukh. I've just chipped in just now to get this done. So without much ado, I would like to invite my esteemed panelist, Dr. Amit Bhatt from Pune, Dr. Arun Chandrasekhar from Calicut, Dr. Jyoti Kode, Dr. Amit Avasthi, and Vijay Patil. Hi, Jyoti, it's good to see you. Yeah. So, Bao, yes, Bao Sahib is my colleague, and it's a pleasure to be able to moderate one of the people who has trained with you, Dr. Albina Nisar from Actrec, and Dr. Priyanka Samal from Bhubaneswar. So while, in the interest of time, while we could just get started, might take the permission. So we know that chimeric antigen receptor T cells and also the bispecific T cell therapy is an emergent treatment, and it involves genetically modifying the autologous or allogeneic stem cells in the case of CAR T cells. And also trying to generate antibodies, whether or not bispecific for the T cells and the cancer antigen. And these modified T cells attack the normal tissue cells and expressing similar proteins on their surfaces, leading to cross-reactivity. And also, this can also translate into adverse events for the patient, compromising sometimes the true benefits that the CAR T cells can provide. And though the results are substantial, but for those people who are dealing with CAR T cell therapy, we really struggle with the problems that are induced by the cellular therapies. And we know that the CAR T cells therapy has emerged in the treatment of pediatric B-cell acute lymphatic leukemia, non-Hodgkin's lymphoma, and to a certain chronic lymphatic leukemia, follicular lymphomas. And we know Tisacel, or the Eliana study for ALL was, Tisacel was approved in 2017, and we have the approval for relapsed refractory diffuse large B-cell lymphoma based on the data from the Juliet study. And we also have the availability of BiTEs in our for B-cell malignancies, and more promisingly for multiple myeloma. And these are basically the targets that are used or utilized for BiTE therapy, mainly the CD19, the CD20, the CLEC2A, WT1, and so on and so forth. And you have, based on the Zuma study and the Juliet study for non-Hodgkin's lymphoma, and of course the Eliana study for pediatric ALL patients, these have been FDA approved, and this is based basically on the very dramatic responses that one achieved in terms of having durable responses which are sustained beyond 6 months and over the period of 1 year. And that was distributed in most of the studies, like the Zuma study, the Juliet study, and also the Transcend NHL 001 study. So currently, we have almost 10 commercialized CAR T cells available presently in the market. And in India, we have, of course, Tata Memorial led the way in terms of in June 2021 when they initiated the study. And also now it's commercially available for treating leukemias and lymphomas, the CD19-directed CARs. And Narayana Health in Bangalore with Immunil also has their product for the CD19 CARs just now. And Origin and the trial which we are all working, and I'm also one of the PIs for the study, wherein we have the CAR T cells for myeloma study, and it's just completed the phase 1 study with very encouraging data. So the question is cellular therapy living up to its promise, or are we facing fundamental roadblocks? So I'll start with Vijay. So, is cellular therapy living up to its promise? What are your thoughts?
So I think for CLL, ALL and lymphoma, it's living up to the promise. In solid tumors, it has just started. So there are multiple challenges which need to be overcome in solid tumors. So for hematolymphoid tumors, yes, it's living up to the promise.
So, Bao, because you are with basically mainly with hematological malignancies, why do you think that with hematological, any thoughts on why hematological malignancies have been better equipped to be susceptible to this form of therapy?
Yeah. So I think we besides the beginner's advantage, we have clean targets for hematological malignancies. Inherently, these cells are susceptible to T cell attacks, and probably that's the reason what I can see as of now. And definitely they are living up to success. For example, we had in myeloma setup, we had novel agents where response rate were used to be in the range of 30-40%, but now we are seeing response rate crossing 80-90% for CAR T with good depth of response. So definitely they are living up to the hope we had.
Priyanka, your thoughts on?
So I'm basically a hemato-oncologist, so we have been using these BiTE cells as well as CAR T cells. Yes, definitely there was no hope for patients who were actually in the triple line or 5-line refractory, and still now actually transplant hold its place even in the CAR T failed patients, and it is the opposite way around also. The patients who have failed allo, they are also have an advantage of having a CAR T therapy. So there are definitely a lot of things to come up, but then the roadblocks in our setting is we opt for these options late, and second is affordability. So these are the two things, I think these are challenges for us.
But do you think this using it as an option, as a late thing, because most of the CAR T cell therapies have shown even benefits whether you're using it upfront? or whether you're using it later. So is that probably, is it really one of the
Sir, we
issues
have
that you
used
think?
for patients who are actually all refractory. So in those cases, the patients end up coming to us late, which is a challenge for us because patients who are in a better condition, they tolerate it well. As you know, the CRS and the ICANS, the toxicities are more when the patients have an advanced or a refractory disease.
Yeah.
So these are challenges which we are facing. But if it comes up to a second line or a earlier course, possibly it will be better for us as well as the patients to handle the complications as well as for them to get the benefit out of the therapy.
Arun, yeah.
So I do community oncology practice, so we have a team who does the CAR T-cell therapy. So the difference which I've seen CAR T-cell do right now, which makes me think is when a patient has the first line, when he has progression, I start counseling them. I start earlier. Before, maybe I used to do it much later, but then in the first refractory setting, I kind of tell them we have this option which is there. Do you need to wait? I don't think so, but think about it by the time we start a salvage regimen. And patients, they kind of become attuned to it. It takes a time, giving a costly therapy like this, it takes time for acceptance. The other issue of using CAR T-cell earlier, that was a problem with myeloma, where like some of the detractors who said with the CAR-Titude you had lifelong issues like Parkinsonism, which could be detrimental. So are we doing it too soon? So probably myeloma, I might stick to, probably 3 lines or 4 lines. But things like lymphoma, I think earlier is better.
Yeah. So, I get that point, but my point was that CAR T cells, irrespective of what is the phase of the refractory disease, still it seems to have its independent activity because the mechanism of action is completely different at the cellular level. So, Vijay, you have been, you've been focusing on solid tumors mainly for CAR T cells. What are the challenges actually that we are facing in solid tumors? Is it because of the heterogeneity of the antigen expression and it's not just one, or is it?
So there are multiple factors.
Yes.
The first factor is the trafficking factor. As opposed to liquid tumors where there is no trafficking issue. In solid tumors, if the tumor has a lot of collagen deposition, you don't know whether the CAR-T which has been given intravascularly is going to reach up to the tumor or not.
So you say that there is a lot of at the tumor-related deterrence to availability of the CAR-T cells for the target.
Correct. The second thing is, sir, the tumor milieu. A lot of tumor milieu in solid tumor is immunosuppressive.
Yes.
And to convert an immunosuppressive into a cold tumor into a hot tumor, you need to look at higher generations of CAR-T, like 4th or 5th generation, or you may have to add certain interleukin-2s or interferons.
So there should be a lot of manipulations that you need to actually to augment that. So in that bargain, are we going to encounter more of toxicity?
In that bargain, obviously, we are going to encounter more of a CRS and more of a ICANS. That is going to happen. Second is the selection of target. As Bahu rightly pointed out, we have clean targets in hematological tumors. We have 2 types of targets, like tumor-specific antigens or tumor-associated antigens. We have very few tumor-specific antigens. When you have tumor-associated antigens on target, but off-tumor toxicities actually do happen with, like for example, the CEA CAR-T programs in initial stages in 2 or 3 large academic institutes had to be closed not because of efficacy related, but because of high toxicity related, because of colitis, until we started having gated CAR-Ts for that matter. And the last is antigen escape, or tumor heterogeneity for the antigen. If you have an antigen escape or there are more than one antigens which are associated, then a single antigen associated CAR-Ts are unlikely to be effective in solid tumors.
So, Dr. Hari, I would like to add, what Priyanka and even Dr. Arun mentioned that earlier this option is applied because the longer the other treatments, the more the immunosuppressive microenvironment turns. So early they tap, okay, if they can go for CAR-T. Second thing, what Dr. Vijay mentioned, that, the antigen heterogeneity, like you may start with one CAR-T selecting a patient, but later on the antigen heterogeneity changes. And at the same time, you don't see that abscopal effect, like especially for solid tumors, which you see in radiotherapy or probably oncolytic viruses, that this particular in CAR-T, it may not be, because CAR-T will just reduce the tumor, but it will not release completely the antigen, which later on can go to metastasis, which are mainly seen in solid tumors. So these could be the reasons, some failures.
But also, just a thought, is it because of different antigen expressions at the metastatic site of the, the heterogeneity of the metastasis vis-a-vis the primary tumor?
Yes, sir. So for example, in breast cancer, we all are aware that when there is metastasis, we see a 33% discrepancy in the, so it becomes very difficult because which clone are you trying to target in that situation?
Okay.
So can I add?
Yeah, please, yes.
From a basic science point of view, I feel that there is already an established suppression of tumor microenvironment is, it's not really in the favor of CAR-T persistence and there is exhaustion. But I feel a lot of focus has always been on hematological malignancies because it was easy in terms of immunoengineering techniques. But now there's a shift and there are more molecular techniques being used to, because single target CAR-T is definitely not going to work. It has to be combinatorial, even if it's dual target or it's logic gated. For solid tumors, it has to be a combinatorial approach because the environment is completely different from hematological.
Yes, take your point, yes. Yeah.
Can I add to this? I think Dr. Bijay Patil has already added most of the point.
Yes.
Three major challenges the way I find in the solid cancer, why the success taking time. It has a dense environment, the microenvironment, extracellular environment is very dense. The second one is the tumor suppressive, because the PDL1 or CTLA4, this actually suppress. And what happens that CAR-T is exhausted over the time. And the major reason why it is much more successful in the hematological cancer is that we were lucky to find a marker, something like CD19. So it is super expressed compared to the normal cells, because you want a target which can only express mostly in the cancer cells compared to the healthy cells. And you don't want healthy cells to be, So I think now many such targets we identified, Claudin 18.2, one of the very beautiful example. And we need to identify many such markers which are predominantly present on the solid tumor cancer compared to the non-cancerous cells.
Thank you. Thank you for those pertinent points. Yeah. So I'll just come back to the solid tumor. Recently, the data was published on GI cancers and the large data. What is your take on that? And why? I mean, people are saying that 5 months. At 5 months, one of the longest sustenance of response is 5 months, but they were all relapsing still. Would you call that as basically a success? Or if you look at the way that patients are being treated with multiple lines of therapy, even a 2-month survival or progression-free survival with chemotherapy is considered a positive trial. So from that perspective, is it still something that has to be a...
So when we talk about success, it has to be with respect to what was that the baseline. The patients which were taken in that Claudin study were 63% were post 3 lines of therapy. In those patients, the expected life expectancy is below 4 to 5 months.
Yes.
And if you look at the results of any of the drugs which are used in 3rd line or 4th line, which are even FDA approved, the response rates are in the single digit. In those patients, if you are going to get response rate between 36 to 50% and a median PFS of 5.6 months, but the duration of response of 9 months, then it is going to be labeled as success. But I think more important than that, what is important is the new Chinese press release which came for the first clinical trial for Claudin 18.2, which had a head-to-head comparison of patient of gastric and gastroesophageal tumor post 3 lines of therapy, randomized either to Claudin 18.2 single infusion CAR-T or to paclitaxel or nivolumab or apatinib, and which proved that there was a PFS and an OS benefit. So that puts it into that, this is something which looks to be very promising.
So so the way that we probably need to think in the future is to bring it more into the front line rather than wait for more.
That may have to do with lymphocyte exhaustion also. The more the lines of chemotherapy you give, the more the lymphocyte exhaustion and depletion you get. And you must have seen, sir, when we harvest patient of multiple myeloma which are post 2 lines of therapy or post 4 lines of therapy, the total volumes which you have to take are more when they are post more lines of therapy.
That's right. So, so
what you mentioned about exhaustion.
Yes.
So
I wanted next question was for that actually, when limitations of the CAR-T cells and antigen escape and exhaustion. So would you like to?
So the exact exhaustion is what Dr. Vijay mentioned. too much of, we want the proliferation of these CAR T cells so that there is sufficient tumor kill. But the too much proliferation may put them into exhaustion. Now these exhaustion markers again are actually immune checkpoint markers. So somebody mentioned like again, yes. So first start with the first line as a CAR T, and then you come up with the immune checkpoint therapy which can, take care of the limited.
But one of the things that we have said about T-cell exhaustion is actually chronic antigen stimulation, and that is something which is happening after you infuse the CAR T-cells, and that is one of the reasons why you are talking about failure of CAR T-cells also.
Yeah.
So any thoughts on that? Yeah.
No, what is the heterogeneity is what? See, first initially you know something is working, but later on the tumor understands, yes, something is coming and they are trying to push the tumor off. Then they change the antigen repertoire on the surface. This is where the antigen heterogeneity comes into picture. This is what the rate, because later on it ends into failure there. Yeah.
And about T-cell exhaustion, which you said, what is the, I mean, is it different for hematological malignancies? I'm just putting a thought. Is it different from hematological malignancies?
So
because you see in myeloma, for example, you see responses that are lasting for 2 years, in fact. Yeah. So, but we don't see this kind of things for in cases of solid. But I think it is too early for us to say that. But we are still learning as far as solid tumors are concerned. But your thoughts, yes.
Yeah. So I feel that there's still a lot of data which is pending, and there is still a requirement of a lot of consolidation of all the data in the field of solid tumors especially. There's still no FDA approval for solid tumors, so it's very early to say, but because it's mostly patient-tailored, so I feel it has a lot to do with the immunometabolism of the patient also. It's not as simple as, just the suppressive tumor microenvironment and solid tumor profile, but a lot of it is not only relevant to the tumor, but also to the production process. So if you take care of these elements in the design of the CAR itself, which has been already, there are studies about that. In CD28, you just introduce a null mutation, it will be taken care of. Then during the in vitro expansion also, during your production process also, you can make alterations and you can change the way you add cytokines, different cocktail of cytokines. Those could also help in the antigen exhaustion and persistence of cells also. And then also the selection of the sample. So when you're taking an apheresis sample, the quality of the sample is the most important part of it, which is relevant for both hematological and solid malignancies, but more so for solid tumors because you're already working with a different profile of the cells.
So I just put forward some of these points. One of the antigen escape is that it decreases this, because of the chronic stimulation, I mean, it decreases the cytokine production, upregulation of inhibitory receptors, and also altered transcriptional profile. And some of the strategies, as we have described over here, is that you can actually engineer the CAR T cells with optimized signaling methods or target metabolic dysfunctions. Manipulating the metabolic profile of T cells is a way to look at it. And integrating CAR T cell therapy other than immunomodulatory strategies like cytokine therapy or immune checkpoints along with the CAR T cells may be to augment the CAR T cell activity, and also preconditioning the T cells, modifying the T cells ex vivo to enhance the anti-tumor activity is another way to go about it. So these are some of the other things that I felt it's really translates into one, as Vijay has very clearly and a colleague has mentioned about why the tumor microenvironment is so difficult in solid tumors sometimes, and also the CAR T cell trafficking on the infiltration, which is a challenge for solid tumors, and also the CAR T cell associated toxicities that makes it a little bit difficult, so that you are dealing with another problem after the CAR T cells, which is like chronic and many a time like an autoimmune disorder. So that's a problem. And so the tumor microenvironment components, the immune cells, the stromal cells, and the extracellular cells, all these have to be taken. I mean, hypoxia, isn't it? Hypoxia. Any thoughts on that? Yes.
So, in fact, I can tell you we have one paper which is coming on hypoxia from our center, and we have observed it even during production and manufacturing process, that it really remits the expansion profile and the type of the phenotype, which is eventually also important for the final efficacy of the product, in vivo product. So there are even as simple as lactate or pH during manufacturing. Those are also very vital, and we may miss the manufacturing elements, but the manufacturing elements also decide the type of the product and eventually the efficacy of the product. So it's multifactorial, I would say, actually.
Yes. Yeah.
So that's important, sir. Both the at peculiar cancers, peculiar strategies have to be adopted. Like for example, the results of hypoxia-resistant CEA CAR-T, they give both intravenous also, intraperitoneal also. The results of intraperitoneal CAR-T were much better than the intravenous CAR-T, just suggesting that if there is an access which is made local, it obviously would give same thing in GBM. So when you give intracavitary with Rickman catheter on Omaha reservoirs, the responses are much better than what you give with peripheral. So I'm sure as we evolve in solid tumors, we'll come across these situations just like the way surgeons do. We might be doing a scoring of the abdominal, that there are limited meds, and we will be using those scores to justify.
So when you say that, I did see that you had. It seems to be that the CNS CNS tumor seems to be amenable to CAR T cells manipulation.
So that's actually a surprise, because the amount of immunosuppressive environment which you have, but the results which are coming, they actually show that.
But actually, I'm not surprised, because when you look at the ALL data, for example, there was 100% availability of CAR T cells in the CSF, and there was not a single patient who had a CNS relapse following the CAR T cell therapy, or vice versa, patients who had CNS disease had complete clearance of the disease in the CNS. So that's probably one of the things. But I'm just looking at local administration of the CAR T cells into the CSF.
Correct, sir.
No, we have looked at using chemotherapy. Is it a different thing when you're looking at, you used to use wafers, for example, to administer? How is that different? I mean, that did not work really, isn't it?
So the mechanism of action, as you told, is completely different
Yes,
for chemotherapy versus.
Okay, get your point.
So I can tell you for the mechanism of, the infusion of the drug. So we're doing the studies on mice right now. So it's obviously going to be very difficult when you do it in, humans, but then there are established trials already which are being done internationally. So I think more important is the type of the product which is going to work, whether it's going to be CAR-T or a oncolytic or a combination of both.
Okay. Yeah.
Can I just add, like, beyond all these points also, like, is it I would like to ask Dr. Vijay and Dr. Arun also, that is it that patient accessibility, or, because it's a little bit costly therapy, you think patient population is not getting enrolled more, where you can actually show the success probably?
I think we are
is it the number?
We are still in the phase, I mean, other than, of course, in the space of myelomas, or for the non-Hodgkin's lymphomas, or for that ALL, I think as far as solid tumors, we are still very much early in it's not about this thing. People are still looking for the treatment. It's just that we have not still been able to decipher which is the best way to optimize and deliver this, and what are the antigens, etc.
Just before that, also one more point, the same lane, that after chemotherapy, that medical supervision, whatever is required, and after CAR-T therapy, medical supervision, how long or how easy or better all these clinicians you can also even.
Yeah, so that is a challenge. Bao, will you take up that? Because that is a challenge, because managing toxicities is incredibly expensive. I was there for the presentation when they first discussed about only using bispecific blinatumomab and inotuzumab for treatment of ALL. So they said that it is $250,000 for the treatment, and it is $350,000 for the supportive care. So that's where we are here. Yeah.
So definitely, but I think, sir, we have moved a long way. Now with the cellular therapies, including CAR and BiTEs, we are on the same page, where we are same set of toxicities. And with our background of managing similar cytokine release related phenomena, be it interleukin therapy, be it post-transplant phenomena, I think we are better geared to handle these things. But definitely one has to be thoughtful. One has to be anticipate these toxicities. One has to be ready with the management plan, like you would have a scoring system, CRS, ICANS, etc. What is grade 1, what is persistent grade 1, and it would be kind of monitored very closely. So definitely with the kind of approach of very meticulous charting, monitoring, early intervention, we can manage all these toxicities. I think the thing which we are still not able to overcome is the cytopenias, the hypogammaglobulinemia which we see with CD19, and the infection, which still remains a problem, because once in a while, typically a person who has low counts to start with is likely to develop all these complications, and that's the area where we are facing issues.
Priyanka, yes.
Sir, in hematological malignancies, when we are using it, possibly CAR T is somewhere we are trying to go away with the transplant. So almost when you counsel a patient regarding transplant, this is the transplant cost, and then post your therapy, this is the cost you have to come across for the next 6 months or 1 year. That is how the counseling for the CAR T as well as the BITE, especially at the outset only you say the patient so that he is ready for and accepts whatever the outcomes are or whatever the toxicities are.
So are we currently well equipped to manage CRS? How you've been
experience from the humanized CD19 CAR, I think it has been easy to handle the CRS.
Are there any infusion-related problems that you face? I think that should be more with BITEs rather than with CARs, because CARs are autologous.
Correct. But I think, sir, one thing to keep in mind is every CAR product is a different product. The moment we switch from one target to another target, and also the other variables of CAR construct would be important. Second thing is, yes, sir.
Just a question regarding the infusion. One of the issues, one of the other centers, my colleague said, was the flow. It got stuck in the tubing. It was, I don't know if it's common, but the team from from Mumbai had told that this could happen, where the infusion rate was going very slowly and they had to take it out and then inject it.
So we routinely give the CARs as an IV push. We never give it as an infusion. We just give it. In fact, we put the bag and then take out the thing and then push it into the patient. Yeah. You had to say something.
No, I actually was saying that there is an established mechanism, and if there is one product which has faced an issue, so that's just one. Okay. So. This, of course, is not a production issue, because they will always have a supplementary bag along with that, or an overwrap bag which should take care of it if you're talking about that. But during infusion also, I think there's already an established protocol for infusion. So I was talking more about the production.
Okay. So is there anything? Yeah.
One of the things you must remember that many other times they may have port or all. You always use a big tubing, big channel, and what Sir said is correct. It is better that you withdraw it from the syringe and then push it. Withdraw from the syringe and push it so that it gets finished faster. But I think, sir, one of the things which need to congratulate the IIT team also, the Immuno-Oncology product, I at least have not come across much amount of CRS or ICANS. It is pretty smooth to give, and even if the patient has a few compromised organ functions, still it's easy to give. Now, your thoughts?
Yeah, so definitely in terms of safety, this is much better than other CD19 approved CAR. And we have not seen grade 3 or more CRS, etc.
Okay. Yeah. So I just put up the physiology. In the interest of time, I think we are running late. How much more time do we have? 2 minutes. Okay. So I think I will not go through the slides. So I'll just rush through the grades of. Is there any way to predict CRS, and are we now well equipped to manage CRS? Because that is something that is. With the how much has tocilizumab actually helped in managing CRS? Do you get those kind of grade 4 CRS? Have you ever encountered that?
Yeah, so we have to not get there. Basically, if you get there, it's a very difficult situation, and basically you don't want to go to a situation where you have to use steroids. So definitely, I think the starting point, besides all these things, tocilizumab with a persistent grade 1 CRS. If you start early in time, you are going to prevent that cascading and prevent all these things. So acting early is very important, and once you move beyond grade 3, grade 4 situation is difficult, and similarly is for ICANS.
Yeah, and how have you ever encountered ICANS in patients? I personally haven't so far.
Not yet.
And in solid tumors?
You do encounter ICANS. In fact, what I learned about ICANS is we do the ICANS scoring, but one of the first things the patient tells us is unable to open its mobile.
Yeah.
Because of the complicated designs which the smartphones require, that is one of the first things the patient is going to do.
So one of the first thing also is the handwriting.
Sir, but handwriting, when the sister goes and asking for handwriting, that time he's going to write. The patient, the first thing he does in isolation, he says good morning to his relatives. So if he cannot open the mobile, that's the first indication that he's developing an ICANN.
Yeah.
No, sir, we didn't encounter any ICANNs, but yes, cytopenias prolonged, that is.
Oh, that is something which even we have struggled with actually, with the cytopenias, which is what I'm coming to. I don't have time because it's we are running this thing. But overall, when we look at the kind of the data that has been published in terms of severe CRS is more feature of the CAR T cells rather than the TCEs or the T cell engager therapies, which is the BiTEs and etc. But if you look at different systems, probably it's the maximum probably for leukemias where you get CRS much more. Probably it's got to do with the volume of the disease. Is that the reason? Any thoughts on that?
So definitely. Prior to infusion, disease burden, etc., would matter for CRS.
So we don't encounter so much in solid tumors, which I mean, in solid tumors, when I say in hematological malignancy, the lymphomas actually, and very negligible as far as the immediate CRSs and the ICANNs with the BiTEs. But the main problem that we encounter with BiTEs is infection and also HLH. How many times have you encountered HLH?
So, sir, we had a couple of cases of difficult to handle HLH. Definitely, one should be thinking of HLH when you have cytopenias and your other features of hemophagocytosis. And again, aggressive management is...
Would you like to take up this next question? Because the new concept of immune effector cell hematotoxicity.
Yes, sir. These are also, you see, they have classified as 30 days, 30 to 90 days, and then late effects. So this can prolong right from the very beginning when it is because of the lymphodepletion. Because of that, you find some amount of cytopenias, but usually resolves by day 30, day 60. Then later on, whichever comes up, that is mostly because of the B-cell aplasia, and we keep on monitoring regarding that. Difficult to handle, but with the growth factor supports, we continue with the cytopenias.
Okay, I get it. And how many times have you had to use immunoglobulins for our patients for treatment? Regularly. Is it more for the bispecifics or more for the CAR?
Equally for both.
Equally for both, yeah. Because in my experience, it has been more for bispecifics rather than for the CAR T cells, yeah. So I think these are the sequence of infections. Just give me just one more minute, yeah. So expect the timing of reactions that are there. This is the graph that we see, and the infections are something that we encounter for a long period of time, and even beyond 1 year. And many a time, my colleagues, at least in the West, said that you tend to lose patients in myeloma because of infections rather than because of the disease itself. Especially that's true with the bispecifics. So with that, I conclude. I thank my panelists, and I hope it was a useful discussion. It was something that I put together.
Thank you, sir.
Thank you very much.
Thank you very much, Dr. Hari.
Thank you, Manoj.
We would like to invite all the chairpersons to kindly come on the dais to take a photograph. Thank you so much. Now we come to the last talk of the first session before heading to the tea break. This is a sponsored session by Immuno-Act, and I would like to call Mr. Karan Gera for the topic From the bench, factors involved in autologous CAR-T manufacturing and emerging strategies. Please, sir.
Well, good morning, everybody. It seems that all the esteemed speakers and panelists that have come before me have pretty much made my job very easy here today. We've discussed allogeneic CARs, we've discussed autologous CARs, we've discussed CARs in solid tumors, we've discussed oncolytic viruses. But when it comes to the real world, when it comes to the now when commercialized applicability of CAR T-cell therapies and their manufacturing in the real world, especially when we're talking about autologous therapies, there are several factors that come into play. And quite often, more often than not, and in fact quite significantly, these can impact outcomes. So, how are conventional immunotherapies manufactured? Conventional immunotherapies, you usually manufacture them on a Pichia pastoris or a CHO system, or sometimes even in bacterial systems. Beautiful thing about these is you could just scale them up. You know, you could just have them in a single bioreactor, and then you had new strategies which involved continuous perfusion-based manufacturing, which allowed your scales to linearly increase. And that gave people a huge advantage because with the benefit of scale, you could bring down cost, and that gave you a lot more wherewithal in order to provide access. But the situation in CAR-Ts, the situation with gene-modified cell therapies as a whole, especially with the autologous kind, is notably different. So, three major components. There are two components which are off the shelf, and then there's one component which is personalized. So, off the shelf, you have your retroviruses, which are either your gamma retroviruses or your lentiviruses. And they've been the most preferred form of delivering your payload, delivering your genetic payload into the cells. So, how do you manufacture them? Now, ideally, you would want to go to where conventional immunotherapies are, where you have one beautiful looking cell line that does everything you want it to do, that you can grow it straight from the start, and it'll give you your gene of interest, and it'll manufacture to scale, and it'll give you the recombinant protein with the titers that you want. But that's not always the case with CAR-Ts. Until you move to producer or packaging cell line systems, wherein your retroviral vector or your lentiviral vector is able to manufacture continuously based on what your needs are, you'd still need to transfect your HEC platform cells every single time, and that's transient transfection. So with lentiviruses, that involves a GAG-Pol plasmid, an envelope plasmid, LTR plasmid, as well as your gene of interest. Now, vector manufacturing is not as challenging as it used to be 15 years ago, but it still has a lot of hurdles. And preferably, it still proceeds in adherent platforms, in fixed bed platforms. Now, the problem with fixed bed platforms is that they're not as easy to manage and scale as, say, a suspension platform. So, what you typically have right now is you'd seed, and then you'd go to the next stage, which is cell stacks, and then eventually, you know, you do a nuclease treatment, you clarify the harvest, then you'll go through concentration and diafiltration, you'll try and maximize the titer that you get, the purity that you need, go through sterile filtration, and then aliquot into your fill and finish. The goal, scaling beyond this, is to move to fixed bed bioreactors. Now, when you move to fixed bed bioreactors, costs become a challenge because some of these systems, while efficient, are still quite expensive, especially in the Indian context. So balancing titer, purity, and the consistency of your viral vector production also does factor in to the scalability of the platform itself. Now, would it be good to move to a suspension platform? Of course. But modifying HEC293 T cells to become suspension capable and to retain their high titership in a suspension culture is not that easy. But these are the processes that are involved there. Hopefully, with time, there will be a lot more ubiquity, as we have seen with recombinant proteins, in the availability of packaging cell lines, wherein you only need to transfect your gene of interest, or producer cell lines where you would just straight away, thaw and start manufacturing, and the scalability of vector platforms themselves. But that's not what we're discussing today, because these are still the off-the-shelf components. So what's really interesting? What's really important? What's really important is the personalized component. So when you talk about hematological malignancies, which are currently being treated with CAR-Ts, you're talking about relapsed refractory multiple myeloma, which is by far the worst. Many patients have had several lines of treatment. They've had DARA, they've had VRD, they've had 2-3 kinds of IMiDs, they've had 1 or 2 proteasome inhibitors, they may have failed an autologous transplant. Their T cells have been through hell and back again. And somehow you still need them to manufacture CAR-Ts. That's a bit of a challenge, isn't it? The situation is even worse for lymphomas, when these T lymphocytes are not only supposed to be active in the peripheral blood, but they're also supposed to reach the lymph nodes, and they pretty much get exhausted there. Now, your typical process used to be a lot longer. However, in due course, and even for us, our manufacturing process for autologous T cells has become shorter. We have progressed from a 19 to 20-day manufacturing plus release timeline to now a 9-day manufacturing timeline. And there are many even commercial scale CAR-Ts, like for example, Axicell or Brexucell for that matter, which have reduced that manufacturing timeline to 8 and 7 days and keep the remaining for release. So reducing the manufacturing timeline is very essential, but it does depend on a bunch of factors, because you want to choose the sweet spot for when you're able to activate the right potential of T cells, for when you're able to transduce them, how long you're able to transduce them, when you feel that the dose that you have established is going to be adequate enough, which, by the way, is different for different CAR-Ts. Very different for different CAR-Ts based on their construct design and based on some other factors. So how do these factors interplay? And not just that. You're done manufacturing, but you got 11 quality release tests to be done. And you don't want them to take too long, while at the same time, you want them to be performed in time for the drug to be made available for the patient. Because remember, most of these patients are going to be undergoing some form of bridging therapy. If they can avoid the bridging therapy and they're already in low disease burden, it would be preferential for them to reach the treatment soon. But that's not always the case, especially when you're going to scale. So, what are the actual factors that affect CAR T-cell therapy manufacturing? One is the disease burden, prior treatments. The higher your disease burden, the longer the duration of your disease burden in terms of these tumors, the more there is a chance that the T cells that you're going to get are a lot more exhausted. Prior treatments, if you've had severely lymphotoxic therapies, which have a very long half-life, like say for example, bendamustine, every CAR-T manufacturer's nightmare, then you may get the right count of T cells, but their transducibility with vectors would be low. And their ability to expand eventually would also unfortunately be subpar. But you can't expect physicians to just, do away with drugs such as bendamustine, especially when earlier in their line of treatment, a patient was rendered transplant ineligible and they had to receive a BR regimen. So what do you do then? Also, T cells are distributed in different subsets of maturation. So if you just look at the CD4 and CD8 T cells, you're going to see them across a whole subset of maturation, from T naive to T stem cell memory to T central memory to T effector memory and to T effector T cells. And then the ones we all wish weren't around. That awkward guy in the room, we call them regulatory T cells. We don't want them around in our CAR-T product. They could do more harm than good. You're good in the regular environment, but we don't need you in this context. It's like that. So all of these factors can actually impact what your CAR-T manufacturing profile looks like. Ideally, with the T-cell maturation subset, you want a good balance of all. You do not want only naive cells because you want them to start acting immediately. You don't want only effector cells because you want them to persist for longer. So what their proportion is in your received leukapheresis subset will also impact the quality, durability, persistence, and overall activity of the product. Apart from that, there are process-related caveats, whether or not you're using a manual system, whether or not you're using a semi-manual system or semi-automated system, or whether or not you're using a fully automated system. What are the interventions that you're doing on each stage? Are you using beads? Traditionally, people use antibody-coated magnetic beads. Problem is, it's not until you wash out the beads that you can actually expect the T cells to expand. So now people are moving to, colloidal polymeric nanomatrices because they can be soluble. They can do the job and then allow you to be able to expand your cells in order to reach the right dose and then get them out there and get them ready for formulation and for release testing so that you can get them to the patient soon. Then, process components like cytokine cocktails. Traditionally, you use IL-2, but there have been studies that have shown that by using a good combination of IL-7 and IL-15 incubating, you'll get a better subset of T cells. You'll improve their robustness in terms of expansion and in terms of activity. When we first started with CAR T-cell therapy development all those years ago, we didn't realize these things. We didn't even define what CRS and ICANS were. But these technologies are now improving better. What also is really important in the process development aspects is the time duration of each and every one of your events. What is the ideal time to select and activate your T cells? Are you activating them for too long? If so, will they reach their exhaustion profile and will their in vivo expansion be limited? Are you keeping them transduced for just the right amount of time? Are they hitting that transduction efficiency sweet spot? Not too heavily transduced, because that could provide undesirous effects in the cells and in the body later on, and it may increase the chance of undesirable insertions, or not too low in its transduction that it does not express enough CAR for it to do the action that it was meant to do. You need to have the right balance there as well. The others, how quickly do you do the bead separation? In the process of bead separation, are you losing T cells because of clusters? These are all the processes that get impacted, and it's all that forms a part of process optimization, which is why autologous CAR T-cell therapy manufacturing is not just a science, it is artistic prowess. And finally, the vector itself. Now, it doesn't necessarily mean that a high multiplicity of infection means that you're going to get a more potent vector. Sometimes, based on the kind of cell that you're trying to target, a high multiplicity of infection may not be desirous, or a low multiplicity of infection may not be desirous. You again need the right balance. It's all about finding that balance. Of course, if you don't just have those methods, it's also preferable to extract your T lymphocytes early, like this study, which actually showed that an early extraction of T lymphocytes led to the better overall survival of patients who had LBCLs. The idea was that these are a better distributed subset of T cells. So what's coming next? What's coming next is T-charge, is faster manufacturing, is preserving of stemness, is the decrease in the duration of using lower incubation time of beads, is using soluble beads so that you can keep your T cells naive, you can keep some in the effector stage, you can keep some in the memory stage, and then immediately get them moving in order to go to release testing. In the case of Novartis, what they're doing is they're doing transduction and selection and activation on the same day. What did that lead to? It led to similar responses to Tisacel, but better persistence. So this is just an initial readout of their phase 1A, 1B trial, which is now in a pivotal trial. Ultimately, we know that faster manufacturing is going to benefit us. But what we will also need is better automations in terms of manufacturing, better interventions in terms of the speed and robustness of quality testing, lower time for release testing, better parameters in order to adjudge release testing better, meaning do we need a 9-day sterility test? Perhaps we don't. Perhaps we can use it better and more robust 5-day sterility test. Examples like those. Of course, there are also enhancements to manufacturing itself, such as GOT2 inhibition. So Sotio Biotech is working on a GPC3-directed CAR, and they found that by inhibiting GOT2 expression, they improve T-cell persistence in vivo. Again, PD-1 inhibition, we all know the devil on the T cell, and you want to stop the devil on the T cell. So PD-1 knockdown in your T cells before they are infused in the patient could be advantageous in terms of expanding T cells and improving their persistence. And finally, removing regulatory T cells. So it's all about finding that balance, Manual versus automated, what's going to work cost effectively? Accelerated versus full duration manufacturing, because you can reduce the T cell dosage in accelerated manufacturing, but the problem is it can also lead to T cells which may not be immediately acting and which may take a little bit more time to act. Process automation, it'll reduce your personnel, but it could increase your capex costs to begin with. And centralized versus point of care manufacturing, how long can you proceed with centralized, and at what tipping point will it be better to move to point of care? Finding the right balance is the way. We'll get there because we have to. Cell and gene therapy is the future, and that future is now. So we'll do all we can if we can do it together. Thank you very much. Bye-bye. Yes.
Yeah, I had a very short question. What do you feel is the most challenging aspect when you are manufacturing the product in your experience with Immuno-Act, and what do you want to advise the collection centers, the POC centers for your better product manufacturing?
Extract lymphocytes early. Extract them early. Have the patient counseled if they are at high risk, that if you are going to be at high risk of relapse, you may probably want to take that investment and exercise an apheresis early. There is a risk that we may not be able to expand later, but it's a risk worth taking. An earlier manufactured product is definitely a better and more persisting product, especially for the NHLs, especially for those hard to reach DLBCLs.
Thanks, Karan.
Ooh, didn't know there'd be more questions, but hey, I'm always open to them.
So my question is regarding short-lived and long-lived CAR-Ts. So this concept of using RNA instead of lentivirus for transient transfection and expressing a CAR construct for a short term, in terms of their efficacy, are you familiar with it?
Well, you can use mRNAs in order to, express a CAR. It's not the expression of the CAR itself which is short-term. It's whether the T cell will live long enough.
Correct.
So what really depends on whether you can preserve the longevity of the T cells, and that means that you need to transduce their younger versions as well, or convert them into younger versions. So Dr. Albina was talking about a paper on hypoxia, which involves improving the stemness of T cells by keeping pulse hypoxia in your manufacturing process. And then when you transduce with either a virus or even an mRNA sequence, those T cells will then bank into your thymus memory, and they'll later be able to expand. So it's not a question of what agent you use to transduce, it's more a question of what to do once you've transduced.
Are there any trials with those short-lived, transient, transfected?
You mean by transducing T cells and then eliminating the expression of the CAR?
Yeah, right.
So there is something called a suicide switch, which is sometimes included in a sequence. Mostly these involve an apoptotic Cas9 switch that can be used. Basically, you induce that switch as a part of your construct, and when you feel that you need to inhibit its activity, you give a small molecule that will initiate. Those trials are underway.
So I would like you all to please proceed for a short tea break. It's for 10 minutes. I hope we all come back energized and, really encourage our speakers by clapping a lot, Okay, thank you. to call upon the stage the chairperson, Dr. Vamshi Krishna sir from Hyderabad, Professor Nilakshi Mahanto ma'am from Guwahati, and Dr. Prashant Kumar Parida sir from Bhubaneswar.
So, a very warm good morning to all here. We are starting our symposium, and the topic today is evolution in advanced urothelial carcinoma treatment to achieve maximum survival benefits in patients. The speaker for this topic is Dr. Vineet Talwar from the Department of Medical Oncology in Delhi. I request Dr. Vineet Talwar to come and take the stage.
Thank you, madam, for the kind words. And at the outset, I thank the organizers for giving me this opportunity to speak on a topic which is a burning topic nowadays. And this is a bourgeois topic in the sense that it has been evolving in the last about 3 to 4 years. It remained static for about 1 or 2 decades, after which it has now become a hot point. So these are the selection therapies for the advanced bladder cancer patients. You find that the percentage of patients not receiving first-line treatment, and you have countries like England, Denmark, and Germany also. So I would be talking about the efficacy, safety, and the patient eligibility in the selection of therapies in these patients, and also some of the real-world evidences whenever we treat these patients. Basically, I would be dealing with the first-line treatment in the urothelial arena, whether it is the platinum-eligible or platinum-ineligible patients. And I would be focusing on basically 3 trials. That would be the EV-Pembro, the GEM-CIS followed by switch maintenance, and the GEM-CIS-NIVO trial. So you have this current landscape where you have patients who initially take chemotherapy and then they are switched over after the 4th or the 6th cycle to maintenance Ivolumab when there is no progression of disease. These are in the platinum eligible conditions. And we also find that up to 90% of patients who receive the first-line platinum-based chemotherapy achieve a response or a disease control whenever you give these patients Ivolumab maintenance. And high response rates are observed in patients who are platinum eligible and treated with first-line platinum bladder cancers. And that necessitates the need for Ivolumab maintenance. And that also is an important therapeutic option to address the challenge of chemotherapy resistance in these patients. So with that preamble, I will discuss the Javelin 100 trial, which was in unresectable first-line metastatic urothelial cancer patients who initially got either CIS or GEM or carbo or GEM. This was a 1:1 randomization. And after which they were given Ivolumab. This was 10 milligram per kg, IV every 2 weekly plus best supportive care or best supportive care only. The primary endpoint was overall survival, and the secondary endpoints were progression-free survival and also the quality of life indices. And you find that it met its primary point. It was 23.8 months versus 15 months as far as the OS was concerned. And 5 months versus 2 months, that was the progression-free survival. And now we have the long-term follow-up also. We have a 29.7 month overall survival if you see from the start of the treatment. That is, if you combine the time when you take the switch and the time prior to that. So this had a lot of advantages in the sense that either you could give cisplatin or you could give carboplatin. There are a lot of patients who are cisplatin ineligible, so those patients could be given carboplatin. And the duration of the platinum-based treatment could be 3 cycles, 4 cycles, or 6 cycles. All of them had good responses. And the patients who had a complete response, partial response, or a stable disease, they could be taken under the ambit of switch maintenance. And you can see the treatment-free interval length. Either you could start at 4 weeks, or 8 weeks, or up till 10 weeks. And these are the safety outcomes. They were almost similar to what it is seen in other IO drugs. And this is the long-term conditional survival with the AE probability. You can see patients after 1 year and after 2 years of avelumab treatment. They were about 34% after 1 year and 20% after 2 years of avelumab treatment. So now switching gears after this switch trial, the second trial which I am discussing is the GEM-CIS and nivolumab followed by the maintenance nivolumab. And here you find this is a 1:1 randomization, again upfront patients in which chemo is combined with immunotherapy and continued till toxicity or intolerance. A word prior to this is that there were 3 trials earlier in which chemotherapy was combined with immunotherapy drugs, and all 3 were negative trials. So this was the first trial after the 3 negative trials where you could get a survival benefit. And here you find that there is an overall survival benefit of about 3 months with a very slight PFS benefit. And this is about 2.8 months OS versus 3 months progression-free survival. Actually, it was a post-hoc analysis of a larger study in which this was taken as a survival outcome. And these are again the ORR rates, which are about 57.6. And 10% patients had PD as the best overall response. So question comes is there a benefit of upfront IO addition or not? And in the subsequent therapies, a limited number of patients were given maintenance avelumab in the control arm. That is also a point of contention in this study. So 74% of patients with GEM-CIS carbo arm received subsequent ICs in the JB100 arm, where in this case it was just about 10%. The third trial which I'm going to discuss is the EV-Pembro trial, which is the new kid on the block, and it has taken the whole urothelial landscape by storm. And this is basically a phase 3 open label study in which you had previously untreated patients. Now this you can use in cis-eligible, carbo-eligible, or platinum-ineligible patients also. And here you had EV, that is enfortumab vedotin with the pembrolizumab versus GEM-CIS. This was 6 cycles, or GEM-Carbo, which was 6 cycles. The thing to note here is that after the chemo in the control arm, only 30% of patients received avelumab as maintenance. That again could be a point of contention in this study. And this was continued until progressive disease or unacceptable toxicity, where Pembro was given about 35 cycles and EV was continued henceforth. The dual endpoints were progression-free survival and the overall survival, and the key secondary endpoints were the toxicity and the quality of life indices. And you find that it met its primary endpoints. The OS was a fantastic 31.5 versus 16 months. And this got a standing ovation in ESMO, and the progression-free survival was 12.5 months versus 6 months. So you can say that the outcomes were doubled when you give this combination against the pitted standard, which was conventional chemotherapy. So this was fraught with its own caveats or own side effects, the treatment-related adverse events. On the left-hand side, you can see that 97% of patients had adverse events and 35% patients had discontinuation. So you started with 100 patients, 65 could only proceed. And the interruptions were about 70%. So it's not a very easy drug to handle. And dose reduction was in about 40% of patients, and 4 patients died during this trial. So roughly about 40% of patients, either they stopped the treatment or they were discontinued, or there was about 70% interruptions with the dosages which were being managed. So this was there when this trial was there. And another important thing to note was that those patients who had skin toxicities or side effects, 40% of the patients had residual grade 2 skin reactions, even after the subsidence of the immediate adverse events. And the other thing is that 50% of patients had residual grade 2 neuropathy. Now we can very nonchalantly say that patients can have grade 1 or grade 2 neuropathy, but you can imagine an elderly individual with a grade 2 neuropathy, inability to write, and this is residual neuropathy. This is not the acute phase. This is after the acute phase. And as I told you, about 60 to 70% patients had grade 2 or grade 3 adverse events, and about 40% had grade 3. So this is the current landscape. If you have a platinum eligible patient, you can give EV Pembro, or you can do the switch maintenance with Avelumab. If you have a patient who is cisplatin ineligible, in these patients you can use EV Pembro, or you can use Pembro alone or Atezo. Patient eligibility remains the key point in these studies. If you talk of the switch maintenance, about 90% patients could be enrolled. If you talk of the EV Pembro study, 80%. But if you talk of the GEM-CIS, Nivo followed by Nivo maintenance, you find that only 50% patients were eligible. So I did not want to scare you with the side effects of EV Pembro. Suffice to say that in our clinical practice, how we can fine-tune and sift out those patients who can be eligible for the EV Pembro combination, because that is the combination to look on where you have almost doubling of the OS and the PFS. So this was proposed by Dr. Enrique Grande in an editorial in EAU, where if you have a patient who's not in a very good performance status, if the hemoglobin is more than, the glycosylated hemoglobin is more than 8, creatinine clearance less than 45, any corneal or retinal abnormality, because with the EV you find most of the patients have dry corneas, and we give some eye lubricants prior to the initiation of therapy. And if you have grade 2 or more sensory motor neuropathy, these patients might not benefit qualitatively, although quantitatively they may benefit. So these might not be given the benefit of EV Pembro. So they should be fit as far as these criteria are concerned, and they can be conveniently taken over by the switch protocol. We have real-world evidences where we can learn how the control and the real-world fared, and they are available across the world. You have United States, Europe, Asia Pacific, and you find that the progression-free survival and the OS were consistent in the RWE studies and the registration trial as far as the switch maintenance was concerned. And you can see that second-line EV achieved an overall survival of 40.8 months. So if you give the switch initially and after that you give EV, you get 40 months. If you give EV alone, EV and Pembro alone, you get about 30.1 months. If you give switch maintenance, you have 28 months. If you give GEM, CARB, or GEM, CIS, and Nivo, you get about 26 months. So that is the situation which is there nowadays. And in EV Pembro, the real world studies were there, but unfortunately they could not compare with the registration trial. There were differences in the overall response rates in the 12 months PFS and OFS, unlike the switch study. So this is a snapshot where you can see, and the thing I would like you to see is the grade 3+ adverse events, that is the third from the last, and the discontinuation rates. You find that although that EV Pembro data is a fantastic data, there are a lot of discontinuations and grade 3+ adverse events. So this is where the EVITA criteria, which I alluded to you, that can be used, and you can choose out patients who are going to actually benefit from EV Pembro. Otherwise, you can give them switch maintenance. Now the question is, I'm talking only of switch maintenance and EV Pembro. How does Nevo-Gem-Cis fare in this? Those patients who have got a high lymph nodal burden or lymph node only disease, these are the ones which can be taken up under the ambit of Gem-Cis and Nevo upfront. And these are the median overall survival trials, which I have just now told. You have 40 if you combine line 1 and line 2 with EV at the end. Then you have about 30-32 months where you have EV Pembro. You have about 30 months when you have the Javelin switch. And if you have about 26 months if you have Gem-Cis and Nevo. So I'll conclude here that in the rapidly evolving landscape, it is important to consider all treatment options to optimize the treatment and get a qualitative benefit from the treatment modalities which are available as of now. And it is a bourgeois branch, meaning thereby that every day you're getting newer and newer trials and you're getting better and more handsome outcomes, so as to say. But then you have to see qualitatively and quantitatively and match them. Avelumab maintenance can be tailored according to the practical considerations. Due to the toxicity profile, significant proportion of patients may not be suitable for EV Pembro. So you have the EVITA criteria where you can find out which patients will actually benefit. And first line treatment impacts options for second line choice. Meaning thereby if you lump everything in the first line, you are left with sweet nothing in the second line. There you have to go again back to the conventional chemotherapy drugs. And there are trials to say that if you have given immunotherapy in the first line, giving it in the second line may be detrimental, so as to say. So these are all the things which you have to consider whenever you embark on treatment with the metastatic urothelial cancer patients. I thank you for your patient hearing.
Thank you, sir, for the excellent deliberation on the therapeutic evolution of advanced urothelial carcinoma.
Sir, we would like to request to take all the questions at the end of the session, so
yes.
we can continue.
Thank you. So the key takeaways from his talk is the standard of care platinum gemcitabine response may not be durable. So by adding IO drugs like avelumab, we can have better long-term outcome. And IO drugs like nivolumab can be added to the standard of care cisplatin gemcitabine, which has improved OS and PFS, but we have to wait for the long-term data. And even platinum ineligible patient, we have options of EV plus Pembro, the chemo-free regimen, but we have to define who are the candidates for EV ineligibility. So these are the key takeaways. We can go to our next session, and next topic is lessons learned from combination of IO in GU cancer, and our next speaker is Dr. Amit Joshi from Mumbai.
Thank you, organizers, for this opportunity. So we'll quickly review the immunotherapy, especially the dual immunotherapy in the current era in GU oncology. So I will just start with 1 or 2 slides of basic of the how the T cell gets activated or deactivated in the normal cell and the body. So ultimately, it's the T cell exhaustion or inactivity by the combination of PD-L1 and PD-1, which makes the T cell inactive against the tumor cell. And this newer PD-1 inhibitor and PD-L1 inhibitor makes the T cell active, and that ultimately leads to, and this phenomena happens at the tumor cell level. So there are two axes of the T cell activation. One happens at the tumor cell, and the other axis happens at the lymph node. So the CTLA-4 axis, which seems to be more active at the lymph node level, and because of the inhibition of the CTLA-4 by the CTLA-4 antibody, this T cell remains activated. So in a tumor cell, both the PD1, PD-L1 pathway and the CTLA-4-1 pathway both makes the T cell inactive against the tumor. So if we simultaneously block both the PD1, PD-L1 axis and the CTLA-4 axis, the overall benefit or the response of the immune system may be much better in tackling the tumor, which will give the whatever the survival benefit we are seeing with the immunotherapy. So instead of combining one pathway of the PD1, PD-L1, if we combine both the anti-PD1, PD-L1 with the anti-CTLA-4, probably the response will be better. And that what the lessons which we have learned over a period of time. So when we see the combination of immunotherapy, there are some trials which are positive trial, some are 1 or 2 trials are negative trial, and there is some trial which is phase 2 data and which has shown the way how the immunotherapy will be used in future. Since the melanoma and the renal cell carcinoma are the 2 prototype of the tumor which are immune derived, most of the work in a immunotherapy milieu is done in these 2 tumors, that is the renal cell carcinoma and few in the urothelial in a urological malignancy. So first discuss about the role of the combination immunotherapy in a renal cell carcinoma. Just before starting, I just want to show the highlight that with the use of the tyrosine kinase inhibitor, the VEGF TKI, the overall survival in BEST series is close to around 2.5 to 3 months. So when the patient doesn't take the immunotherapy, the OS is close to 2.5 to 3 years. So that is the benchmark from where the immunotherapy will improve this survival. So 2 to 2.5 years the benchmark for the VEGF TKI, that is sunitinib, pazopanib, whatever we used in that scenario. And from this, when we plan the immunotherapy or plan the treatment for RCC, all medical oncologists are aware, we divide the patient into 3 groups with the IMDC criteria, depending upon various factors, and then as per the factor, if it is 0, it is favorable, intermediate and poor. And if you see the survival for a patient who falls into the poor risk is close to only 8 months. So the overall survival for a poor risk RCC in a TKI era, VEGF TKI is only close to, median OS is close to 8 months. So then we will know how the immunotherapy or how it is really have improved the survival. So positive trial which has stood test of time, and we all know that this percentage has stood test of time. So this is the Checkmate 214 is the most important trial in terms of RCC which has stood the test of time. So this trial, the long-term data of close to 99-month follow-up data was presented. And this was presented in ASCO 2024, the 90-month follow-up data. So if any trial is or new drug is presenting the data 90 months, that means it has stood the test of time and it has passed the test of time. So it's a 90-month follow-up data, standard treatment design, patient of advanced RCC, clear cell RCC, IMDC risk, all 3 groups were taken, and then they divided into 2 groups. One group received the sunitinib, which is the VEGF TKI, and they compared the combination of nivolumab plus ipilimumab. Nivolumab is the PD-1 inhibitor and ipilimumab is the CTLA-4. So the combination of a PD-1 inhibitor with the CTLA-4 inhibitor versus the VEGF TKI. And this trial and the final data, the 8-year follow-up data. So if you see the overall survival in the all the 3 risk group, the median OS is close to 52 months. So close to 53 months in the ipilimumab arm compared to the only 37. So there is a clear-cut improvement of a median OS of 20 months in patients who receive the combination of IO plus IO. When we further divide the intermediate and poor risk, which we saw that the median OS was close to only 8 months in the TKI era, the median OS in the Nivo-IP arm was close to 48 months. So there is a clear-cut improvement, which is very rarely seen in oncology with very few chemotherapeutic drugs. So in patients of a metastatic clear cell RCC falling into the intermediate and poor risk group, the combination of a ipilimumab plus nivolumab, especially in the intermediate and poor risk, has improved the survival risk, and it has stood that, and this is the 90-month follow-up data. So there is no doubt that the combination of immunotherapy in this scenario has improved the survival. Similarly, there is some benefit in terms of the favorable risk. When this was initially presented, the hazard ratio was more than 1, but in this follow-up after 10 years, the hazard ratio has fallen to 0.9, although still it is the upper boundary is close to 1.3, but it is showing. So what I meant that even favorable risk is now started showing some benefit in terms of survival. So similarly, the PFS, which is seen more in intermediate and poor risk, whereas in favorable risk, as we discussed, the PFS was not improved by NeoEP, but in intermediate and poor it has improved. And see the duration of response. If you see the duration of response in a NeoEP, it is close to the median duration is close to 82 months, close to only 20 months. So that means those patients who are responded to immunotherapy, the response will be sustained, and that is the reason the survival has improved to test of time. Similarly, the lung alone metastatic disease, the OS is quite much better compared to the other organ, the liver and bone. So this criteria sometimes we take in decision making. Patient presenting with a predominantly liver metastasis, we may not prefer IO plus IO combination, but in a patient who is presenting lung disease, not much symptomatic, we may prefer IO plus IO combination with a median OS of close to 50 months. Similarly, the side effect profile, if you see, these are the side effect time pattern with the side effect. All the side effects are mostly in the 1 year. This is the month axis, and this is the grade. So all majority of the side effects when we start the immunotherapy, it usually happens within 1 year. If you cross 1 year, the side effect profile probability is less. But in sunitinib, this is the sunitinib. If you see the side effect profile, and those medical oncologists who are doing that, we know that the any VEGF TKI will have associated toxicity in terms of skin toxicity, the endocrine toxicity, renal toxicity. So that means even after follow-up period, the toxicity remains a major challenge in patients who receive VEGF TKI. Whereas in immunotherapy, if you cross 1 year, only the toxicity which is seen is mostly the endocrine toxicity, that is hypothyroidism, and few renal toxicity. So that means from the toxicity point of view, initial 1 year is the difficult time. When you cross the 1 year, you can able to handle those patients with the immunotherapy without much problem. So in summary, the hazard ratio for OS with Nevo has stood test of time and remains stable. And those patients who respond or get a deep and durable response, the probability of long-term control. So practically now we are looking towards a cure in some subset of the patient of a clear cell RCC, especially those who disease remain controlled even after 3 or 4 years. So now we can discuss, or we can talk to the patient regarding the probable cure in a metastatic patient, especially in some subgroup of the patient. That what I discussed, that this trial mainly benefited the intermediate and poor risk group. But with this long-term data, we are now seeing some hint regarding the good risk or favorable risk also. So in a fit young patient, although falling in a good risk, if they want a deep and durable response, ready to take some toxicity, now there is some data that even those patients can be considered for a EP-Nivo, even they fall in a good or favorable risk. And if you see all the cross-trial comparison of all the IO plus TKI, so Checkmate 214 is the trial that we are discussing. Similarly, the Keynote, Pembro-XZ, clear trial of Pembro-Lenva, and Checkmate of 9ER. So the longest follow-up is seen with the EP-Nivo. And if you see the overall survival for all these trials, it is close to median OS is crossing the 4 year in all of the trials. So that means and if you see the corresponding arm of the TKI is close to around 3 years. And this is despite the 50% crossover of the TKI arm receiving the immunotherapy at the time of progression. So that means we are very clear that immunotherapy has substantially made an impact in management of clear cell RCC, and the survival is, the median OS in metastatic is reaching close to around 4, and now we are looking towards the median OS of close to around 5 years. So that means in the current era, in a clear cell RCC metastatic, either a combination of immunotherapy or a IO plus TKI is must, and it has definitely stood the test of time in terms of improving the survival. Similarly, the situation what we deal sometime in RCC, we get the sarcomatoid features. So previously when we used to deal with RCC with sarcomatoid, the median OS was used to be around 1 year. now with patients who are having a sarcomatoid feature, even the Checkmate 214, the median OS is close to around, median PFS is close to 26 months. So that means the tumor which was used to be a very bad tumor, now the median PFS is close to around more than 2 years, and the OS is definitely going to be much more. So the tumor which was poor biology, now it's with the right treatment, the survival is much better than the original clear cell RCC without. So that means it has just to test of time, but definitely everything comes with with some sort of other phenomena, that the toxicity which we are discussing. Similarly, the around 30% patient may need steroid, around 1/4 may need interruption. So managing a immuno-related toxicity phenomena may need some discussion, some sort of a management strategy, how to tackle this pain, because 1/3 of the patient needs a disease interruption or use of steroid. So that is very important, how to tackle the toxicity of the immunotherapy related phenomena. Similarly, the other problem which we face, the progressive disease. So when this trial was initially presented, the main problem was that around 1/4 of the patient presents as a progressive disease at the time of initial evaluation. So when we start giving IO plus IO, 1/4 patient progressed by the time the first assessment done. So that problem was one of the criticism of that trial. So the other group of the trial, the investigator decided to do this study, the Cosmic 1-3 study, in which the Ipi-Nivo was combined with cabozantinib. So one arm received the Nivo Ipi with placebo, and the other arm received the cabo with, so triplet in first line RCC. And this was the initial data of this trial was encouraging, although the toxicity is going to be the challenge. So this trial was a positive trial in terms of the PFS. It is still not reached in the triplet arm. Similarly, the PD, the progression which was seen in around 25% in this triplet arm, the progression was seen in only. So the problem of a progression was tackled when we combine a TKI with the IO plus IO in the first line, and this was seen in only 8%. Similarly, the toxicity which I discussed, when we combine two drugs and then add another drug, so around, if you see the grade 3-4 toxicity, more than 3/4 patient will develop some, so that is, we have to need the be careful when we deal with this drug. And definitely the overall survival for this data has not been reported, but definitely there is a hope that when we combine IO plus IO with TKI, the PD problem may be similarly, the negative study, that was the positive study. The negative study was the Checkmate 914, that in similar phenomena when they try to combine the IO plus IO in an adjuvant setting, that was the metastatic setting. In Checkmate 914, they tried if we can combine the IO plus IO in an adjuvant setting. This trial was a negative trial. The combination of IO plus IO in an adjuvant setting compared to the placebo. Unfortunately, if you see the DFS was the endpoint, it was both hazard ratio 0.92 and crossing the unity, and the 24-month disease control was more or less same. But this was one of the setbacks. We thought that this will be a positive trial, but this was a negative trial. And probably reason for negative trial in this, the patient who only 55% patient completed these. When you combine IO plus IO, the toxicity will be more. And in this trial, for whatever reason, they have defined only 6 months of adjuvant treatment, not 1 year, which is seen in Pembro. So this trial was a setback trial, and this was a negative trial of combination of IO plus IO. If you see the adjuvant setting, only one positive trial is from the pembrolizumab, the keynote trial. So still the adjuvant era is still open, only one drug is approved. Similarly, we know that all patient is not going to respond. So this is one of the trial which I feel that, just 2 minutes, last 2 slides. This is the trial in a prostate cancer. So giving immunotherapy to all patients is not going to benefit. In this study, what they defined the immune positive score. So patients who are having a MMRD deficient, so they defined the IMS positive patient with MMR positive, or a DNA damage repair positive, or a high tumor lymphocyte infiltration. So if there is any one of the factor is present in the tumor of a prostate cancer, those patients is going to respond. So this is one of the phase 2 study, and this has shown that if you select the tumor as per the tumor characteristic, not all patients. So this was a phase 2 study, and if you see the response of this study, the median OS is close to 16 months in a CRPC setting. So instead of giving all patients, if you define the patient as per this criteria for a metastatic CRPC, which is a cold tumor as per that. So this is the way to go ahead in this scenario, and in the future direction, that what I discussed, how to decrease the toxicity. So there are 2 or 3 major trials are all going. Means the ipilimumab is given every 12 weekly, not 3 weekly, which may decrease the toxicity. And similarly, the personalized tumor approach, like a TMB high versus inflammation cold dead. So ultimately, at some point of time, we will have this type of trial that first do the study, see whether it's an immune-rich tumor or not, inflammatory cold tumor or not, try to manipulate the tumor, and then give the immunotherapy. So I think this is the future for this, and definitely the immunotherapy has made a deep impact in urology, and we are seeing at least in the RCC, the survival has definitely improved, and it has stood the test of time over a period of time. Thank you.
Thank you, sir, for excellent discussion on the combination IO in genitourinary cancers. Now we can take few questions.
If no further questions, we would like to proceed for the group photo session. I would like to request all the chairpersons and Dr. Vineet Talwar sir and Dr. Amut Joshi sir also to please kindly come for the photo session. Thank you, sir. Thank you, ma'am. With no further delays, we would like to proceed with the next session before the lunch. That is I would like to call upon Dr. Swadeep Mohanty and Dr. Siddharth Patnaik, sir, to kindly chair the next session.
Good afternoon. I invite Dr. Krupa Shankar as a moderator for this session.
I'd like to request our panelists, Dr. Piyush Bajpai, Department of Medical Oncology, Delhi, Dr. Vikas Talreja, Department of Medical Oncology, Hemato-Oncology, Kanpur, Dr. Sachin Shekhar Biswal, Medical Oncology, Bhubaneswar, Chinmay Kumar Pani, Medical Oncology, Bhubaneswar, Dr. Shri Gopal Mahanti, Department of Medical Oncology, Cuttack, Dr. Amit Joshi, Department of Medical Oncology, Mumbai, Dr. Ashwin Tamhankar, Department of Surgical Oncology, Mumbai, Dr. Biswajit Nanda, Department of Urology, Bhubaneswar, Dr. Gautam Panda, Department of Medical Oncology, Bhubaneswar.
All right, so now that we're all set, very good afternoon to one and all. And before I begin, I'm going to take this opportunity to congratulate the organizers, led by the ever-dynamic Dr. Ghanshyam Biswas, for putting together this wonderful academic extravaganza and also for having all of us here. So I have this arduous task of going through this panel with the role of looking at the role of IO in GU cancers. And I've decided to focus on metastatic bladder and renal cell carcinomas. And I have quite an elite and a distinguished panel here to take me through this. So obviously, this is why we can't resist talking about immunotherapy, because it's here to stay and it's so hot right now. You just can't keep it out of any solid tumor malignancy. So let's begin right away with my panelists. So do you believe there's a role for biomarker testing in bladder cancer? If so, what are the biomarkers that you would like to look for? Anyone? Or Piyush sir, we could begin with you.
Yes, of course, biomarker testing is important. As you have already laid down a couple of biomarkers over the FGFR3 and HER2 expression, because we have the ADC, HER2, ADC, FGFR3, of course, we do not have it right now, but of course, patient access could be there. Otherwise, also BRCAs is what we usually also would want to do.
Okay. So anyone else on the panel looking for tumor agnostic markers as well, like MSI, TMB, NTRK, BRAF, RET, and so on and so forth? Would you
definitely agree that yes, for all solid malignancies, it is a norm to get a testing done. However, for bladder cancer, inherently we know that the positive predictive value of MSI and TRK is probably 1 in 1,000. So FGFR is a targetable, drugs are importable, so it is definitely worth a shot. But for most of the patients, I believe, I
Agreed. So, since we all keep talking about FGFR, so would you like to look at FGFR fusions, rearrangements,
I think first thing is that we discuss with the patient whether they are going to afford. So even if FGFR comes out to be positive, how many patients we are going to get the drug? So at this point of time, in a straightforward patient, I don't think so. If patient has exhausted all the options, they want to explore. Otherwise, in a run-of-a-mill case, I don't think so that is still there. Unless we get a specific HER3 expression and then so
exactly as part of tumor agnostic therapy as well. But then remember FGFR, I'm sure a lot of us would have used lenvatinib, when we have, when people have not been able to. Yeah, please.
So it is indicated. The question is at what time? And do you have the lag time for waiting of the NGS report to come? Yes, probably we do prefer to do concurrently, but how many of us are actually doing NGS upfront before starting or before deciding any kind of therapy? I think my take on is start the therapy, take a discussion henceforward and send for the NGS, wait for the report to come, then you discuss if the patient is affordable and keeping in view the data set in front, I think we can.
If cost is not a constraint, how many of us in the panel would want to do an NGS right up front, including a PDL1, MSI and TMB, if cost is not a constraint?
So if the patient is getting infotumab plus pembrolizumab as the first line option, I would definitely recommend.
So fair enough. So I think the key thing here is, remember that, when we talk about the molecular subtypes of bladder cancer as well, remember they are luminal, basal subtype, and then P53 driven as well. Luminal is where FGFR expression comes in. And, post multiple lines of therapy, if you have somebody who can't afford a FGFR inhibitor, we have used lenvatinib as well, which is a pan-TKI or a multi-targeted TKI, which can also action on the FGFR. So, those are things that you could look at, do it, and remember that not all mutations are created equally. So you got to remember to distinguish between, FGFR fusion versus a rearrangement versus an FGFR mutation. So immune checkpoint inhibitors in bladder cancer, why are we not looking at PDL1? Do we believe that, a bladder cancer behaves differently? Is that why we're not looking at it? Or do you believe that we should look at it probably because the magnitude of benefit might be higher? You just spoke about using expensive drugs. So probably if somebody is likely to benefit more, would you look at doing a PDL1 and gauging, okay, this patient is likely to benefit more from IO as opposed to another patient who doesn't have any PDL1 expression? Given that it's not a perfect biomarker, we're still understanding the tumor microenvironment, the other factors like MSI, TMB, TILs, tumor infiltrating lymphocytes as well. Thoughts from the panel? Amit sir, yeah.
So I think the only place where I want to consider is the adjuvant immunotherapy, adjuvant nivolumab, because the benefit has constantly seen in a PD. Otherwise, in a metastatic second line, all has benefited. And if you see the real benefit, it's not the contrasting difference between PDL1 positive versus negative. So generally don't do it in an adjuvant setting when the patient has some sort of a tolerability issue, whether we are not sure whether it will benefit or not. In those scenarios, I consider for doing PDL1.
Perfect. So I think even in the adjuvant setting, the approval is irrespective of PDL1 expression. But like was rightly pointed out, the magnitude of benefit definitely is higher for patients who have a higher PDL1 expression, at least more than 1%.
The ESMO has approved only for the PDL1 positive.
Correct, but FDA has approved it for for all comers. So the intention to treat population did include patients who were PD-L1 negative as well. So I think it's a fair statement. And this is how far we have come. We've started off with just platinum and then MVAC. And then finally we are in the era of combinations where we're looking at IO, not just IO alone, plus IO, or even IO plus other ADCs as well. So that's where we are today. And if you look at it, we've started off with a dismal survival of close to 11 months, and now we're talking about close to 2.5 years for metastatic bladder cancer. So, let me again ask this question. We already heard about the data from, where Vikas pointed out EV plus Pembro. So does platinum eligibility still matter? Because the trial included all comers, either platinum eligible or ineligible. So does this still hold good in your practice? Let's say probably, let's not bring in the cost here again. Your patient can afford every drug in the world and is willing to go on EV plus Pembro. Do you still think platinum eligibility matters? We'll begin from the far left this time around.
No, sir, I don't think platinum eligibility matters because if the patient is being planned for EV plus Pembro, we can start right away, even if in ineligible patients.
Okay, so you're saying that it will still hold good, irrespective of whether the patient is going to be platinum eligible or not. Yeah.
Yeah. Otherwise, I want to comment that if we use N4 plus this Pembro upfront, then what will be the next option? So options will be squished. So in that case, platinum eligibility still we are using, and upfront we can go for the Gemcis plus followed by Avelumab. Then second line we can go for EV. So
yeah, so I think
in that case, if I am trying to use the first line Gemcis followed by this one Avelumab, then I will go for the platinum eligibility.
So how would you choose? I mean, what would you do?
In my practice, I go for platinum eligibility.
So you go with platinum eligibility.
Not yet started with EV plus.
So in that sense, would you add Nivo plus Gemcis? Would you add Nivolumab in the first line setting in platinum eligible? Because that data is also there, and that's a positive trial as well. I'll show you that as we move along.
But longer data, what we have is with Avelumab data. Gemcis followed by Avelumab. We have the longer data.
I think one way of looking at it is if you have somebody who's platinum eligible, and if they are willing to go on platinum, so you could either choose between EV plus Pembro if they can afford it, or you could even add Nivo plus Gemcis as well. If they're not eligible, then you do Gem Carbo and then follow it up with Avelumab maintenance. That's another way of looking at it. But then that's your NCCN as well says that's a preferred option, whether it's platinum eligible or not is EV plus Pembro. But I think a word of caution here. They're just guidelines and not just actual rules. So EV, the drug, I'm not going to get into the mechanism of action of the drug, but it's definitely one of the most potent drugs, especially as an ADC, which incorporates the cytotoxic agent being monomethyl auristatin E, which is a microtubule inhibitor. So that's the data, pretty impressive, doubling of the PFS and the OS as well. So 30 versus 15 months is where we are right now. So is this good enough for us to say, okay, fine, this should be the new standard of care? Yeah, because you already have the mic in your hand, so I'm sure you're wanting to make a comment. Go ahead.
Metastatic bladder cancer, again, is a huge, I would say, Pandora box. For us, it is very important that we have to differentiate between those who have an oligometastatic or somewhat near curative potential treatment. And that's where cisplatin is still one of the major drugs, because cisplatin does perform superior in terms of the carboplatin. Once we label it and term it as a palliative intent, I believe cisplatin and carboplatin are equivalent in terms of the efficacy-wise, because the response rates, whatever they create, are short-lasting, and they need some kind of immuno to back upon.
But before we get any further, I think this is an interesting comment. So how many of us here, in the audience included, believe in the concept of oligometastatic bladder cancer? Because, we talk about other oligometastatic subsets, right? But in bladder cancer, do we still believe that oligometastatic disease?
Very rare.
All right. So
I also agree that it is a very rare concept, but yes, for last 2 years, I have seen 1 or 2 bone mets giving an NACT-like intent with Gemcis and Nivolumab. And they have done beautifully. They have done. We have done surgery. They are doing well so far now.
But we don't know if they could have benefited. The same benefit would have been achieved with EV plus Pembro in the first line.
When the immunotherapy wasn't there, the concept of it wasn't there. Probably at that time, oligometastatic was a whole metastatic. But with the coming of immunotherapy, I believe you have to.
Yeah, agreed, Vikas. But then, I think the point is, conventionally going back, the time tested, even the Vesper trial looked at MVAC versus Gemcis. MVAC still holds good. How many of us are actually practicing MVAC or doing it in practice? Any show of hands on the panel? Anyone doing dose-dense MVAC? You are doing it, please. Can you tell us your experience with dose-dense MVAC and with growth factor support?
patients who are young and fit, I am using DDM-VEC. I use them.
So what proportion
in
of patients are you actually able to deliver your dose-dense MVAC and also complete all cycles?
For last couple of months, maybe 6 to 8 months, I'm using DDM-VEC in patients who are young and fit or slightly
the ballpark figures. The ballpark figures on an average percentages.
On an average, I am using 60% of the patients. I am using DDM-VEC and 40% as the GEMSIS. And one comment about the NGS, basically, we should remember we are living in a LMIC, and I think AR receptor should also be done because there is data regarding efficacy of the androgen inhibition in this case.
Absolutely, key point. Agreed there.
Again, one point regarding the cisplatin eligibility and ineligibility. Cisplatin ineligibility does not only mean that we are not able to deliver cisplatin. It also means that patient has some comorbidity which might hamper the outcome. So it also indicates that patient might have some decreased OS, which I think is not seen with the cisplatin eligible patients.
Agreed. I think, sometimes platinum eligibility defines also sometimes people may not be even eligible for even carboplatin as well. That's a key point. Agreed. And then again, coming back to your point that you made, right, about AR receptor as well. So that's more common in the luminal subtype where FGFR, along with FGFR, you could also look at the androgen receptor as well. So I think, the verdict is, this probably looks like a clear winner in the first line setting. The other options are you could look at doing avelumab maintenance as well, even if the patient is not eligible for cisplatin. And so what about second line treatment? I know this was another, let's say you used EV plus pembro in the first line. What are your second line options? I know the NCCN says that that's how you go about it, but in practice, forget about even EV or pembro, any other options that you would like to go for in the second line, even if you've used IO in the first line?
Chemotherapy is still there, sacituzumab as you have shown, but then sacituzumab has failed, Tropics 4 has failed. So of course, at that time, it's the time that you should look at the NGS report probably.
So just the key thing here about saxituzumab govitecan, it was touted to be the next big thing in uro-oncology, but unfortunately, it had an accelerated approval, and now it's been voluntarily withdrawn for its indication from the market in this setting. And that again tells you that you'll have to wait for longer term data to mature before you could actually call it a full regular approval. So it was just an accelerated approval. So that's probably your reaction when you call it FDA approved.
I think so. There is a phase 2 trial now. What they're doing is, as Dr. Talwar sir was saying, that enfortumab and pembro, the toxicities, skin toxicities, residual toxicities were too many. And in fact, a lot of attrition, 37% attrition in the trial. Yeah. So they are actually designing a phase 2 trial which is ongoing where they are just inducing with the EV and they're dropping it off with the pembro being continued, and probably a reintroduction of EV could be done at a later.
Yeah, perfect. I think, that's probably, we will have to look at what the ideal duration is for us to actually look at re-challenging the drug, what the treatment-free interval is likely to be in that sort of a setting. But, quickly here, I'll bring your thoughts on this as well. Because
I have 3 patients on enfortumab right now for last 2 years. And they have been continuing so far so good. And they have presented in a very bad shape in terms of the disease burden. And they have responded phenomenally. I am still confused because I also don't know what to do next after the same. And as we know, that's a cold tumor, not much hits on the NGS at all the time. And I believe the panel and you will enlighten us as how and what to do. Because if you go back to the trial also, post progression follow-up, the data is not that much encouraging right now.
Very true. So, because EV plus pembro has just been approved, and all of the trials that were done in the prior era did not have EV plus pembro in the first line approved at that point in time, which is why, if you look, it's a data-free zone. So, you're likely to use your own clinical expertise and your, what you believe is right for your patient sitting across the board. But I think this is what we're looking at, in the future as well. So, I think, this is definitely the few concerns about toxicity as well, especially in patients who are diabetic and who have neuropathy, and especially who have uncontrolled autoimmune disorders. If it's grade 1 toxicity, I just wanted to highlight, you could just continue and just start them on steroid or topical applications. So I'm just going to complete. So, what about key toxicities with the erdafitinib? Anyone's used it in practice? Any FGFR inhibitors? Any experience with that? Yeah, sure, please.
It causes a lot of skin toxicity, especially the nail changes. And I've seen
Absolutely.
the avulsion of the nails, and second is the hypophosphatemia, that is refractory. So those are and ultimately the lowest dose started with the highest dose, and then I think 3 mg, I don't remember the exact dose, but I have 2 patients, I had a very bad experience. I thought that they could have been better without the drug.
So I think, as a group, these FGFR inhibitors, be it pemigatinib or even infigratinib, and then futibatinib as well, all of these as FGFR inhibitors have this class side effect. So I think, obviously, if you're looking at neutropenia as a concern with Saxi, I'm going to conclude with that and say the future, we're looking at combinations. There are a lot of ongoing trials looking at combinations, especially, when they've progressed. And probably this will give you the answer to Vikas's question as to what you would do when they progress. IO plus TKI is here, or again, IO plus other IO agents as well. We would call it the IO, as in Tamil, IO means, oh my God. So probably, we're in that zone where we're looking at all combinations, and combinations will probably rule the roost going forward. So with that, I'll conclude and say thank you all, and thank you to my panelists for making it so interactive. Thank you. Thank you for your patient listening. Thank you.
Thank you very much. Dr. Krupa Shankar, Department of Medical Oncology, Coimbatore, for moderating the session, along with all the panelists for a nice interactive session. Yes, the evolution of immunotherapy has made a huge impact in the field of oncology, especially in the locally advanced, metastatic, and recurrent disease. Proper selection of cases, smartly choosing the drug, and precision, especially in high TMB and MSI high cases, will definitely going to benefit. Thank you.
Thank you, sir. Now I would like to request the chairperson and the speakers to come on stage for a group photograph. Thank you so much, Krupa Shankar sir. Thank you all the panelists. So for our next session, Symposium 3, which is sponsored by the MSD, this is on the topic of redefining treatment outcomes with adjuvant immunotherapy in renal cell carcinoma. I would like to invite Dr. Professor Soumya Surat Panda sir, HOD Department of Medical Oncology, some hospital. Please give a huge round of applause, please.
Good afternoon, everyone. Standing between you and the lunch. So we'll discuss on redefining treatment outcomes with adjuvant immunotherapy in RCC. So RCC and bladder, the adjuvant therapy has changed a lot now. So earlier the days were that if RCC was there, then you did not think of any adjuvant therapy because the data was very weak, and that's why a comparator arm across all the adjuvant has got a placebo. So we'll cover on the overview of RCC, then we'll go to the stages, and then the adjuvant treatment for early RCC. Then we'll discuss the trial, the keynote trial, and the safety profile. With that, we'll end. So this is an overview of RCC. So RCC for the residents that arises from the proximal renal tubular epithelium, and most are renal cortical. And the most common, it is one of the most common kidney cancers between the age 50 to 70. So the age distribution is very important. And the incidence is increasing with time. Coming to the classification of RCC, it is a huge one, it is a busy one. What we'll concentrate is the major morphological subtypes. There is the papillary RCC, the chromophobe RCC, and the clear cell RCC. And the incidence of clear cell RCC, that is 78%, and the papillary is the next most common, it is 15 to 20%. And why it is clear cell? That is because that is characterized by lipid and glycoprotein-rich cytoplasm. That's why it is a clear cell RCC. Coming to the risk factors for RCC, again you have to remember smoking is one of the risk factors for RCC, obesity, high blood pressure, and family history, and it is more common in men than in women. Then again for the residents, if you have got a case of that, then you might be asked of the genetic diseases like von Hippel-Lindau and the hereditary papillary RCC, the Britt-Hogg-Dubé syndrome, and the Cowden syndrome. Remember, the Cowden syndrome, the patient has a younger onset RCC, and it is more common in females. So it is a bit different compared to the RCC that we see. So if you have got a young RCC and female, and the patient has got, along with that, some skin lesions, other cancers, then you might think that there is syndromic association might be there. So these are the risk factors for the RCC that you have to remember. The survival rate, the 5-year survival is highest for those with the localized disease. Regarding the global burden of the RCC, again, kidney cancer is ranked 15th. You always see, if you have got you are seeing multiple cancers, you will see 1 or 2 RCC coming into your OPD in an adjuvant setting right now, the urologist being, if you have got a urologist in your institute as well. So they will send you across post-treatment mostly. Then the global distribution of RCC, very common in Asia. And coming to the prevalence of RCC in India, again, it is very similar to the global scenario. It is more common in males. And again, the age range is 50 to 60 years. And you have got clear cell as the most common subtype, accounting for around 70%. That's what the data for the global cell. So in a nutshell, stage 1, stage 2, and stage 3, and stage 4. So stage 1 is limited to kidney. You have got a higher survival for that. Stage 2, involvement of perinephric fat remains limited to Gerodes fossa. 5-year survival drops down to 50. In stage 3, there is a renal vein involvement. And in 3B, it is nodal involvement. And the 5-year survival is 30%. So it crashes down from 90 to 50 to 30. And when it comes to stage 4, the survival, 5-year survival is something like 5%. So you want to catch on those stage 2 and stage 3 where the rates are decreasing, and you don't want them to go to stage 4. That's where the adjuvant strategy comes into. So prognosis depends on the stage of the histology cell type. So the clear cell RCC, the 5-year survival is 70%. Papillary is a bit better, 90%. This is the TNM staging for the RCC. Now the stage-wise management, you know across all stage 1, stage 2, stage 3 is the surgery or the radical or a partial nephrectomy, that is the treatment of choice. And regarding stage 4, again cytoreductive surgery is there. It is one of the cancer where you can undergo a surgery even in stage 4, but then it is debatable in the immuno era. And you have got some spontaneous remissions in stage 4. It is one of the cancers that had interleukin-2 interferon right there. So you always knew that it was immunologically a bit, the patients had a spontaneous remissions as well sometimes. So some bit was going on earlier also. We knew that it is the area where the immunotherapy will be there in the future as well compared to other cancers. So high recurrence rate in early RCC is there. So you've got a recurrence rate of 8% and 3% metastasis rate is there. Coming to the recurrence following nephrectomy, there are risk factors. As you know, the partial or radical nephrectomy is the standard of care. Risk factors like disease stage, the size, the nuclear grade, and the regional lymph node involvement. Somebody with a lymph node involvement automatically becomes a high risk. So it becomes a advanced stage 3, it becomes a stage 3. And again, it depends on the sarcomatoid. If it is there is sarcomatoid features, then again it is a high risk. So that is also there. So RCC recurrence worsens the patient prognosis, and adjuvant therapy improves the outcomes in RCC. Current adjuvant therapy agents that was tried prior to the immunotherapy was the VEGF, but it has not been successful in showing a overall survival. You had some small amount of DFS benefit which did not translate into OS benefit, and that's why it was mostly restricted to kind of a very high risk, stage 3, and you can select out some patients if you feel, but again that was a kind of not a standard of care. So that's why all the adjuvant arms had a placebo as a control. So that's why the immunotherapy. And why immunotherapy? Again, you know that the PD-1 and PD-L1 and the CTLA-4 pathways has demonstrated in the metastatic setting. So on that logic, it should work again in the early stage as well. So stage 1A, again surgery. Stage 1B, again surgery, and followed by surveillance. And stage 2 RCC. So if in stage 2 RCC, if a patient has undergone a partial nephrectomy and the patient carries a grade 4 tumor and there is sarcomatoid features, then the option of adjuvant immunotherapy is there. Coming to stage 3, again you have got the data for adjuvant pembrolizumab there. So Keytruda is a PD-1 inhibitor. Always remember the PD-L1 has not been so far very successful. We had Atejo, the I-OSICON 010 didn't work there. And that is almost the inclusion criteria was very similar to the Keynote 264. That's what I'm going to talk about. But PD1 is more active in the RCC compared to PDL1 that is there. And regarding the dosage, 200 milligram every 3 weeks given for 1 year, 17 doses. So this is the Keynote 564 based on which we are now considering adjuvant. If you look at the trial design, very important that you look what was included in the trial. It was a RCC and patients were intermediate or high risk. What is intermediate or high risk? I'll tell you in the next slide. And patients, they were included within 12 weeks of surgery. And patient did not, of course, have a immunological disease. And those patients, they were randomized. M1 NED. M1 NED means patient is having a oligometastatic RCC and you have operated everything. So that is M1 NED, you have taken out. So M1 NED and patient is now randomized into Keytruda 200 milligram IV 3 weekly, and the other arm is placebo IV 3 weekly. Why placebo? Because that has been the standard arm for across all the adjuvant trials. So the TKI has not been proved to be successful. That's why the placebo is there. And the duration, again, you have to remember 1 year duration. That is again very, very important. Why 1 year? Because you had some trials, adjuvant immunotherapy trials like nivolumab, which used 6 months. So you had, and that was that's why Keynote 564 is successful compared to others. That part you have to remember. You had M1 NED, you had sarcomatoid, you had a duration of around 12 months in the adjuvant setting. So those very, very finer points you have to remember that part. And probably that is the reason that it has been successful. Besides, Keytruda has got a difference mechanism of action as well. So those finer points you have to remember. And coming to intermediate high risk and high risk. So intermediate high risk PT2 with grade 4 or sarcomatoid, and PT3 any grade. High risk include PT4, any PT with a node positive. So that was a high risk. So PT4 node positive, that is a high risk. M1 NED, 5% of the patients in the trial were M1 NED. So this is the baseline characteristics. Around 6% was M1 NED, and mostly high risk features were there. Sarcomatoid were around 15% of the patients were sarcomatoid. So you had a trial which had included M1 NED, you had a trial which had included sarcomatoid, and you had a population which was very high risk. So with this background, the primary endpoint we will discuss. DFS was a primary endpoint of the study with a secondary endpoint of overall survival. So whether DFS was an acceptable primary endpoint, so answer was yes. So DFS was a definite primary endpoint because it correlated with the OS. And now that Keynote 564 also has demonstrated OS benefit, so therefore the doubts were a little bit, now it has gone away that you have got a drug which shows a DFS benefit, which has got a OS benefit. And so Keynote 564, superior DFS with Keytruda versus placebo, that was the initial results. 24 months, if you look at the DFS, it is 77% versus 68%. And the patients, the p-value was definitely significant. And then you had got an exploratory analysis of 30 month. Again, 30 month was around 70% versus 65%. And the subgroup analysis across the age, gender, and the PS. And the PDL1, of course, PDL1, again, we don't do in RCC. The positivity rate of PDL1 in RCC is around 25%, so for that matter, but we don't see that. Irrespective of that, we will give them the adjuvant. And M1 NED definitely had a higher benefit with a hazard ratio close to 0.28, but remember that was only 5%. It is not driving the whole population, of course. And then you have got the difference in nephrectomy. But this forest plot, that should always be remembered while interpreting a forest plot, that this was not powered to detect the differences in the treatment effect in the subgroups. So if the primary endpoint is positive, then you take the trial as a positive. And so this is the subgroup analysis of the DFS as per again, the forest plot showing the irrespective of the gender and the ECOG PS. This is the exploratory. So again, the updated is showing the same thing, that across all, the benefit is still maintained with time. This is the OS data. So the overall survival with adjuvant pembrolizumab, the hazard ratio was 0.68. This came out in April 2024, so nearly a year back. So in that, they had clearly demonstrated that even after 4 years, the OS was 91% versus 86%. And the p-value was significant. The hazard ratio was 0.68. So the DFS is there, the OS is there, and both your primary endpoint and your secondary endpoints, they are positive. So you had a trial. So what we were worried is that whether the patients were adequately treated in the control arm, that is the placebo arm, whether they received the immunotherapy or not. Again, that was that part also a little bit calmed down after we saw that The placebo arm, if you look, around 88%, 85%, they were exposed to immunotherapy when the patient progressed. So that's again, the trial was kind of what we do in the real world. If the patient progresses, then we keep it the immunotherapy at the end, and that has been done. So the crossover is allowed, and 85% of the patients, they received a immunotherapy when the patient progressed in the placebo arm. Besides the PROs, so the patient reported outcomes, so no detrimental quality of life deterioration was not there. However, the treatment discontinuation will come to that. So the treatment discontinuation is around 20%, remember that. So in the NIVO trial and the NIVO EP trial, that had around 30%. So again, the treatment discontinuation is 20% in this particular trial. So due to the adverse events, so that part you have to remember that. So around 1 out of maybe 5 discontinuing that. So the cause of discontinuation being the adverse event like fatigue, diarrhea, pruritus, these are there. But specifically, if we look at the immune-mediated adverse events, so again, as you expect, the endocrine abnormalities, hypothyroidism, hyperthyroidism, pneumonitis, that was not very high. It was grade 3 and grade 4 were very less than 10%. And it was kind of a safe drug, and kind of the side effects were expected compared to the other adjuvant IO trials in the same context. So coming to the summary, so indication, Keytruda as a monotherapy is indicated for the adjuvant treatment in patients with RCC right now, in intermediate and high risk. So when it comes to PT2, means with a high risk, so that particular group of patients, when it is not node positive, when it is not T4, so those patients in our context, they go into the tumor board in our hospital, and then we discuss about that part, discuss with the patients, and then go to that. But you have got a DFS benefit, you have got a OS benefit. Across all, you see the margins are still good. So that's why most of them still go ahead with the adjuvant therapy, and most of the patients are also willing for the same thing. So Keytruda as a monotherapy is indicated for the adjuvant treatment of patients with RCC at intermediate high risk or high risk of recurrence following nephrectomy, or M1 NAD, of course, following resection of metastatic, that is M1 NAD, following resection of metastatic lesions. And the patient population, as I told you, PT3, any grade N0, M0, PT2 with a grade 4 or sarcomatoid features, and high risk, as I told you, PT4 or any node positive, and M1 NAD. So these were the population that we are giving them the adjuvant. And the efficacy is there, DFS benefit and OS benefit is also there, and the discontinuation was around 20%. With that, I conclude my talk. Thank you for hearing me.
Thank you so much, sir. So before we break for lunch, the lunch has been organized in the ground floor near the exhibition hall. And I would like to request you all to be back in the next 30 to 35 minutes by 2:10 p.m. Thank you.
There's an announcement that the basic science posters will be evaluated today during the lunch time.
hypertensive, presented with hematuria for 2 months and increased urinary frequency and mild dysuria for 2 months. Physical examination was unremarkable, no palpable abdominal masses or suprapubic tenderness. He had grade 1 prostatomegaly. So, oh, can somebody display it here, please? Yeah, so I'll repeat, a 62-year-old hypertensive came with painless hematuria, physically nothing remarkable, and he has grade 1 prostatomegaly. So how would you proceed? Vikas?
So, cystoscopy to see the cause of the hematuria, do the urine cytology.
So I'll butt in between. Cystoscopy showed ulcerative lesion in the posterior wall, and this was a high-grade urothelial carcinoma invading the superficial, basically CT2. What else would you like to do? Cystoscopy, then?
So I think so the workup is complete. CT Uro and MRI has been done. Planned for. So has the muscle cut been taken? The superficial muscular
They have it. They have it.
Okay. Perfect.
So Kripa, anything else you have to add at this point of time?
So as far as imaging modality is concerned, the CECT.
We'll come to that.
Oh,
Anything, in this basic workup?
So, I would probably look at getting IHC done as well. So sometimes there can be differentiation or de-differentiation as well, which we got to account for.
You would look for some divergent histology?
Exactly, divergent or even de-differentiated histology.
So that would be the next question. Sahu, from the pathology point of view, whether only this histopathology is enough or anything else?
So I think as far as divergent histology is concerned, you could have squamous differentiation or an adenocarcinoma differentiation. And as far as de-differentiated, you could even have a small cell de-differentiation where your treatment can actually slightly get altered as well.
Fine.
So that's one. And also as far as the response to therapy, we know these are all pretty aggressive tumors. They don't respond as well to neoadjuvant therapy if you have especially a divergent or a de-differentiated histology. So I think for that reason, I would definitely look at those parameters.
So, Sahu, coming to you, anything else apart from this?
I think at this point it looks...
so you have a non-metastatic situation,
yeah,
you have these markers. How
so basically a CCT chest, if it is that's okay or X-ray is okay, I think we have enough to proceed forward.
Vamsi, would you like to do FGFR at inception?
No. In such a case of PT2 and 0, no. Not for treatment purposes. As part of later, I might as well send the final cystectomy specimen.
Data says that in NMIBC, the expression of FGFR is the maximum. So that won't enthrall you here? No?
No, at this point, no. And this is MIBC, this is not NMIBC.
Yeah, yeah. So even in MIBC, you have about 5 to 7% expression. Anything? Any one of these markers at inception?
Right now, none of them have any therapeutic altering potential. So for now, none of them. But as Dr. Vamshi said, that for possibly a retrospective analysis or something for a data collection, it might be good to have a look at these samples for future. But for clinical Monday morning practice, none of them.
So I think we are good with the IHC.
The only thing I'd like to put forward is it's a T2 and 0. So if I'm thinking of something as a systemic therapy at some point of time, I would love to definitely document the PDL1 or CPS, whatever you would like
That's
to
exactly what I asked.
Yeah, so if you're looking at that, yes, I would like to. The Niagara had its own criteria of putting
We'll come to that. We'll come to that.
So, that's the only thing that proved. So my point is, PDL1 is the very likely thing which I might ask, which I'll ask for. The role of ctDNA is not done now. At upfront, will I ask? No. Post-surgery, probably, but not. We'll talk about that if time comes.
Yeah. So this is basically a T2 N0 M0. And what are these staging investigations now? I think, again, we can start.
So I think so. The CT Euro has been done. Cystoscopy has been done. Complete evaluation in terms of the local imaging has been done. as Dr. Sausa said, that the chest modality need.
Any role of PET-CT? Normally the patient's attendants get a PET-CT for you, but...
So I think the only role of a PET-CT is because it is incorporating a good CT scan, it's a single thing, you can do it. I mean, beyond that, we don't stress upon it, and if a CT abdomen has come, we don't ask for one more. We just do a CT chest.
Right, right. So, upfront surgery versus neoadjuvant chemotherapy, one-liner, Kripa?
So, I think even in all the neoadjuvant trials, including the SWOG study, they've looked at including patients with T2 disease as well. So I think, here also I would look at probably offering them neoadjuvant therapy if the patient is fit enough, especially cisplatin eligible. So I think I would definitely look at using neoadjuvant therapy here.
So as you can see, if you do radical cystectomy, you have a local control, but the 5-year overall survival is again dismal, 30 to 60%, and almost half of the patients die of metastatic disease. So, is there any evidence for neoadjuvant chemotherapy?
Yeah, I think neoadjuvant versus adjuvant, if you have to give chemo, it is neoadjuvant, and there has been a OS benefit of around 5 to 6%. So I would probably always offer a neoadjuvant to a cisplatin eligible. So that's important. The GFR is going to play an important role if it is beyond 50 or 60. 60 is the cutoff, but 40 till 40 or 50 also people have tried it in divided doses. But so a good GFR and cisplatin eligible, I think neoadjuvant is the way to go forward.
Agreed. So you can see this forest plot here.
So the only caveat to that is you don't have a trial specifically done for T2 N0. So if it is a T3, T4, N+, I don't think there's any doubt you should go for a neoadjuvant. For a clear T2 N0, if there's any borderline thing or cisplatin is not very good, I would be happy. I would be fine to do a surgery as well.
So if a person is, physically good and you have a platinum sensitivity, I think all of us agree we'll give neoadjuvant. Now
we agree to give it.
A lot of times we have discussed about DDMVAC and GC. So, Vamsi, Vesper, just for the sake of documentation, what are the caveats and what are the plus points for both?
So I haven't used DDMVAC. I don't know whether it's just a bias on my side. I know centers which are giving it and are comfortable with it. I don't have personal experience, so I generally give GC.
So this is your
personal
current practice,
yes.
right?
Yes.
So,
same here, but looking at the data and having read a little bit in the recent past, I think DD-MVAC should be the way forward. And if you're giving a neoadjuvant skeleton, should be, and that has proven to be superior in all levels of trials, even in the retrospective, in the prospective data set, that is the regimen that's most active. And it's, my colleagues who have done it tell it's doable in our scenario also. So I feel GEM-CIS is definitely inferior to dose-dense MVAC. And
so
that should
Kripa, do you think the Vesper was able to give the adequate data for the inferiority or the non-inferiority?
Vesper, I look at Vesper in addition to all the other data set which is already there. And every data set is pointing to the same indication.
All other data did not have a head-on.
I understand. So
I think
let's have your thoughts.
So I think it definitely comes at the cost of increased toxicity. Even if you look at the Vesper trial, the number of patients who went on to complete all cycles of adjuvant therapy was definitely less than 50%. So in the setting of a clinical trial, if only around 50% are going to complete all these cycles of therapy, I think real-world data will definitely be much less.
Yeah,
so always argue saying that despite that, you were able to show an improvement in DFS and overall survival as well.
So before you say something, if you have a super fit patient with all the criteria met, will you give DDMVAC, will you give GC, and why?
Today, yes. I would like to change my practice. Point number one. Point number two, taking something from what you said, if you look at even the latest of the data set from Niagara, and look at that 70 to 80% started adjuvant chemotherapy.
We'll come to
no, I was coming to something else.
Yeah.
Only 50% completed the scheduled adjuvant therapy. So even with GEM-CIS, you don't find a lot of people completing a therapy, and especially in the adjuvant setting. The neoadjuvant is something else.
Yeah, sure. Maybe if you're lucky enough to have super fit patient in practice, I think all of us would really want to give dose-dense MVAC. But I guess in practically, and when we come across your operations, very rarely do you get to see patients who will be fit enough to tolerate even dose-dense MVAC with growth factor support.
Between CARB and CIS?
So definitely CIS has shown superiority in terms of response rate, which is our primary criteria. also in terms of the loco-regional and the DFS survival and the overall survival. So if the patient is not suitable for cisplatin, I would plan a upfront surgery and then possibly make him. Also, we'll try to make a patient cisplatin eligible by trying the split dose GemCis protocol. Also, because when you give an inferior regimen, your outcomes do tend to hamper in long terms. And that's where it is better not to give rather than give an inferior regimen. And between the dose stands and...
so in those patients who have a low GFR, you try to change to
split
cis.
The dose.
You split the dose.
Split the dose.
Till what level?
So...
till what GFR?
So anything which is below 60, I would split the dose in day 1, day 8.
Suppose it's 45. Would you
split the dose? We'll split the dose to day 1 and day 8 for most of my patients. At the end of the day, it is important for the patients to continue and reach up to the 3 or 4
I think
cycles.
the NCCN guidelines are also in conformity. Wherever you find that the patient is not eligible for cisplatin, you try to either operate or refrain from giving carboplatin. Yes, sir.
So I was coming to that. The split dose doesn't have any precedence. It does not have a level 1 evidence. We don't know what the split dose. We only have few recent trials telling from 40 to 60, you give the 35 per meter square. But if you look at it, where that evidence has come from, I could not find one.
So, Kripa, what do you do in your practice? What are the most common causes for cis ineligibility?
I think more often than not, it's the age and the performance status, and again, definitely the renal function as well.
Comorbidities with it.
Absolutely.
Yeah, Vamsi.
The location of the tumor matters. If you're close to the VUJ or if obstructing the VUJ, those are the places where you have more challenges in a bladder.
So these are the trials which are going on in cis eligible and cis ineligible, and one of them which we are going to discuss is the Niagara trial. So immunotherapy. Do you already have the experience of using immunotherapy in MIBC?
We have used it in one or two. We have used a couple of patients. No, we have used a couple of patients. One who is cis ineligible, upfront surgery. The second patient who was neoadjuvant chemo did not have a complete response. So those are the two settings we have used.
So, Saud, till which T stage would you be comfortable in sending the patient for surgery if it is ineligible?
If it is ineligible for cisplatin, T3 N plus N0 would definitely has to go for surgery.
T4?
T4, the adherence would matter where it is going through, and that is a surgeon's call.
So T4 you have to do a multidisciplinary this thing. Yeah. So this was the adjuvant trial, Nivo versus placebo, and here you find that there is a DFS benefit. That was the primary endpoint, and approvals for patients with PDL1 greater than 1. This was the criteria here, which is very, very specific. And this is the ambassador trial again, similar endpoints, and this also had the DFS benefit here, but the OS result is not there. So these are the unmet medical needs. You have 50% recurrence.
So the other thing is the IM vigor. I mean, if you are talking of Pembro, we should talk of IM vigor. Because both the IM vigor and the ambassador have exactly the same populations.
Yeah.
And both have come negative.
Correct.
So, you know, it adds to your thing that it is a negative way to go.
I think if you look at the ambassador, it has own issues. It was observation versus giving Pembro. It had 30 to 40% patients in ambassador who withdrew consent, and that was huge. And if you look at the post-treatment in ambassador, if around 50 to 70%, 60% received the immune checkpoint.
Later on, the crossover.
No, in ambassador, in crossover.
Yeah.
So if you look at it, open label observation, and all the trial was totally, if I'm not wrong, done in US, where people had access to the standard of care. So what this trial tells me that the PFS is dot at 0.7 around, which all the trials have seen. And when you have access to the drug post-progression, does it affect? And ambassador has a big issue of consent withdrawal, which is 35-40%, which is huge.
Kripa?
So the other thing which Sahu sir is not saying, it is his favorite, he has forgotten, a lot of censoring in that trial. There was a pretty big censoring component in the ambassador.
So the more you dissect, the more, things you get.
But that's
out
the
of it.
same even in the adjuvant RCCs as well. I mean, like, informative censoring was a key concern there as well. And the other thing is, the US versus the European divide. FDA has approved it for all comers, even if you are adjuvant nivolumab. EMA said only more than 1%, PD-L1, for adjuvant nivolumab.
So here, when you have a DFS benefit in the adjuvant setting, there was a thought that why not in the neoadjuvant setting or a perioperative. So this was a network meta-analysis where I found that neoadjuvant chemotherapy may achieve a better PCR and pathological downstaging than NAC or NAI alone. So,
just the point in between the ambassador and the nivolumab.
I think ambassador agreed.
So the point that it hasn't achieved the overall
we don't have much time, so
is because of the when we do the intent to treat analysis out of it, maximum of these patients have received in the first line infotumab or a very potent therapy at the end of the day. So post progression
upfront.
In the post-progression.
Okay.
Post-progression. So that's why the overall survival...
See, that is the problem with the open-label trial, one of the caveats. So coming to the concept of perioperative IO plus CT. Vamsi, your thoughts. Then we can quickly have everybody's thoughts.
So as a concept, it's good. I mean, there's nothing to say against it. And the trial is simple. If you look at the trial, it's pretty straightforward study. Of course, the only question in this is why, if you look at the patient population, they have used good risk and high risk also. So you had a lot of T2N0. This study actually had 95% node negative patients. So that way they have probably selected a better group of population, and it's good that they still got the result.
So, Sau, I would like to have your expert opinion on EFS in this.
I think Niagara positives first. It's a positive trial.
It's a positive trial.
Okay, so there are 2-3 concerns for me with the design. For me, the EFS. There was three things were the EFS endpoints. One was the progression during treatment or post-surgery treatment.
Or recurrence.
Or recurrence.
Yeah.
Second was the death. Third was something that really bothers me is the date of expected surgery who did not undergo radical cystectomy.
Need to elaborate on this.
Yeah, I want to. Because it tells you that I have planned for, as I get it, I want someone to explain it to me further. As I read the article more, there's a planned date for a surgery and someone misses it, it becomes an event, which triggers an event here. And if you look at the persons who have not undergone that, in that percentage, it's around 5 to 6% more in the control arm who have not undergone surgery at that point. So that would have affected the trigger.
And even in those 5 to 6%, the criteria was not, medical always. Criteria was on the, the patient's consent for surgery or not. So even that bias is there. So Kripa, let's go to the next one, PCR. What are your views about PCR?
Definitely, sir. I think the perils of PAT-CR, I think, definitely it's not a great surrogate endpoint. That's number one. And then again, this
for OS, it's not a good surrogate mark.
Correct.
Right.
And then again here, calling it dual primary endpoints. Co-primary endpoints is always a better, because, you're beating both. Dual, you're settling for either one. So I think, that again was another criticism of the trial. And then again, you're choosing out only platinum eligible patients. You're completely excluding all the patients versus platinum ineligible. I think probably, yet another criticism, we could have probably looked at even platinum. There are a lot of trials looking at even addition of IO plus chemotherapy, even in platinum ineligible patients as well, ongoing trials.
The next question, the comparator arm, you gave chemotherapy and third, finished.
That's always, I believe, they haven't included the adjuvant immuno in the comparator arm, and that's also, I believe, a number of...
that design, when they designed it, it would not have been approved, to be fair to the trialists.
When they designed this trial, it was prior to the adjuvant studies.
That's
the problem. So that's why I wanted to ask.
Yeah, so second, the one more thing which Kripa's told is little bit not okay with is they expected 50% in the immune checkpoint and the chemotherapy arm to have a PAT-CR. Unfortunately, it did not touch that mark. And they had to go for a reanalysis of 40 or 50 patients who missed. That was something which was unplanned for. and this trial does not give me one big answer in a maybe country like India, how much component of benefit happens with the neoadjuvant, how much component happens with that?
No, they had clearly mentioned that they are not trying to find out whether the neoadjuvant or the adjuvant. They are just trying to tell you that if you give immunotherapy, is it going to make any change post-surgery? Agreed. So, Vamsi, did it make any change post-surgery?
So again, if you look at the PATH-CR, I mean, when you see a thing, they had estimated a difference of at least 10%, it didn't get it. Then they did a reanalysis, said some patients, we reanalyzed the sample. So, this is not what they were looking for. So that's it. So I don't think PATH-CR really matters. This was something that I did talk about how, look at the number of patients who are T2 in the study.
Yes,
40% are T2 N0,
even the
N1 is only 5%. So, you're taking a very good risk patient, and there you have shown some benefits. So you can take it either which ways. You found a little difference because it is a good risk group, or you can say even in a good risk group, you found a good benefet. So you argue whichever you want.
Kripa, anything on the renal functions, creatinine clearance?
I think this is the split renal dosing that they tried as well. They were pretty adventurous, even looking at even CC more than 40 and giving them weekly.
When you look for some evidence, I think this would be the one.
Yeah, definitely.
So this was the event-free survival. It is there. It was positive. This was the PCR, and we have discussed. They had done a reanalysis and showed that later on probably there was a benefit of about 8 to 10%. And this is the overall survival by the intention to treat population. Adverse events, Vamsi, anything?
By and large, fairly okay. I mean, I don't think adverse events in any immunotherapy is going to really strike us as surprising right now. We are used to immunotherapy.
Anybody else wants to give any? So, Sahu, this question is for you. How would you decide which patient should receive perioperative or adjuvant therapy? IO.
The T and the N does matter. Point number one, the characteristics of the tumor. Second would be the platinum eligible, cisplatin eligible, ineligibility.
So if I were to say that there are a lot of T3 patients in this trial and they didn't make a dent as far as the post surgery results were, how would you argue that?
I didn't get you.
You said that the T stage matters.
Yeah. So what I meant was, anything less than T2 would not receive. That's what I meant.
That point is well.
And that's what I meant. That's what I meant. And the second point is, if someone is not eligible for cisplatin, and I'll be little skeptical till 40, I'll quit at 50 probably in my practice. 40 is little too low for comfort. So those patients probably go for surgery.
So
and then
when the patient comes to you in the clinic, you want to give perioperative. So which would be the criteria? You'll say 1, 2, 3, yes, I will give.
As I told, the tumor stage. Number 2 is the cisplatin. And eligibility. If these two are met, and lot of finances, definitely.
So I think this is where the challenge is, intuitively, you would want a patient who has more disease with a bigger nodal burden to get your perioperative, but that is the patient who has not been adequately covered in this study. So that's a bit of a counterintuitive way of looking at it, but I agree with Saucer. If it is a cisplatin ineligible, matter closed, you directly go for a surgery and adjuvant.
The other thing is, suppose you do the surgery and you find that it is T1, would you give a perioperative or
Why PT1?
Would you
Why PT1 or PT1?
PT1.
Why PT1?
Post surgery, why PT1?
Yeah. Why PT1 I would still continue. Because we have given it with the intent of giving for 1 year. So I won't change it based on the...
Kripa.
I think the question should be, would you rather give it in the neoadjuvant or in the adjuvant? I would look at doing it in the neoadjuvant setting and get to, see if the patient has got a PATH-CR, then probably may not require 1 year of adjuvant.
So would you give perioperative in this patient?
So, as I said that they have used the split cisplatin dosing. I believe that's a real world scenario. They have picked up the patients and they have picked up in the extreme ends to their justification. So they have picked up the T2 patients also, they have picked up node positive patients. So they might have covered a whole spectrum of patients. So, and they have demonstrated a unanimous result.
As a thought process, is it a good thought process or is
your question was
it is a real world practice.
Your question was someone started off with the neoadjuvant component of the Niagara and then achieved a YPT0T1.
That is basically the criticism of this trial. That suppose
as per this, you need to continue.
Yeah.
They did not look at
They
it
did not stop.
No, we cannot stop.
Yeah.
The other thing which we sometimes tend to forget is the UTUC. This did not include UTUC, whereas the others included upper tract. So that's the other place. This is pure bladder.
So these are the advantages. This is the first study to see a perioperative approach. Included patients with UC and some with divergent differentiations. It's a practice-changing trial, but it has its own caveats. PCR, EFS is a problem. Whether this 10% differentiation in PCR after correction would translate into significant differences in OS, that remains to be seen. Should patients get adjuvant durvalumab, these categories, once you have started, you have to continue. ctDNA would be a very good incorporation after the surgery where it would make a dent. And question of what to do after progression, if you lump everything in the beginning, later on, again, that remains a problem. Last word, Kripa.
So I think, just like how we have in lung cancer, we have a trial looking at only perioperative versus, pre-op and then continuing in the adjuvant as well. So there's one positive trial for that as well in lung cancer.
Perioperative, you have a good example in
correct.
stomach. Flot.
Exactly.
Flot is there.
So the only thing that excites me is the ctDNA. If that trial of
it excites you.
of Atizo comes positive, we would be not giving lot of patients adjuvant therapy. So that's what I'm looking at, the data.
I think we're truly in the era of precision medicine and personalized therapy. So risk-adapted escalation and de-escalation of treatment strategies is here to stay. And the ctDNA would definitely go a long way in helping us achieve that.
Anything?
I think so. We are over the time limit.
Okay. So I thank you. I thank the esteemed panelists, and we close the discussion. Thank you.
Thank you. Thank you, sir. Thank you. So with this, I would like to now invite Dr. Shrigopal Mohanty, sir. Sir, I would like to invite Dr. Vineet Talwar, sir, for a group photo session. Thank you, sir. So this is another sponsored symposium by Johnson & Johnson's. I would like to invite Dr. Shrigopal Mohanty, sir. He's a medical oncologist from Bhubaneswar.
So, good afternoon, everyone. So I'll be discussing on a breakthrough in non-small cell lung cancer, the promise of amivantamab for exon 20 insertion mutation. So these are the disclaimers. So as we all know, this the pattern of this lung cancer, most common cancer among the worldwide. And if you talk about lung cancer, we know that 85% constitutes non-small cell lung cancer, and 15% are small cell. Among non-small cell, 40% are adeno, 30% are squamous, 10% large cell, and approximately 10% other variants. So these are the common mutations what we see. The adenocarcinoma, as we all know, EGFR, all-cross, and red mate, and apart from that, KRAS mutations. Among the squamous cell, we see commonly the TP53 mutation, PIK3, the P10, FGFR mutations. These are common. So when you talk about adenocarcinoma, as we all know, there are first, second, third generation TKI for common mutations, whereas for exon 20 insertion mutations, when we see in clinical practice, that's a challenge definitely. And that was the area of unmet need. Now we have a promising drug, amivantamab. I will go through this drug. So this is the NCCN guideline, which recommend that the comprehensive genomic profiling based NGS test upfront while managing advanced non-small cell lung cancer. So as we all know, this NGS will help us to detect this exon 20 insertion mutation. So let's look into this mutation. Basically, there are two types of exon 20 insertion. One is in-frame insertion, second is duplication. And most of the exon 20 insertion, they confer resistance to the typical EGFR TKI, except this helical region 763A locus, this mutation, which confers sensitivity to first and third generation TKI. So how this exon 20 insertion mutation causes the resistance? Basically, it causes the conformational changes in the EGFR binding site. And the binding pocket, it reduces the size of the binding pocket, and it reduces the affinity of TKI. So if you look at these other rare mutations, apart from the exon 19 and 21 EGFR mutations, this exon 20 is the third most common EGFR mutations. And these are the studies, two studies. First one is showing that among the 3,000 patients, this exon 20 insertion mutation presence was showing a poor prognosis compared to exon 19 or 21 mutations. In the second curve, we can see that presence of exon 20 insertion mutation, the efficacy with EGFR TKI was inferior compared to the exon 19 or 21 mutations. So definitely this exon 20 insertion mutation, this cohort of patients needs some novel therapies. It was required previously because in the first line in this cohort of patients, the platinum-based chemotherapy was the standard, and of course the long-term efficacy was not promising. We can see that the median PFS with this platinum-based chemotherapy was about 6 months, and if you look at the median OS also, it was something between the 12 and 16 months. Whereas if you look at other EGFR mutations, we have much better efficacy. So there is unmet need. And again, in the second line setting in this cohort, we don't have any promising options. We didn't have. So if you look at the TKI efficacy in this cohort, so being it first or second or third generation TKI, everything failed to show a promising efficacy. Whereas adding cetuximab to afatinib has shown some efficacy up to the median PFS of 5 months. Again, that is not very promising. We all know that. So amivantamab is the promising drug in this cohort, what we have got in our armamentarium. And this is a fully human bispecific antibody that target EGFR and MET receptors. So this binds extracellularly to these two receptors, so it doesn't get affected by co-mutations in the EGFR TKI binding sites. So three mode of action of this amivantamab. One is direct toxicity by activating macrophage and natural killer cells. Second is by lysosomal mediated receptor degradation. Third is by binding to EGFR and MET receptors. It inhibits the downcell signaling pathway and cell death. So one more advantage of amivantamab is this. This inhibits the receptor crosstalk between EGFR and MET pathway. As we know, there is a receptor crosstalk between this MET and EGFR. By binding to both of these receptors, amivantamab prevents this receptor crosstalk. Let's look at this post-platinum progression in the second line, subsequent line setting, how this role of amivantamab is. This is the Chrysalis study. They have looked into this use of amivantamab. This was a phase 1-2 open-label multi-center study. The primary endpoint was objective response rate. So post-platinum progression, they have compared this amivantamab to placebo, and the baseline demography you can see 50% patients are Asian patients, 50% are non-smoker, and 50% smoker. Here we can see that the objective response rate with amivantamab in second line, subsequent line was 40%. Median PFS was about 8 months, and median overall survival was 22 months. With amivantamab, if we see about only chemotherapy, then we can get some OS of about 12 to 16 months. Whereas here, about 10 months of OS benefit with this, although this is a cross trial, but again, there is some advantage. So if we look at this quantity of benefit, the magnitude of benefit based on where the EGFR exon mutation insertion happens, the most benefit was noticed when the insertion was seen in near loop region. Again, if you look at the long-term clinical benefit of amivantamab in second subsequent line, 40% of patients, they had sustained clinical benefit. Which group derived the maximum sustained benefit was patients with good PS, patients who responded well to the treatment, and those didn't have any baseline RAS/RAF/MEK pathway alteration. Looking at the toxicity, grade 3 hair toxicity was seen in about 16% of patients, and treatment toxicity related to discontinuation was only 4%. So definitely this is well-tolerated drug. And typical EGFR-related toxicity, as we know, that RAS, mucositis, and MET-related toxicities was peripheral edema and hypoalbuminemia was seen. And if you look at the CNS activity of amivantamab, about 11% had a intracranial progression, and 4% had both intracranial and extracranial progression. The median time to progress intracranially was about 4.5 months, and median time to progress to the systemic progression was 5.5 months. In 50% of patients who progressed intracranially after amivantamab, they received SRS, and post amivantamab progression, the median duration of continuation of this drug was about 4 months. So this was the second subsequent line. Now, if you look at the first line use of amivantamab, this is the Papillon phase 3 study, which compared this amivantamab plus chemotherapy versus chemotherapy alone. So this was a phase 3 study, and in the first line setting, advanced non-small cell lung cancer with EGFR exon 20 insertion. The primary endpoint was median progression-free survival by the BICR review. The secondary endpoint was objective response rate, duration of response, overall survival, time for next treatment, and the safety profile. So this is the baseline characteristic. 60% are Asian population. So smokers were 40%, non-smoker were 60%. Now look at this median PFS. It was 11 months with amivantamab plus chemo versus 6 months with the only chemotherapy. Hazard ratio for PFS was 0.40. There was significant PFS benefit. So there was consistent PFS benefit. We can see in this forest plot, there was, irrespective of all subgroups, there was a median PFS benefit with amivantamab plus chemotherapy. And if you look at the objective response rate with amivantamab plus chemotherapy, the 70% was the objective response rate with the amivantamab arm and 47% in the chemotherapy arm. Duration of response, median DOR was 9 months in the combination arm versus 4 months in the chemotherapy arm. The p-value was significant. Time for treatment discontinuation, it was 13 months in the amivantamab arm and 7 months in the chemotherapy arm. Hazard ratio again 0.38. It's very significant, we can see here. Curves are well separated. Now looking at the progression. So irrespective of whether it's a distant visceral mets or nodal recurrence or like any lymph node recurrence or soft tissue. So irrespective of sites, the recurrence rate was lower with amivantamab chemotherapy arm. Time for subsequent chemotherapy, again we see the clear separation of the curve, hazard ratio 0.35. So median time to subsequent therapy was 17 months in the amivantamab arm compared to 9 months in the chemotherapy arm. Now looking at the PFS2, again the hazard ratio is 0.49, curve well separated, very clear benefit we can see here. Interim overall survival, it was not achieved in the amivantamab arm. It was 24 months in the chemotherapy arm. Hazard ratio is 0.67. And there is a trend towards improved OS because you can see the tail end of curve is good, well separated. Now looking at the safety profile. So EGFR associated toxicity, as we discussed, rashes and the mucositis diarrhea. Mate-related toxicities are hypoalbuminemia, peripheral edema. And definitely this is a well-tolerated drug. The major toxicity was the infusion-related reaction. So from this Papillon study, we can see that this amivantamab chemotherapy options is a very good option, which significantly improve the PFS versus chemotherapy in first-line setting in this exon 20 insertion in the first-line advanced non-small cell lung cancer. So this is the NCCN guideline, which says this amivantamab plus chemotherapy is the standard first-line option in this cohort, and amivantamab alone is a second or subsequent line option is the way to go. Now, what is the, the challenges what we face is the infusion-related reaction, which is seen about 2/3 of patients. And out of them, 98% are grade 1, grade 2, which are well managed. This infusion reaction contributing to treatment discontinuation was seen in 1% of patients only. And 95% of this infusion reaction was seen in the first cycle of infusion. So skin nail toxicities, most of them were grade 1, 2, which was seen in about 75% of patients. Grade 3 hair toxicities was seen in 1 to 3% of patients. Now, how to prevent this infusion reaction? This is a study, skip your study, a phase 2 study. Basically, this study, they compared this dexamethasone low dose 4 mg in the 1 day prior to chemotherapy and the before chemotherapy versus dexamethasone 8 mg BD 2 days before, 1 day before, immediately before chemotherapy versus Montelukast 10 mg on the day -4, -3, -2, -1 and on the day before chemotherapy versus methotrexate from day -7 to day -3. So this was basically designed, and low dose dexamethasone, they didn't cross the stage 1. Dexamethasone 8 mg, they crossed from stage 1 to stage 2, then they entered the expansion stage. Montelukast entered the stage 2, whereas methotrexate failed to enter the stage 2. So finally, the 8 mg dose of dexamethasone BD from day -2, day -1 and immediately before chemotherapy, that gave a very promising result. We can see here there was a 3-fold reduction in infusion-related reactions, which was reduced from 67% to 22% with use of this dexamethasone pre-medication. So concluding this study, prophylactic dexamethasone 8 mg orally BD 2 days before chemotherapy, 1 day before chemotherapy and 1 hour before chemotherapy, it's a promising options, which reduces the toxicity by 3-fold. And to conclude, this amivantamab plus chemotherapy is the first-line standard option, and amivantamab alone is the promising options in the second subsequent line setting in advanced non-small cell lung cancer with EGFR exon 20 insertion mutation. Thank you, everyone, for patiently hearing.
Thank you, sir. Now, moving on to our session 3, that is on GI. I would like to invite our chairpersons, Dr. Lakshmaya Kesi, he is a medical oncologist from Bengaluru. Dr. Lalatendu Sarangi, he is a surgical oncologist from Bhubaneswar. Dr. Bharat J. Parekh, sir, he is medical oncologist from Ahmedabad. And Dr. B. K. Smruti, sir, he is a medical oncologist from Mumbai.
Good afternoon. Dr. Sarvajit Mukherjee, medical oncologist from Miami. He exclusively practices GI oncology. Going to speak, where are we, what is the status and the current future?
Thank you, Dr. Sarangi, and it's a great honor to be able to present here. I'm currently at Roswell Park in Buffalo, New York, where I'm an associate professor of oncology, and I also co-lead the GI translational group, but I'm making a transition, as you just heard. So the topic of our discussion is immunotherapy in gastroesophageal cancer. Where are we now and what's in the future? So keeping in mind this year's theme of the conference, Immuno-Oncology Choosing Wisely and Precisely, we are going to talk about biomarker testing and biomarker-guided therapy in upper GI cancers. We will talk about some of the challenges and the future directions. Now, the most important biomarker for immune checkpoint inhibitor therapy in upper GI cancers is PD-L1 testing, and we will be using the terms CPS and TPS quite a bit, so let's just refresh our memories. So CPS actually looks at PD-L1 positive cells, tumor cells, lymphocytes, and macrophages, and they divide them by viable tumor cells, whereas TPS looks at PD-L1 positive tumor cells only. So you can see that there's a little bit of discrepancy, and different trials have used CPS and TPS, as I will elucidate in my lecture. So over the past few years, we have seen a lot of approvals of immunotherapy drugs in upper GI cancers based on some of the landmark trials, both in the metastatic as well as in resectable setting. In my talk, I will talk about some of these trials, and we will then further delve into biomarker analysis and subgroup analysis. So let's start with CheckMate 649 that led to approval of nivolumab in gastroesophageal adenocarcinoma. This was a global open-label phase 3 study. They included patients with advanced or metastatic gastric, esophageal, or G-junction adenocarcinoma. Patients could not have a known HER2-positive status. Patients were randomized to EP-Nivo, Nivo plus chemo, or chemotherapy. EP-Nivo arm was negative, so I'll present data on chemo versus Nivo plus chemo. The study had dual primary endpoints, OS or PFS and PD-L1 CPS score greater than or equal to 5, but they also looked at OS and PFS in all randomized patients as well as those with a PD-L1 CPS score of 1 or greater. And here are the results. You can see that there was a survival benefit in all randomized patients with a hazard ratio of 0.8. But then if you look at biomarker enriched populations, PD-L1 CPS score greater than or equal to 1, and the hazard ratio improved to 0.77. And then if you look at those with a CPS score of greater than or equal to 5, the hazard ratio further improved to 0.71. So the FDA approval was for all comers, regardless of the PD-L1 CPS score. But if you look at the NCCN guidelines, you will see that category 1 recommendation have been reserved for those with a CPS score of 5 or greater. And several European guidelines also recommend using the checkpoint inhibitors in those with a CPS score of 5 or greater. So again, we have to choose very wisely, especially in resource-limited settings where cost is an issue. Now, moving on to HER2-positive disease, KEYNOTE-811 study was again a global randomized study that included patients with gastric or G-junction HER2-positive adenocarcinoma. Patients were randomized to receive chemotherapy plus trastuzumab, which has been the standard of care in this population for over a decade, versus chemo plus trastuzumab plus pembro, and dual primary endpoints were overall survival and progression-free survival. As you can see here, there was a benefit among all comers, and those with a PD-L1 CPS score of 1 or greater had even better outcomes. Initially, FDA gave this drug an accelerated approval. It was approved for all comers, but then based on a further subsequent analysis where they did not see any benefit in those with PD-L1 CPS score of less than 1, the indication was reversed. So these days, we check for HER2 as well as PD-L1 before adding pembrolizumab in this specific patient population. So far we have talked about adenocarcinoma, but immunotherapy is effective in squamous cell carcinoma as well. Checkmate 648 was a randomized global study that included patients with esophageal squamous cell carcinoma in the metastatic or unresectable setting. Patients were randomized to receive nivo plus chemo, nivo plus EP, or chemotherapy alone. As you can see that both immunotherapy arms improved overall survival compared to chemotherapy alone without significantly increasing grade 3 or grade 4 toxicities, as you can see here. Similarly, if you look at all comers, there was a benefit. However, if you look at the TPS score of greater than or equal to 1, the benefit was even greater, both in terms of overall survival and progression-free survival. No talk is complete without discussing MSI high subgroup. As you know that this subgroup across histology benefits from immunotherapy. So one of my colleagues, Dr. Joe Chow, did a subgroup analysis of some of the first generation immunotherapy trials in upper GI cancers. And he showed that patients who have MSI high status benefit greatly from the addition of pembrolizumab, as they say that if you can drive a truck between the Kaplan-Meier curves, it tells you that it is effective, and that's exactly what's happening here. So far we have learned about 3 biomarkers, HER2, PD-L1, and MSI high, and it is extremely important to check for these biomarkers. At our institution, we routinely check all 3 biomarkers from the get-go. Now, there are some challenges, as you all know, especially with PD-L1 testing. CPS scoring, we spoke about different cutoffs, 1, 5, 10, etc. But CPS scoring is somewhat operator dependent. So a CPS score of 3 to one pathologist would be CPS score of 7 to another pathologist. At our institution, we have implemented a policy where pathologists don't give us a specific number, they give us a range, like 1 to 5, 5 to 10, or greater than 10, things like that. So we'll always have to take it with a grain of salt. Secondly, PD-L1 expression is a dynamic biomarker. One of my former colleagues did analysis to show that PD-L1 expression has temporal and spatial heterogeneity. So it depends on whether you are getting the samples from pre-treatment biopsy versus post-treatment tissue, whether getting it from primary or metastatic site. Thirdly, PD-L1 expression is assay dependent. As you know that there are several approved assays, including 22C3, which is what we most commonly use, SP142. And although there's a general concordance between these assays, they are not all the same. So we have to be very careful about how we interpret these results. And last but not the least, we see this quite a lot, that biomarker testing is not routinely done. So as a group of academic oncologists, it is our responsibility to increase awareness about biomarker testing for both treating clinicians as well as patients. Now we will switch gears quickly to talk about immunotherapy in resectable setting. As you know that the standard of care for a long time has been chemoradiation in squamous cell cancer. For gastric adenocarcinoma, we have been using perioperative chemo. In Asia, sometimes we do upfront surgery followed by adjuvant chemo. And for G-junction, the practice has been somewhat mixed. Some patients get chemoradiation, some patients get chemotherapy. But of course, that's going to change following the ESOPEC study. Regardless, you can see that despite advances in multimodality treatment, the cure rate at 5 years tend to be less than 50% for the most part, at least in the Western population. So there's definitely room for improvement. So since immunotherapy showed great promise in the metastatic setting, investigators started testing it in the adjuvant setting as well. So Checkmate 577 study was again a global randomized study that included esophageal adeno as well as squamous cell carcinoma patients. Patients received neoadjuvant chemoradiation followed by surgery, and those who do not have a pathological response, which is about 80% of our patient population, have a high risk of relapse. So these patients were included in the study, and they were randomized in a 2-to-1 fashion to receive nivolumab versus placebo. The primary endpoint of the study was disease-free survival. And you can see here there was nearly doubling of disease-free survival from 11 months to 22 months. However, if you look at the specific histologies, you will see that squamous cell population benefited much more than adeno. And on a further subgroup analysis, the authors showed that higher the PD-L1 expression was, more the benefit was from adjuvant immunotherapy. So based on this study, adjuvant immunotherapy or adjuvant nivolumab became standard of care for this patient population. Now, based on the success of adjuvant nivolumab, trials started using it in the neoadjuvant setting, like every other cancer. Unfortunately, we haven't seen that much success in this patient population. EA2174 study that used nivolumab concurrently with chemoradiation was a negative study. Similarly, Attraction 5 that used nivolumab along with chemotherapy in the adjuvant setting was also a negative study. Vestige study took a risk adaptive strategy and they didn't find any survival benefit. Keynote 585 used immunotherapy in the perioperative setting along with chemotherapy, and although immunotherapy improved PAT-CR, it did not translate into survival benefit. So we are currently waiting for the final results of 2 other studies very similarly designed as Keynote 585. One is Matterhorn study and the other one is Dante study. Initial results showed improvement in PAT-CR, but we are yet to see if that leads to survival benefit. And just like in the metastatic setting, MSI high population responds very well to immunotherapy. Neo-NPGA study looked at MSI high T2 to T4 gastric or G-junction cancers. These patients did not receive any chemo. They received EP-Nivo in the frontline setting, and they went on having surgery, and they saw that the PAT-CR rate was 60%, and at 12 months, there were 30 out of those 32 patients who were alive and without relapse. So in the MSI high population, we are not even using chemotherapy these days. There was similar data from Durva-Tremi as well, so we have been using immunotherapy in this patient population. Now, we have so far talked about survival and PAT-CR, all clinical endpoints, but the question is whether immunotherapy improves quality of life. So we did a meta-analysis of 11 randomized control studies involving 820 patients. We saw that when combined with chemotherapy or alone, immunotherapy improves several health-related quality of life metrics, including pain, dysphagia. So we can be more confident in using immunotherapy in the right patient population. Now, future directions. We know that immunotherapy modestly improves survival. However, resistance is common, so we have been looking at different mechanisms. One of them is co-expression of different checkpoint inhibitors. We looked at the bulk RNA-seq data from our own patient population, and we saw that several co-stimulatory and co-inhibitory checkpoints are expressed along with PD-L1 and CTLA-4, as you can see here. So one possible strategy could be combining multiple checkpoints. For example, TIGIT has shown great promise in this situation. Similarly, different targeted therapies like FGFR2-directed therapies, anti-claudin therapies, or even VEGF TKIs are being investigated to overcome resistance to immunotherapy. I would like to finish this with saying that so far our talk has focused only on checkpoint inhibitors, but there are several types of immunotherapy that are being tested, including bispecific, T-cell engagers, oncolytic viruses, CAR T-cells, vaccines, ADCs. So there's really exciting time ahead in this field, particularly for upper GI cancers. So in general, we would say that immunotherapy is indicated for the right patient population, but biomarker testing is the key, and we should advocate for immunotherapy in the right patient group. With that, I would like to end my talk. Thank you all for listening, and I would be happy to take any questions in the future.
Thank you, Dr. Sarabjit Mukherjee. That was an excellent talk of how immunotherapy has made inroads into the upper GI cancers. And I think the questions, is there time for questions, or we would take it after the panel discussion?
Yeah, ma'am, there is a time for question and answer after the panel discussion.
Okay.
I think, Sarvati, you stay back. Okay. There's a time for the question and answer after the panel discussion. Thank you.
Now we'll go for the next session, panel discussion, case-based GI cases. Moderator is Dr. M. Vamshi Krishna, Department of Medical Oncology, Hyderabad. The other panelists are Mohit Sharma, Medical Oncologist, Delhi, NCR. Gunjesh Kumar Singh, Department of Medical Oncology, Ranchi. Avinash Rakli, Surgical Oncology, Mumbai. Rajesh Shinde, Surgical Oncology, Mumbai. Sirish Alurkar, Department of Medical Oncology, Ahmedabad. Lipista Samasti, Medical Oncologist from Bhubaneswar. Siddharth Turkur, Medical Oncologist, Raipur. Vinit Gupta, Medical Oncologist, Bangalore. Chira Rajan, Radiation Oncologist, Bhubaneswar. Henrik Karl Justin, Department of Surgical Oncology, Sweden. Sangram K. Panda, Department of Surgical Oncology, Cuttack.
Good afternoon, everyone. I'm reminded of that scene from Sholay where Gabbar Singh says, "कितने आदमी थे? इतने लोग और मैं अकेला, ये बड़ी नाइंसाफी है." So, but we will try to discuss. I have 4 surgical oncologists, I have a radiation oncologist, and 6 medical oncologists. So we will discuss something which is coming to all oncologists, not only medical oncologists. Okay. So we will go with the first case. Is all about ctDNA. Has the genie been let out of the bottle? MRD testing in CRC. So case 1, 82-year-old gentleman, diabetic, CAD on meds with a normal cardiac function, COPD meds. He had melena, evaluated, cecal mass, non-metastatic on PET, underwent a right hemicolectomy. So surgeons are all there on the stage. 82-year-old, any issues with operating for anybody? All of them are happy to operate a 82-year-old, absolutely fine. Great. So this is the report. Cecal mass ulceroproliferative, it's a T3N1A. So I'll start with Dr. Avinash sir. So Dr. Saklani, patient comes to you and says, Doc, you have removed it. It is T3N1A. What do I do now? I think we need a few more mics here. I can see only one mic there.
I think stage 3 cancer, the standard of care is adjuvant chemotherapy. Obviously, for its risk and age, the degree of benefit and risk has to be done. So you will refer it to a medical oncologist.
So you will refer it to your medical oncologist for chemotherapy. Excellent. Dr. Henrik, would you agree with this? 82-year-old guy, he's fit. You operated him, obviously. Would you say, look, you are 82, the benefit of adjuvant chemo is 3 to 5%. The side effects of chemo are 3 to 5%. That's what we all know. How would you take it?
So if it's a frail patient, definitely not. But
you have operated him.
Yeah.
He's not frail. You know, he underwent a heavy
Usually
colectomy.
it ends up with adjuvant chemotherapy, even in Sweden. Yes.
So you would advise an adjuvant chemotherapy. Wonderful. Mohit, your name is on top, so yeah, you're there. So Mohit, patient comes to you. Would you say, yes, my surgeon has sent, I cannot disappoint them, I will give chemotherapy?
82 years old, I think here comes the role of ctDNA.
Okay, you will do a ctDNA.
Yes.
Okay, very good. Do the other medical oncologists agree on the panel? Dr. Gunjay?
I think surgeon said he will let medical oncologist decide whether to give chemo or not.
Fair enough. Dr. Gunjesh? ctDNA, Mohit says, would you agree or would you say, no, you have undergone a surgery, we will go ahead with chemo?
So if possible, sir, ctDNA. Otherwise, if patient is fit, I'll go for either Capox or single agent 5FU is okay for me.
Single agent capstabine in a stage 3. Any data for that? Yes,
More
single
than
agent
70, the patient is more than 70 years old, that the benefit of oxaliplatin is already doubtful.
Okay.
And the patient is frail, so I do. We are just.
He has undergone a surgery.
Probably 82 years old, there is non-cancer related mortality risk is also
Okay.
high. So I don't want excessive toxicity. I would be on the side of not giving the.
You would say not giving. Okay, so you can see I have put 5 topics. You can
Vamsi,
do. Yeah.
Any MSI data on this?
MSI stable, sir. You can go, so MSI is stable and there is no, there's a pathogenic mutation in KRAS and TP53 and APC for whatever birth that is. Right, so I have given you options here. Dr. Alukar sir, since you asked, you can do a ctDNA, Guardant, Signatera, whichever else you want, and decide therapy based on the report. You can do a ctDNA and say, look, I'm doing it, but I will not give you any therapy. It's only prognostic. You will do a ctDNA, but still treat irrespective, like Gunjesh says, that I will give chemotherapy. You would not do anything, only observation, or would you not do ctDNA and observe? Treat. What would be your choice, sir?
So doing ctDNA is a dicey,
Okay.
test because there are a lot of pre-analytical issues there.
Okay.
How you centrifuge it, when you take the sample, how soon you centrifuge it. The recommendation as per the cap is 4 hours. So in a clinical setting, I don't know in hospital how much it really occurs, 4 hours. So that is, so I would maybe do a ctDNA, do a serial ctDNA later and see if at all there is a rise.
Okay. Dr. Lipshita, your take on this. And what would you do in practice? This is a real patient who came to me some time back. This is not an invented case.
Sir, in practice, I prefer giving single agent capecitabine.
Okay. Single agent capstabine.
Yes.
All right.
Sir, if the patient was frail and would not be able to.
He has undergone a hemicolectomy.
Yes, so
I
it's
think
not
all medical
frail.
oncologists are more scared than surgeons. That is what this panel has proven. We are more worried about the age and the frailty. You know, they went and operated. So,
Yes, sir.
you know.
So the ctDNA has a role, but the problem is if the ctDNA is negative and the recurrence happens, so that is the area where we
Okay.
will be dicey.
Okay.
So doing a ctDNA is.
So you
not
would
a
go
good
with
thing.
probably no ctDNA and treat, if I can understand correctly. Dr. Vineet sir, your thoughts on this? There's no 100% right answer, obviously.
Right. So in my practice, I would probably, if he's actually fit enough to undergo a surgery, give him a Xelox.
You would give him Xelox. Okay.
Because
Any
if he's actually
ctDNA?
fit enough.
All right.
Now for ctDNA, I'm not using it in practice
Okay.
because the role is really prognostic. It's not predictive. So it's not predictive of benefit for the therapy.
Okay.
So it's prognostic, yes, but
Okay.
I'm not using it in routine practice.
You are not using it in routine practice. Okay. So I'll come to the next question, Dr. Vineet, since you're from Bangalore, the city of startups. I think all of us get at least 5 people coming and telling us, oh, we have a ctDNA which is as good as anything there. Now, all of us are aware we have Signatera, which is a tissue-based testing, where you actually do the ctDNA on the tumor sample and look for it, or you have a tumor agnostic like the Guardant. And multiple Indian labs are doing multiple tests. So what would you prefer, as just as an example?
I'm not using it in practice.
No, but if
I have no
supposing
preference.
you don't use it. Okay, Mohit, you said you will use it. you said you will use it, so I'm asking. So some Indian lab comes and says, look, we are as good as Signatera or Guardant or whatever. Do you agree with that, or would you like only the, the approved one?
If I'm doing ctDNA, I'll prefer sticking to Guardant or...
If you are doing it, you would prefer sticking to the... Okay. Alurkar sir?
Guardant would be the choice.
For you, Guardant would be the choice. Excellent. So again, there's a very interesting thing because we have now data which says that when you have MRD negative patients, at 4 weeks, you actually have no benefit, and this was in stage 2 and 3. This is a paper from Nature Medicine in 2023, and they said that who were MRD negative, they had no benefit from adjuvant chemotherapy. The risk reduction is only 3.4%, which is not a huge number at 18 months. This is in high-risk stage 2 and stage 3, whereas MRD positive patients did have a big benefit. So I take the point of Dr. Lipshita that if it comes negative, you're probably safer. If it comes positive, you have committed, you have to give chemotherapy after that. So in essence, if somebody said 82, we would just give CAPE, we will not give Xelox, you might as well not do the test and go ahead with your test. You're not wrong in doing that because you have decided to give the patient treatment. You do this test, the later question would be, why did you give only a CAPE? You could have given a Xelox. You could have done the full treatment. So I think that's something we need to remember when we discuss this. This is the Pegasus study. Now again, this is a post-surgical biopsy guided treatment. So Dr. V, this is also predictive now. It's not just prognostic because people are using it to decide how to treat. And in stage 3 and high-risk colon, what they found is they gave the adjuvant phase for some patients, then decided whether to give CAPE or not based on the liquid biopsy. And they found ctDNA positivity of around 26% in all the patients. In low risk, it was around 18 to 20%. In high-risk stage 2-3, it was around 25%. In T4, it was as high as around 56%, but only 14 patients. Yes, please go ahead. Mike, please.
So Yuman Fang's group from City of Hope just published a few weeks ago a serial assessment of MRD assessments, right? Have you read that paper from JAMA? And what they showed was that MRD positivity led to only 1.6% increased cure rates in patients with directed therapy, or when they were actually operating on metastases for a curative intent.
Okay, that's oligometastatic. We are talking of stage 2 and stage 3.
Right. So the point I'm trying to capture is that if in the metastatic setting the benefit is so minuscule, the benefit when it comes down to adjuvant is going to be far lesser in quantum. So that is the point I was trying to make.
Point taken. So again, I think we have to be a little cautious when we are doing this. Again, doing it in every case, we have to discuss with the patient what we are doing before deciding. This is another paper which was the, forgetting the name, this is the Guardant study which looked at stage 2 colon therapy. Now in this study, they had only 41% high risk, 59 were actually low risk. And then when they looked at the primary side, the T3 was 86%. So less of T4, mostly T3. And in this study, they found that they could reduce the reception of chemotherapy from the oncologist. So number of patients who received the chemotherapy was much lower in the ctDNA guided group with no difference in the outcome. And 92% and 91.4%. So T3 is mostly N0. So this group, you do get some benefit by doing the test. That's what this paper showed. For patients who had negative ctDNA, did much better than the positive ctDNA. So what did we do? We discussed all this with the patient. Patient happened to be from Dubai. They had come down for a surgery. We said ctDNA, it came negative, and we have kept the patient on observation. We have not given patient chemotherapy. So again, based on all this, but I think everyone has a point that you should discuss these issues before doing it. As of now, I am not doing any lab that comes in. I would like some more validation because it is not an easy test to do. Guardant does something called a methylomics, and they look at epigenomics, which is not seen by every lab. So I am a little cautious in doing it. So that is the case one. Now this is a 54-year-old gentleman, diabetic, history of RTA, multiple bone fracture. Yeah. Yes, yes, please.
In this case, he was an 82 years old male.
Yeah.
If he was, say, 60 years or 65 years,
yes,
then also we would have done the same thing, only ctDNA. Or
T3, N1A, I would have given chemotherapy. I wouldn't have done the ctDNA. Because again, the idea comes that if it comes negative and you don't give chemo, there is still a chance he may relapse, like Dr. Lipshita said. So unless, you take it in writing, video consult and so on and so forth, and something happens, you're always at a risk. But I think that's the way we are going to go forth. The idea is you do serial ctDNA. So again, that's the next thing. Just don't do one ctDNA and decide your treatment for the next. The patient has to be prepared for serial ctDNA. If they are willing for that, then still it is an option. But it is not the standard of care as of today, at least I believe so. Right, so this patient came with a CS sigmoid with 4 bilobar liver mets, MSI stable, all RAS wild type, FOLFIRINOX 4 cycles, significant resolution in sigmoid. So surgery was done. Surgeons agree with this plan. 4 bilobar liver mets, you would be happy to operate later and do all the metastatectomy. Patient underwent metastatectomy, good response in the sense he recovered very fast from surgery. YPT3N0, liver mets were not bushed there, but completely resected without any, margins positive. Plan was for more cycles of chemotherapy, so I'll ask the question again. I will not ask the surgeons this time, I'll ask the medical oncologist. So starting with Alurkar sir, again, what would you do? ctDNA, decide therapy based on the report. So if positive, you would intensify and say, I am adding targeted therapy. Remember, we have not given targeted. Or you do a ctDNA, but only do it after your entire 8 months is over, 8 cycles is over for follow-up. Or you just not do that and follow up conventionally with CA and imaging. So
I think I would go with the chemotherapy only.
Only chemotherapy, no
I think
ctDNA.
There is no data concerning the correlation between ctDNA use of anti-VEGF or say.
So we are not using anti-VEGF here. We have not used
targeted therapies. I'm saying targeted therapies, yeah. But there's no data correlating. There's more data with chemotherapy.
Okay. Mohit. No ctDNA, only chemotherapy. Gunjesh?
So, no ctDNA that.
Siddharth, no ctDNA.
In Altair trial also this.
Which one?
In Altair trial recently presented, they have seen with ctDNA monitoring and giving trifluridine in those patients, it has not.
It has not worked. Okay, understood. So intensifying did not work, so you would not do it.
So complete chemotherapy in monitoring, we can do some ctDNA because it can show early rise in comparison to CA and radiologically also. So
what would you do if it comes positive? Would you change chemotherapy if it came positive?
If it came positive, I'll be more vigilant and we'll discuss with the treatment, we'll discuss with the patient if there is any symptoms and we can start only if there is radiological or clinical progression.
So Dr. Vinny, this is exactly the case you were saying. So you would say that no, it's only 1.6%, I will not do it. Exactly. Dr. Lipshita, your patient does it after the chemotherapy is over and it is positive. We know that it gives you a lead time. It actually doesn't give you any interest in it. If it comes ctDNA positive, what do you do? Patient goes to Bombay, Delhi, or even Hyderabad, some medical oncologist does it, and you cut with the report. What would you tell them? Go back to Mumbai. Easiest answer. Come back to Bhubaneswar. Okay. So I think that's always a challenge because patients do such tests and then they come back. So this is the CIRCULATE study. Now, CIRCULATE study actually included stage 4 patients who had undergone surgery. So again, just to kind of bring out that these patients who are oligometastatic were included in the ctDNA studies, and there also ctDNA is predictive of a good response. It's prognostic for a good response and pretty big numbers, not small numbers. So for this patient, actually we have sent a ctDNA which came negative with all the caveats, and I don't say that if I have done the exactly right thing. You cannot do, it is fine. Because again, we are always in an issue as to what do we do if it comes negative or positive. So how do I treat as of today? This is purely my personal view. I would go with a T1, T2, N0, no ctDNA for sure. T1, T2 with node positive N1. T3, T4 and N0, probably yes, I would at least offer the option to the patient saying that there is something called a ctDNA which you can do. It may suggest something. If it is T4 with a node positive or a frank T4, I would probably not do it because like everyone says, you tend to give chemotherapy there. And finally, if it is metastatic at this point of time, no. Oligometastatic, I would tend not to do it, though I have done it in one patient. So this is my case. So any of the panelists would like to agree, disagree, or fully agree? Yes, Gunjesh.
Important point is, sir, like in breast cancer, we are doing this Oncolytic DX, like T2, T1, N0, and ERPR. We are skipping this chemotherapy. So in that scenario also, stage 2, low risk, if ctDNA is negative.
Stage 2, there is no, those stage 2 people have not been included in the clinical trial for the ctDNA. The low risk patient, anyway you will not give chemotherapy.
So almost, sir, in that paper, that NEGM paper, in the standard risk also, the patient received
high risk stage 2.
11% of patient received, the low risk patient
Correct.
received chemotherapy also.
Correct.
So they concluded that those 11% of patient can those who received chemotherapy can be omitted chemotherapy in those cases.
So in general, T1, T2, N0, you not give chemo any which ways in a colon.
Correct, sir. Correct.
So that you don't do a ctDNA. My point, if you're not giving chemo, don't do ctDNA, sir.
Vamsi, you're such a rich hospital. All of these are just cohorts of patients. So ideally for your hospital, which is so rich, you could do ctDNA for everyone and then you could use it for prognosis. And India is such a big country and then you could get more data than just out of this 2,000 patients. Sorry?
TMH has to do it and tell us what to do. I think as a private hospital where the patient spends for it, it is always difficult to, do it something and then say, no, we are not going to do anything. So T1, T2, I'm going to tell, no, I'm not going to give you chemo. And then I do the test. I may get a false negative. I may get a false positive. It is existing. So those are the reasons we don't. Right. This is a client one. Okay, I'm calling him a client because he's a 51-year-old gentleman. He's a CTO of a company. So knows everything about tech. No family history of cancer, no smoker, no complaints, BMI is good. Wants a routine screening for malignancy and says that, look, I don't want to do a colonoscopy. I mean, if you insist, I will do it under sedation, but otherwise I don't want. Would anyone at this day today recommend a CT-based DNA testing for colorectal cancer? I'll go back to the surgeons because this patient will come to you. Dr. Henrik, Sweden is full of internet startups. It is famous for internet and startups.
But you consider ctDNA a screening method then?
Yes.
Well,
no?
No.
No. Okay. Dr. Saklani, there is an NEGM paper, which is why I'm asking.
I think in Wales, actually, they're doing it as a part of a pilot project. So it can be done as a part of pilot project to trial,
Not
but
routine.
not as a routine.
Okay. Dr. Rajesh, would you do it or would you say
I think I would prefer a fecal immunohistochemical test over
fecal
ctDNA.
immune test rather than and a colonoscopy?
Yeah.
Vamshi, can I say something?
Yes. Yes, sir.
Let's say you detected something, some marker of colorectal cancer. How do you localize that tumor? Why are you asking the surgeon?
Perfect, sir.
So just doing a marker for a cancer is pretty useless if you cannot visualize and you cannot act on it. So in a colon cancer, even if this man owns half of India, let us say, and you do the test and say your ctDNA for colon cancer is positive, you are only going to introduce him to start selling away shares. So tomorrow morning, if you have invested in his company, your shares will only drop to the rock bottom. Okay. Otherwise, it's not of great help to do this because you can't localize where the tumor is.
Point taken, sir. There's an NEGM paper which has come out and potentially FDA is approving it. So that's the reason I brought it out for us to discuss. Dr. Vineet, yes. Yes.
So in defense of this question, you would be inundated by CTOs and large money bags who will come to you and want to reduce and mitigate all the probability and risk of cancer in their bodies. And that is why whole body MRIs, whole body CT scans, and pan-genomic testing is becoming such a rage in Silicon Valley. To that end, I think I would, if they are willing to understand the pros and cons and the caveat emptor principle of medicine, I would offer this test to him with that notion.
Okay.
Absolutely.
So that's exactly, yes.
As a surgeon, I will tell, if patient is, there is no family history, nothing, and patient on for screening purpose, why circulating DNA? Why not a virtual colonoscopy?
He will show you this article.
Patient deny for colonoscopy.
He will show you this article. This is an article from NEGM. It is not an article.
I understood, but in a practical basis, why circulating DNA? Why not virtual colonoscopy? You can do virtual colonoscopy.
You can do virtual colonoscopy.
Yeah, it's very simple.
Okay. We just introduced something called a swimming pill, which goes inside and swims around and photographs the entire stomach. So, capsule endoscopy is there.
Screening only, sir.
It's only for screening.
Not taking biopsy, only screening. Why not virtual colonoscopy?
So we had a very interesting discussion. So I'll tell you, we had a very interesting discussion in a group which said that a simple blood test is so much easier than doing a pill or a colonoscopy. So, that's the argument. So, sir.
I think, Vamsi, I'll help you out. A screening tool should be one which should be able to screen with good sensitivity and should have no morbidity. Okay. Even for colonoscopy, perforation rate is 1 per 1,000, which is quite a thing. CT colonoscopy in India, it's not very well developed. It is good software. You're talking of quite a large section of population, not possible to do.
Yes.
So, and fecal-based test, the uptake in India is very low. Okay, even in the Western world, it's about 50-55%. In India, if you ask someone and say to study a stool sample, it's not going to happen. So I am with you, Vamsi. It has a place somewhere.
Okay, we can replace. I have one supporter. Okay, so again, I am personally a bit on the fence because this paper came in NEGM. Now, what they looked at, now interestingly, they evaluated 7,800 patients in which 65 had colorectal cancer, 1,116 had advanced precancerous lesions. So that's almost 1 out of 6 patients who had an advanced precancerous lesion. You're not catching cancers, you're catching precancerous lesions. The sensitivity for a stage 1 cancer is around 11 out of 17, 87%. Sensitivity for stage 2 cancers was 100%. Sensitivity for a stage 3 cancer was 100%. In this cohort.
Which subsite of the colon was most sensitive to this test?
I'm sorry.
Which subsite of the colon means cecum
I
or
don't think that
or
really makes a
descending
difference.
sigmoid?
How does that make a difference?
Vamsi, yes, how here? I think this slide only tells you that this test should not be promoted. Go back.
Yeah, Vamsi, one more.
Go, yeah, this one. just look at, you are doing a screening test. And what I
the
want
definition
is,
of a screening test must be interpreted differently if you take this definition from NEJM. At least the editor of this NEJM article would not have understood what is the meaning of a screening test. Detecting stage 1, stage 2, stage 3 cancer doesn't mean it is a screening test. That's a stupid definition of screening test.
And second
point,
so
you can write that back to the NEJM article which you are quoting.
And the second thing is,
if I can, Dr. Sahu, one second. If I can answer Dr. Raghu, 1,116 had advanced precancerous lesions. The definition of a screening test is you should be able to detect precancerous lesions. However, this is what happened in the test. The test had a positive ctDNA in only 83% of, only 13% of advanced precancerous lesions. So if somebody wants to debate on this, saying that you are not catching precancerous lesions early enough by doing a ctDNA with only 13%, I agree with them. Sahu sir, your
can
point.
I say something here?
Ma'am, Sahu sir and then ma'am.
Yeah, so that was one thing which you spoke. The second thing is here it just tells you that the higher the bulk of disease they're catching more DNA. So it's not a screening modality. They're pushing the bar too low, and the sensitivity of 80% for the screening is just not acceptable. And with the 1 lakh cost, it is just not.
Okay, so I think that 1 lakh cost we should remove.
So I'm removing.
Yeah.
Even the
remove the cost because in India it is cheap. In Sweden, I'm sure the cost of mammography is also very high.
Even the science does not support this.
Fair enough, sir. Dr. Sangram.
Sir, actually, in this patient is asymptomatic.
He's not a patient. He's an absolutely asymptomatic person.
Patient has no family history. So for screening, mostly for early detections. If stage 1, sensitivity 65%,
85.
65%. See, sir. In stage 1, it's 65%.
Okay, 65. Fine.
And stage 2, it is 80%. So it is not acceptable at all for stage 1, stage 2 for screening, rather than you can do a simple CT scan of CT colonoscopy and see CT colonography.
How is CT colonography simple?
Yeah,
how
if
good is
if you are suspecting there is a lesion, it can be seen on like it's a CT scan. Or else you can do like
so I think like Raghu sir said, we should understand what a screening test is.
Because sir, in screening colon cancer screening, there are 5-6 modalities. You can
Fair
go
enough.
through anything rather than ctDNA. I don't agree.
So I think we are not understanding the context in which this question is being put. The question is, people like Elon Musk do not want colonoscopies. They do not want fecal tests. They want to go to pre-nuvo. They want to go to fast forward. They want to go to healthcare incorporated that would do such tests. And these tests are out-of-pocket tests.
Sir, unfortunately, Elon Musk has to undergo a colonoscopy
no,
if that
but
is positive.
If
that is
correct.
positive.
We understand
to prove
that.
the cancer, he has to
So
undergo
Dr.
a
Sahu,
colonoscopy.
again, the point what I'm trying to make, I by the way support all of you who said don't do it because I believe 13% detection of a precancerous is very low. So I don't think this is ready for prime time, but I just brought it out to see the reaction and at least some people have Madam.
See, first of all, as I said, any screening test, you have to see the cost effectiveness, even though you say discount. Second thing is, if it comes negative, say Elon Musk you're talking about, are you going to say, yes, you're negative, so
Yes.
I will not do a colonoscopy, and tomorrow he lands up that he had a precancerous lesion? What is your answer to that? So are you going to agree based on this that Elon Musk should not undergo colonoscopy? So I think this is a study which has been presented in NEJM. It is thought-provoking, but I don't think it replaces.
If I'm not mistaken, it has been approved by the FDA also. If I'm not mistaken, it has been approved in the US. So that is that. Okay. Jumping ahead.
The second one, in screening as well as your high-risk adjuvant setup, if ctDNA is negative, somebody mentioned about the serial testing.
Yes.
So what is the frequency?
Nobody knows, sir. As of now, nobody knows. Maybe 6 monthly or maybe annually. Annually is what they are recommending. Right. So this is a 46-year-old gentleman, CA pancreas, T3N1. He was operated. Obviously, Whipple's was done. Folfirinox adjuvant for 6 months. 8 months later, single mesenteric mass, rising CA 19.9. We couldn't sample it. 2 months, within 2 months, developed ascites, weight loss. Now has a lot of peritoneal thickening, CA 19.9 rising. So we are treating it as a relapse. We do not have enough tissue. We have sent the earlier tissue, and since patient wanted, we sent a liquid biopsy as well. This is the report. Now this liquid biopsy shows a KRAS, which is what we normally get, TP53, a TMB of 21, and an MSI which is stable. Okay, so I think this time I'll ask the surgeons to step back. I'll ask only the medical oncologist here, because this is a pure medical oncology question. So my question, starting from this end, Dr. Vineet, MSI high, TMB high, MSI stable, would you act on this report? It's a liquid biopsy, by the way.
Pembrolizumab has been given already in the past?
No, it is only Folfirinox given. Patient has come to you, this is the report.
Yeah, Pembro is approved for this.
Can,
Pembrolizumab is approved for this.
for TMB high?
MSI high.
No, it is MSI stable. MSI high not detected. So that's the whole reason why I'm putting this question.
I've seen anecdotal evidences for this, but I've not seen a
would you act on
study.
it?
I would act.
You would.
I would.
Okay, perfect. Lipshita.
Yes, sir, I would act.
You would act. How would you act? What would you add?
Pembrolizumab.
Single agent?
Single agent.
Pembrolizumab single agent. Sir, pembrolizumab with chemo or single agent? With chemo. Okay. Mohit. The real patient, he has gone around places.
All your patients are like this.
Yes, yes. Not all, some. The ones who keep me awake at night.
I was wondering if I give pembrolizumab, the data is only for MSI high. But there's a limited data for nivolumab plus.
All that is fine, Mohit. We all know. What will you do?
I might, if the performance status is good, I might.
46-year-old fit guy.
Nivo plus EP.
Nivo plus EP. So you will give
Because
Nivo
that is
EP.
yeah.
That's fine. You will give Nivo EP.
Pembro, we have a robust
So you
data
will
for
give Nivo
MSI
EP.
high.
You have
but
said your
for
answer.
Nivo EP.
You have given your answer. Nivo EP.
Chemo plus immuno.
Chemo plus immuno. Gunjesh?
Correct, sir. Chemo plus immuno.
Chemo plus immuno.
Yes.
Dr. Alurkar, sir?
I've never seen a TMB in a pancreatic cancer. Maybe
Very good.
Maybe you have, as you mentioned, you have 5 patients coming to every day with report. I don't have those rich patients. But yeah, I would not
Sir,
know.
you are in Ahmedabad, sir. Ahmedabad is full of rich people.
Ahmedabad is there, but they're all counting money. Anyway, I would not act on this.
You will not act on this. All right. Dr. Sarbajit is there. Since he had presented earlier, can we just ask him? Or yeah, somebody wants to make a point. So Dr. Sarbajit, sorry.
So I would take it with a grain of salt. We have to understand that blood tumor mutational burden and tissue tumor mutational burden are not synonymous.
Okay.
So even if I wanted to use immunotherapy, in the US at least, insurance companies will give me a hard time.
Your patient is a Dubai sheikh who doesn't bother about US insurance.
Okay.
Would you give it or not give it?
I would give chemotherapy alone unless I can get like a tissue that also shows tumor mutational burden is very high.
So can I just show you that VUS? So you see the VUS here. There are at least 30 genes which are having VUS of varied fractions. And that's exactly why the TMB has come high.
Right.
You know, that's why your TMB is high. It is correlating.
Right.
So this is a TMB high. I think that we should not have a doubt on it. Would you still not give any immuno?
I would go with standard chemo.
Standard chemo. Excellent. A show of hands. How many would give immuno to this patient? Any show of hands? Nobody? Immuno, how many would add to chemo or give it? You all said yes. You can lift your hands. You know, you should commit to. No? Okay, very interesting. Okay, so the idea is, again, I did a literature review on this because once this case came, there is a lot of cases, and this is a case of landscape of TMB. If you see the last but third one, there is around 1.4% of pancreatic adenocarcinomas which are TMB high. So it's 1.4%. It's not more than that. It's only 1.4%. And there is other group or other papers which have shown that there's a subset of TMB high with microsatellite stable. So that subset exists. Now, in another paper, they showed that this was around 40% of all MSI or TMB high. So 60% were MSI high, TMB high, 40% are MSI stable but TMB high. And in this, they were mainly in the mucinous and colloid, inconsistent responses to IO. This is a paper which was tested in 21,000. This is by Dr. Amit Mahipal. They looked at 21,000 samples and found 293 were TMB high. In that group who had TMB high, given immunotherapy, the overall survival was around 25 versus 5 months. So again, it is not a phase 3 randomized control trial, but there is some evidence to add it, and this is why we added immunotherapy to this patient. So we are giving chemo plus immuno. Whether it works or not, I don't know. I'll discuss it. Can I have 1 minute to discuss the last case?
Vamshi,
Okay. Yeah.
one comment. TMB in blood, you take the cutoff generally as 15, if I'm not wrong. In tissue, it is 10.
Point
so
made.
FDA approved TMB as a agnostic marker. If you have
Yes.
it irrespective, then you can probably try it.
FDA has not approved it, sir, right now because it's mostly MSI high approval, not TMB.
No, TMB as an agnostic marker, you can give pembrolizumab as per the registration trial.
Yeah. So the last case, 73-year-old gentleman came with abdominal pain. That is the cecal mass, which you can see, a big mass with a tumor thrombus going into the vein. It's a large mass of around 12 centimeter and a single liver met that you can see there. So surgeons this time, Dr. Arina sir, you will operate or, okay, Rajesh, Dr. Rajesh.
I think I will ask my medical oncology friend to give an NACT first.
NACT first, okay. This patient has MSI high with a KRAS mutant. So medical oncologist.
Immunotherapy should be good.
Immunotherapy. So surgeons are more fond of immunotherapy than us. Proven, hence proven. Medical oncologist, what will you give? Immunotherapy. We have only 1 minute. Which immunotherapy will you give? If not, what will you do? So Gunjesh, MSI high, clear pace.
So chemotherapy with pembrolizumab, sir, Kpox and pembrolizumab.
So chemo plus pembrolizumab.
Yeah.
Okay. Siddharth.
Yeah, last.
Finishing.
Yes, sir. Like immunotherapy, I am going to add, but
Okay, how many would use only immunotherapy? Only immuno? No one. There is Nivo-EP data which just came out in MSI high cancers.
EP is like
No, EP is dangerous.
Yes,
Okay,
yes.
All
That's
right.
why
Fine. So this patient we have given, okay, fine, I am running out of time, so I'll just show you what we did. We actually started the patient on Folfox, got the report, added chemotherapy, immunotherapy to it. Don't ask me why, we gave only nivolumab. Why not pembro? We gave nivolumab. And this is the response. So patient, as you can see, that huge mass has become this. The entire tumor thrombus has disappeared. Liver met is just now 1 centimeter, less than a centimeter, close to the diaphragm. So this patient has undergone a surgery. And after surgery, we have given SBRT to the liver met because it was too far up to do anything surgically. And plan is to complete his adjuvant nivolumab for 1 year. Do I have data for that? I don't have data, but that's what we are planning to do. So I thank all my panelists and I thank the audience. Thank you, everyone.
Thank you, Dr. Vamsi and all the panelists. Is Dr. Sarabjit here? Can we have group photograph plus any questions from the audience for the panelist or for the speaker? Dr. Sarabjit.
Thank you so much, Vamshi sir and all the panelists. Now, Vamshi sir really, he really broke it down for us that not just choosing wisely, but precisely also is the theme of immuno-oncology. So, proceeding with our symposium number 6, which is sponsored by BMS. call upon the chairpersons, Dr. Ramesh Nimagada, sir, medical oncologist from Chennai, Dr. Dilipka, sir, surgical oncologist from Bhubaneswar, and Dr. T.P. Sahu, sir, medical oncologist from Bhopal.
The next session topic is live tumor fragment platform to measure ex vivo response to immunotherapy in an Indian patient population. And this talk is going to be given by Dr. Inko German, Vice President, Precision Oncology, Clinical Affairs, Stanford University, USA, at Halifax.
Good afternoon, everybody. I'm Hinko Gierman. First of all, thank you so much to Dr. Carr, Dr. Biswas, and the organizers for inviting me. I'm going to talk to you for a little bit about why we can predict response to immunotherapy. But before I do that, very quickly, you can see up here my background. I think the most important thing is to call out that I have an obvious conflict of interest. I work for the company that I'm going to talk to you about. The first thing that I want to ask you is if anybody knows here how many checkpoint inhibitors have been approved by the FDA in the US. I know we're in India, but some of you are very familiar with the NCCN guidelines and FDA. Does anybody have any idea? There's too many clinicians in the room not to know the answer. Well, does anybody think it's more than 10? It's actually 11, at least last time I checked. So the problem, I think, is not so much that we don't have checkpoint inhibitors or that we're not approving enough of them. The problem, I think, is this, that we do not have good biomarkers today to decide when to use checkpoint inhibitors or to know which checkpoint inhibitors to use. You saw several talks today talk about combination therapies like Ipi/Nivo. Today, a patient that has melanoma has a choice of several immunotherapy options, including Nivo, Ipi/Nivo, and Opdualag. There is no guideline, no biomarker actually to tell you which one of those to use. So doctors look at the frailty of a patient and other characteristics, but they have no way of knowing, not even with PD-L1, what would be the best. And that's a problem we're trying to solve. What I'm showing you here today is a meta-analysis that was recently done on about 91 different trials using those biomarkers, using PD-L1, MMR, and MSI. And the objective response rate in those trials using those biomarkers was 20%. That's across all lines of therapy. What's the best use case? What's the best case scenario? Well, jump to frontline only, then your objective response rate goes up to 30%. Include stable disease. Now you're up to 40%, and some tumors even 50%. Half of your patients is deriving absolutely no clinical benefit from checkpoint inhibitors if you put them on them using biomarkers. That's the problem that we want to solve. So I'm going to show you today a little bit of the data of how we're trying to solve that and how we want to start a study here in Bhubaneswar, India, in collaboration with Dr. Banerjee's lab in DNA to start testing and training that platform here in India for Indian patients. So how do you solve that problem? You probably all have seen many, many papers in the last decade showing predictive factors for immune response. Most successful have been those that use RNA-based or other omics approaches. They're sometimes slightly better than PD-L1 and TMB, but none of them have been really, really accurate. This, what I'm showing you here, has been the only approach that has been successful in accurately predicting response. And it does so because it doesn't actually predict. It measures it. So how does it work? This is from a Nature Medicine paper from 2021 from the Netherlands Cancer Institute, but others have done similar work. And basically what they've done is taken tumors, resected tumors in this case, incubated them in the lab with a checkpoint inhibitor, nivolumab in this case, and then measured cytokines. So what you can see on the heat map is that every column is a patient and every row is a cytokine. There's the usual suspects like interferon gamma that typically we know are associated with T-cell activation, but many, many others. And you'll see that about a third of the patients have a response and most of the patients don't. And they use this in a training algorithm, which is the original cohort in the middle, to establish an algorithm of 11 cytokines. And then using that algorithm, they tested it in a validation cohort here on my right. And the red dots are the patients with a clinical response, which were accurately predicted by the algorithm. Since then, they validated this in another 180 patients, which were presented at AACR 2 years ago. So this is pretty accurate. Its accuracy is over 80%. What's the problem with this? Why is nobody else doing this? Well, the problem with this approach is you need a lot of tumor. And there's only two ways to do that. Either you grow organoids, which means you need 2, 3, 4 weeks time, in which time your patient is already starting treatment, or you need a resected tumor, something that is feasible for many early stage patients, not common in the metastatic setting, which is where you would usually use immunotherapy. So how have we solved that? I'll show you a little bit of data today on how our platform works, where we are able to take a very small amount of tissue like a core needle biopsy, which is much more common in the metastatic setting. This also works for forceps and even FNAs or punches. And in short, we take a biopsy, we keep it alive, so we don't put it in formalin, but we keep it alive and then cut it into small live tumor fragments. We encapsulate them in a hydrogel and then incubate them in the lab, and in 48 hours get a result from whether or not the T cells are being activated by a checkpoint inhibitor. So how do we do that, and how do we preserve the tumor microenvironment, including the infiltrating immune cells? So what I'm showing you here on the left is a 20-gauge core needle biopsy from a lung tumor. We embed it in agarose, and then it goes into a cutting device that we build ourselves, and it automatically cuts a core needle like that in about 10 minutes or so. If you manually cut a resection into fragments this size, they're 300 microns thick, it can take you several hours to do that. So we automate all of that, and then we end up with about 10 to 20 of these live tumor fragment slices per biopsy. Each of these fragments, once we have them, conserve and reflect the histology of the original tumor. So we see in there the tumor, the stroma, and very importantly, the immune cell infiltration. And to date, we have reviewed over 500 specimens that we generated that way from human tumors by pathologists. So here's an example of an analysis that reflects that we are not just keeping the histology in the fragment, but also keeping it intact during the incubation period. What I'm showing you here is the ratio of CD4 positive to CD8 positive T cells, and we're comparing day 0 in gray to day 2 in purple. And you can see these are 9 different samples from, or I should say 9 different patients, each with samples. You can see that in most of these cases, the ratios are pretty similar. Now, you might have noticed the last one doesn't look very good. And you might have noticed that there are big error bars on that specimen. The reason is that that, which commonly happens in tumor samples, is actually a sample that had quite a lot of necrotic tissue. So the fragments that we saw from this patient actually were mostly dead, and there was more noise in that sample, much more noise than any of the other samples. And as any pathologist can tell you, very common for 10, maybe even 15% of samples to be necrotic or even devoid of tumor content. So we do something else. We actually have a non-invasive way of measuring viability and also tumor content, but I'm just going to show you today how we measure viability. Now, for decades, many assays exist that do this. Propidium iodide, CCK8, many of them that you will all know. All of those terminate the tissue. The tissue that I just told you is so precious because we only have a little bit of core needle biopsy tissue, the only way we can functionally predict response to immunotherapy. So how do we do it? We use an imaging technology called optical coherence microscopy. It's similar to what your ophthalmologist does to your eye to measure that your eye is in good condition. And it works kind of like an ultrasound, but now with light waves, and it allows us to see if the tissue is alive. It measures the movement of the mitochondria. What you can see on your right is that we have several of these living tumor fragments cut from core needle biopsies that I just showed you. And the top ones, 1 to 6, we killed them with heat. The bottom ones we left alive, and you can see they're gray. It's because they're alive, their mitochondria are moving. We're measuring that with the OCM. On the left is just a validation with the multiphoton microscope, where you can see in purple the dead cells with propidium iodide, blue is NADH, indicative of the living cells in the bottom. And here you can see a three-dimensional fly-through. I told you that we keep the tumor microenvironment intact three-dimensionally, and this is what that looks like in 3 dimensions when we measure the different cells and their states. The green dots are the living cells, and you can see the difference also when we heat treat them and they die and the green goes away. So I've shown you a little bit of data of how we cut. I've shown you how we keep the TME intact. And here I'm showing you how we measure viability. So now let's get to some data of whether or not this really works. So the first thing we did is we tested this on resections because they're easy to get. And we asked the question, are IO biomarker positive samples enriched for a platform response of our platform? Because if the platform works, that's what you would expect. So we took 60 patient samples, colorectal, bladder, head, neck, and some lung and breast. And you can see in gray the ones that were biomarker negative. So that means your PMMR or your MSS or your PD-L1 score is 0 or negative. And in purple if you were positive. This is a heat map of those 60 samples, and you can see the same thing that I showed you about the Nature Medicine paper. You see about a quarter to a third where you see a cytokine response, and the rest of the samples, not really much happens. No upregulation or downregulation. In the bottom, in the pluses, you can see the ones that were biomarker positive. And when you do the math on this, which is what I'm showing you on the right, you can see that of the biomarker positives, 39% had a cytokine response from the platform, in this case defined by 8 cytokines that are known to be involved in T-cell activation or associated with response in our data. When you look at the biomarker negatives, only 11%. So there's a four-fold enrichment almost in the biomarker positives. And this is, by the way, more or less what you would expect if you're accurately predicting clinical response. Because as you know, biomarker positive patients don't respond to immunotherapy more than half of the time. That was the first slide I showed you. That's the whole problem we're starting to solve to begin with. So I showed you a little bit of data of how we conserve it and that it works for biomarkers. How can we now make this work for core needle biopsies? And how can we mitigate tumor heterogeneity, some of which you saw seeping through in the data? We do that this way. We call it sequential treatment strategy, and what we do is we no longer do a control versus a treatment, the traditional way that you would do an experiment. Instead, we collapse it all into one. So we take all the fragments from the tumor tissue, we put them all in one well, we do the control treatment the first day, we use IgG, and then the actual treatment with a checkpoint inhibitor like anti-PD-1 the next day in the same well. So how do you compare the two? Well, you can't compare fold changes directly. Instead, we measure the slope. So in the bottom, what you can see is interferon gamma. On the left is a likely non-responsive patient, and we measure that by dividing the slope in the purple part, where we treated with anti-PD-1, over the white part, the control. In the left, the fold change is almost the same. It's a fold change of 1. But on the right, you see what looks like a potential responder where interferon gamma all of a sudden goes up. So here's what that looks like for 4 patients on the left that are likely non-responders. They're basically lines that don't have a change in slope. And on the right, you can see 4 likely responders where the slope dramatically changes, about 5-fold change difference. I'm pointing out the one in the bottom there that has the 3 lines. That's an example of heterogeneity. You can see the top line is more steeply going up in the purple part than the bottom 2. Maybe there's more T cells, maybe they get more activated, we don't know. What we can tell is that in the gray part, they kind of behave the same way. So the upper line, the gray line, it's hard for you to tell, but it's also a little bit higher. So it controls for itself. Whatever was in there, more T cells, it was there in the control part too, not in a different well. So when you divide the purple over the gray, you get basically the same numbers. They're all around 7 or 8 fold changes. So this is how you control for heterogeneity, and you need twice less a sample. So I'll end by showing you some of the data that we've started collecting to date. We have several ongoing clinical trials, one with Mayo Clinic. Dr. Dev Mukhopadhyay is here from Mayo Clinic today. He's been key in organizing that. We have a lung trial that has several sites in the US, and finally a basket trial that we're doing with the Hoosier Cancer Research Network, a non-for-profit academic organization, and it has UNC Chapel Hill, University of Wisconsin, and many other sites involved in it. And finally, a biobank study. All of these studies are observational, and we're collecting patients in the metastatic and recurrent setting, and in 2 of the studies also neoadjuvant. We heard a little bit today about the increasing use of immunotherapy in the neoadjuvant setting. And we're also starting to look at adjuvant. We're enrolling over 1,000 patients, and I'm showing you here data on the first 130 so of these patients. These are all core needle biopsies. So this is an extremely small amount of tissue. And what I'm showing you here is that we're seeing similar results. We're able to cut core needle biopsies over 80% of the time, and we get good signal more than 80% of the time as well. We're seeing a group of patients where we see a cytokine response. I'm showing you basically every cytokine that we're getting a signal for, so there's a lot of noise on this graph. And you see a lot of patients to the left where there's basically no signal going on. There's living tissue, we measure that, there's tumor content, there's just not much happening because of the lines staying the same. And we're literally getting the first clinical response data in as of now. We're starting to get this. So we got 2 responders. Unfortunately, both were combination treatment patients. So they're confounded by the chemo component. You don't know which one of them is causing the response. One on the right looks like a immunotherapy responder according to our platform. Unfortunately, that patient actually got pulled off of Pembro due to toxicity after a few cycles. Let's hope that he will or she will continue to respond. On the left is a patient that doesn't look like a platform responder according to us, but they have a response. They have a triplet, so they have 2 chemos and possibly they're responding to that. What we're really going to be doing is collecting this data, and in the bottom I'm showing you in gray the 17 additional patients that we already know are on immunotherapy. About half of them are on mono. Any mono patient should correlate one-on-one with the platform, right? If you're a responder on mono, you should be in the red zone. If you're a progressor, you should be on the left in the white zone. The combo patients, you can still use them. If you progress on combo, you're progressing on everything, including your IO component. So you can use that equally so. And finally, I'm showing you here also the IO biomarker status for the core needle biopsies that we have IO biomarker status for. On the top, in yellow, the ones that are positive. In the bottom, the ones that are negative. And we're seeing the same trend, where the positives are enriched for the platform responders, and the bottom ones, the blue ones, the negative ones, seem to be mostly the ones that are not the responders. So I'm going to end the last 2 minutes with talking about why I'm here in Bhubaneswar. We've started a collaboration with Dr. Banerjee and his lab in DNA. And the goal is to launch this year a study that starts collecting Indian patient samples. The goal of that is to generate evidence outside of the US. We initially started in the US. We're now going to go to India. We're also starting to look at other countries. We want to generate evidence outside of the US on other populations, as well as starting to generate more data. It's literally the largest country in the world in terms of population. And one of the problems, as in many countries, including the US, is access to immunotherapy. Today in the US, lots of people, even with insurance, have trouble paying for the medication. Cancer is the #1 cause of bankruptcy. Making sure that when you get treatment, that it's the right treatment for you is really important, both in the US and in India and everywhere else. And by making sure that you have an accurate way of knowing who should get the treatment, you're making it overall more affordable and you're reducing toxicity for the people who are not benefiting from it. And finally, you can also now start looking for those rare responders in tumors that today are not eligible because of the biomarker. Think of colon cancer. 95% of patients are pMMR, are ineligible for immunotherapy and get chemo like FOLFOX or FOLFIRI. Among them, 5% or so are responders. We hope to be able to find them. So the goal of coming here is to also help kickstart that in India and generate lots and lots of data. We're going to need thousands of patients of data, and we think this is one of the best places to start doing that. I want to acknowledge, first and foremost, all the patients, the doctors, and research staff from all of our sites that help generate the data and that are continuing to do so. We're working on our first paper that in a few months, hopefully we hope we'll have enough data to share those first results with the world. The employees, the organizing committee of this conference, Dr. Banerjee, Dr. Mukhopadhyay, and all of our sites that really have been very instrumental in moving this forward. And I'll close out by just pointing out my last bullet in this summary, the data. Our goal is to make as much data as we can open access and freely and publicly available. So for example, all the cytokine data that I showed, when we publish it, we're going to put it in GEO and other public websites so that all the researchers can have access to it. Thank you so much, and happy to take any questions now or later.
So am I permitted a question here? So I'm a medical oncologist from Jaipur in India, and very happy to know that you're setting up trials in India also. The technique and the procedure which you describe, it appears to be quite complex, requiring a lot of laboratory facilities and high-end equipment. So if we are thinking of, getting it to common usage, then it will have to be a lot more simplified. So is that a plan? Can it be done in a smaller scale, say in a smaller city?
Yeah, that's a great question, right? The question was, this looks very complex. So yes, I agree with you. This is more complex than just placing a DNA sequencer in a lab. So a lot of the work has gone into simplifying and automating that. And we are going to continue to do that. Right now, we already have a prototype of the platform placed at Mayo Clinic in Jacksonville with some of its components. They've been great in helping to do exactly that. We do think, however, that with 2 employees in a typical path lab, this is doable. But you're right, it's going to require effort, but we think it's worth it.
Thank you.
Thank you, Dr. Hinko, sir.
Thank you so much.
Thank you.
I would request Dr. Hinko and the chairpersons to please come and get a picture clicked. Thank you so much. Now, for the next event, which is the Digumati Foundation Oration, I would like to invite our chairperson, Dr. Raghunath Rao Digumati, sir, medical oncologist from Vizag, Dr. Shubda Chiplunkar, ma'am, scientist from Mumbai, and Dr. Hemant Malhotra, sir, medical oncologist from Jaipur.
Okay, thank you very much. Pleasure and honor to chair this oration, the first oration of this conference. I'd like to request my co-chair, Dr. Raghu, to tell us a little bit about the Digumarti Foundation, after which this oration is named.
Thank you very much. I started this foundation in about 2003, initially with the money my wife and I got as examiners. So we used to support a couple of girls in their education up to their graduation. But when I moved to establish the Homi Baba Cancer Hospital at Visakhapatnam, the need became intensive for a patient-friendly campus because we only had a piece of cobra-ridden land, 77 acres of it and nothing else. So I used the foundation to raise a large amount of funds for patient-related activities, and that's how it was born. I then moved to fund this oration for the Immuno-Oncology Society on behalf of the foundation. It's still largely philanthropic, and rest of it is from friends and relatives. Thank you very much.
So for those of you who don't know Raghu, we call him Raghu. His real name is over there. He is one of the senior most and one of the most respected oncologists of this country, and we are proud of you and the work which you do, Raghu. So I'd like to request the past president of the Immuno-Oncology Society of India, Dr. Shubda Chiplunkar, to introduce the orator to you.
Thank you. It's my privilege to introduce Dr. Padmini Sharma, who is an internationally renowned physician scientist at MD Anderson Cancer Center, where she serves as professor in genitourinary medical oncology and immunology. She is the associate vice president of immunobiology and holds the TC and Genetsu endowed chair in cell biology. She is the inaugural scientific director of the immunotherapy platform at MD Anderson, overseeing immune monitoring studies for over 100 clinical trials. In 2022, she was appointed the director of the scientific programs for the James Allison Institute. Her pioneering work includes leading the first neoadjuvant trial with immune checkpoint therapy. She also identified multiple mechanisms of response and resistance to immune checkpoint therapy. Her work also led to the development of multiple innovative immunotherapy clinical trials, including trials that led to the FDA approval of immune checkpoint agents to treat bladder and renal cell carcinoma. She has received numerous prestigious awards, including the Emil Frey Award 2016 and the Coley Award 2018 and the Randall Prize 2021. Dr. Sharma is a member of the American Society for Clinical Investigation, member of the American Association of Physicians, and fellow of the Academy of the American Association for Cancer Research.
So Dr. Sharma's topic today is From the Clinic to the Lab, Investigating Mechanisms of Response and Resistance to Immune Checkpoint Therapies. So all of us are aware that these are very expensive molecules, and they are expensive molecules not only in India but also in the United States. And if you look at cancers across the board, only 1 out of 5 patients is going to respond, other than if you leave apart Hodgkin's lymphoma and maybe melanoma or maybe renal cell cancers. So it's desperately important for us to identify which patient is going to respond and which patient is going to be resistant to these treatments. So that is what we are going to hear from the top researcher in the field. So over to you, Dr. Padmini.
Okay, thank you. Thank you so much for the kind invitation and the kind introduction. Okay, sorry. Okay, better? Okay. So thank you so much for the kind introduction. I'm very happy to speak today on the mechanisms of response and resistance to immune checkpoint therapy. And as you heard, it's very important to identify these mechanisms of response and resistance so we can select the right patients for treatment, as well as determine what will be the next rational combination strategy. These are my disclosures. So just to remind everyone that the immune checkpoint therapy field really started with Jim Allison's work, which was a paradigm shift in terms of looking at how to block inhibitory mechanisms on T cells. So Jim identified the CTLA-4 receptor and showed that blockade of the CTLA-4 receptor will lead to unleashing the T cell response so that you can have anti-tumor responses and tumor rejection. This also allowed us to open an entire new field called immune checkpoint therapy based on this concept of blocking inhibitory pathways. The CTLA-4 pathway was the first, but of course PD-1 and PD-L1 are the next pathways that were then targeted with antibodies. I just want to highlight that the cartoon here is a little bit outdated, but there are still other pathways that you can see that are now being tested in clinical trials. In the US, of course, immune checkpoint therapy has taken off extensively since the first approval in the US was in 2011. Of course, then this became worldwide approval in many countries, including India for some of these drugs, including ipilimumab and nivolumab, for example. But you can see in the last decade or so that we've had multiple approvals across different tumor types, really highlighting the concept that immune checkpoint therapy is not really targeting the cancer. Immune checkpoint therapy is now approved across these different tumor types. Oops. Quit. Okay. All right, sorry. Immune checkpoint therapy is now approved for these different tumor types because these agents don't target the cancer cells, they target the immune system. And then the immune system is capable of targeting the different tumor types. And so the questions that remain, despite the many successes that we've had, are why do some patients respond and others do not? What are the biomarkers that predict response or immune-related toxicities? And we don't have time to talk about that in this talk today, but I'm happy to answer questions later. Are there biomarkers to enable patient selection for treatment with monotherapy, such as anti-CTLA-4, anti-PD-1 alone, or combination therapy, which is the anti-CTLA-4 plus anti-PD-1, which has higher frequency of, or higher response rates, but also higher frequency of adverse events? And then can we increase the number of patients who respond? Are there other pathways that can be targeted to improve clinical outcomes? All of these research questions, of course, are what we're working on every day. So I'm going to give you just snapshots and ideas of how we're addressing these particular research questions. First, I want to point out that a lot of times we utilize this method called reverse translation, and you'll hear more about that in the next talk by Dr. Sangeeta Goswami. But this really takes into the concept that what we're looking at in the laboratory when we're testing in the laboratory is that usually we're using mice, and the mice are inbred and the disease is homogeneous, and that allows us to do great hypothesis testing. But in the clinic, where many of us are treating patients, the patients are polymorphic and the disease is heterogeneous. And so the patient data is only going to allow us to generate certain hypotheses and correlate the data. So this is correlative data when we get it in the clinic. It is not mechanistic data. It is only correlative. And therefore, we have to use the data from the clinic and use that data to then generate a hypothesis that we can mechanistically test in the laboratory. And so this is the concept of the reverse translation, going from the clinic back into the lab. And so because in the US, for example, we have about 2,500 clinical trials ongoing with immune checkpoint therapy, it's very important that we study the patients to understand the potential mechanisms. To do that, we've really relied on these neoadjuvant clinical trials that you see here. basically we're building off the concept of standard clinical trials of phase 1, phase 2, and phase 3 by introducing these pre-surgical or tissue-based trials that allow us then to have access to the tumor tissues so that we have sufficient tumor tissues at the time of surgery for testing potential mechanisms of response and resistance. It also gives us a chance to have a clinical signal in an earlier disease setting so that we can determine whether or not the neoadjuvant setting would be the best place to use the immune checkpoint therapy agent. I want to highlight that the first neoadjuvant trial that we did with immune checkpoint therapy, we conducted in 2006. We initiated the entire process in 2004, but the first trial started enrolling patients in 2006. This is important to note because you'll notice that this date of 2006 was 5 years before we even had any FDA approvals or any approvals of an immune checkpoint therapy agent. And so it's important for you, as you're using these new agents now, to think about how to get them earlier into disease setting, not only to see if it can give clinical benefit, but also so that you can have access to the samples for testing what's happening within the tumor microenvironment. So this is the first neoadjuvant trial that was done in 2006. You can see it was in patients with localized bladder cancer. I also want to highlight that it was a small trial with only 12 patients. You don't need large number of patients in order to learn about the biology. Small numbers of patients studied in depth will give you a lot of biology that you can understand. So these were patients who were already scheduled for surgery because they had localized bladder cancer, and we gave them 2 doses of anti-CTLA-4 prior to their surgery. The clinical trial allowed us to have a clinical signal for safety because it was the first time that we could show that immune checkpoint therapy can be combined with surgery in the neoadjuvant setting. So the safety data allowed us to then generate many, many more neoadjuvant clinical trials, as you've heard about. It also gave us the clinical signal for efficacy because 3 patients developed pathologic complete responses, meaning all of their tumors went away in the bladder that was looked at by the pathologist. And so this allowed us to then design clinical trials for patients with metastatic bladder cancer that then led to FDA approval of immune checkpoint therapy for patients with metastatic bladder cancer to receive anti-PD-1 therapies. But more importantly, it gave us access at the time of surgery to the tumor microenvironment as shown here, whereby we can then study what was happening between pre- and post-treatment samples to understand how anti-CTLA-4 impacted the tumor microenvironment. And you can see from this simple IHC study that you can see there's an increase in CD3 T cells, CD4 T cells, CD8 T cells, and these were granzyme positive, meaning they're activated. And this was also the first time we were beginning to see CD20 B cells. So anti-CTLA-4 was changing the tumor microenvironment with T cell and B cell infiltration. One of the things that we found when we looked at the differentially expressed genes between the pre- and post-treatment samples was that the top 10 genes were related to the immune response, which is to be expected since this is an immunotherapy agent. But the top gene here was I-COS, which had not been previously studied in the anti-tumor setting, and so we were curious about this I-COS pathway. I want to point out that we also did another clinical trial in the neoadjuvant setting with a combination of anti-CTLA-4 and anti-PD-L1 in patients with bladder cancer. And again, this was a small clinical trial of 28 patients. And again, we had pathologic complete responses of 37.5%. And even in patients who had large 3D masses where the surgeons were not sure that they could get good resection of these tumors, we had pathologic complete responses of 42% as a result of the combination of immune checkpoint therapy. But again, importantly, we had access to the tumor tissues as shown here, and so the tumor microenvironment was able to give us a lot of details about what was happening and what could predict responses to immune checkpoint therapy. And what we noted was that patients who had increased density of the tertiary lymphoid structures as shown here, those were the patients who had better responses. And these tertiary lymphoid structures were comprised of cells, T cells, B cells, dendritic cells, and importantly, the T cells were these I-COS positive CD4 T cells as shown in the responders. So what is I-COS? Now, we've identified it in multiple clinical trials. And I-COS is inducible co-stimulator. It belongs to the same family of CD28 and CTLA-4, and its expression is increased on activated T cells. It has a very diverse role that's been reported, and for most immunologists in the room, you'll recognize this as a marker of follicular helper T cells. But a role in anti-tumor response had not been established, and so we wanted to examine what I-COS was doing in the setting of anti-CTLA-4. So this was the clinical data from our patients. And patient data allows us to then have information to generate a hypothesis. And so the first hypothesis we generated was that the ICOS-ICOS-ligand pathway is necessary for effective anti-tumor immune responses. To test that hypothesis, we went back to the lab and we used wild-type mice, ICOS knockout mice, and ICOS ligand knockout mice. And we asked whether or not the mice that lacked ICOS or ICOS ligand could respond in the same way to anti-CTLA-4 as compared to wild-type mice. And you can see that clearly in the top line, the wild-type mice, 80 to 90% of those mice could reject the tumors and have long-term survival. But the ICOS knockout or the ICOS ligand knockout mice had impaired anti-tumor responses, as you can see. So the second hypothesis was, could we target the ICOS-ICOS ligand pathway to improve anti-CTLA-4 or any other immunotherapy? And so we did that in a preclinical model, again, a mouse study, where we combined ICOS targeting with an anti-CTLA-4 antibody in wild-type mice, which is the top graph. And you can see the top blue dotted line. That's when you gave combination therapy. The majority of the mice could now reject the tumors, compared to any of the lines below, which were monotherapies or the controls without treatment. And so the combination therapy clearly had an impact on improving responses. The same experiments done in wild-type mice were done in the ICOS knockout mice, which is the bottom graph. And here you can see that the ICOS knockout mice cannot respond, clearly indicating the importance of the ICOS-ICOS ligand pathway. And so we knew that that pathway was important, and now there are multiple companies that are developing agents to target the ICOS pathway. But important questions we wanted to ask then were, what about other tumor types? Are these tumor types that are resistant to immune checkpoint therapy, is it because they don't have any T cells within the tumor microenvironment or any T cells that are capable of responding to immune checkpoint therapy? And here you can see in this figure, the tumors that do respond well have high mutation loads or high tumor mutational burden. And because they have lots of mutations, such as bladder cancer, lung cancer, melanoma, with those high mutations, it's thought that they have lots of antigens, and therefore those antigens could then engage a T cell response, and immune checkpoint therapy can enhance that response. However, prostate cancer, glioblastoma, pancreatic cancer, those are tumor types that do not respond well to immune checkpoint therapy. And it is thought that they do not respond well because they don't have the antigens that are present for immune checkpoint therapy to then help the T cell response. But as an immunologist, we recognize that T cells only need a single antigen. They don't need thousands of antigens for a T cell response to be generated. And clearly, prostate cancer, pancreatic cancer, glioblastoma must have an antigen because they're cancers, which means they have mutated proteins. So we wanted to ask this question in the setting of prostate cancer. And so we wanted to ask, can anti-CTLA-4 or ipilimumab elicit a T cell response to conventional prostate cancer mutations and antigens, or new neoantigens, which we could identify? And to do that, we enrolled 30 patients with metastatic castration-resistant prostate cancer onto a clinical trial, whereby we could take the tumor sample from these patients, perform exome and RNA sequencing, identify the tumor mutations in these tumors, and then the patients are receiving ipilimumab or anti-CTLA-4 therapy, collect the blood from those patients, and ask whether or not the T cells in those patients can now recognize these different neoantigens or shared antigens such as PSMA and PAP from the prostate cancer patients. And here you can see these are two examples. This is patient number 7 with 2 examples of antigens that were mutated. One is ROG1 and nucleotide exchange factor 37, and one is dihydropyrimidase. You can see that the patients were only able to respond to these antigens after they received anti-CTLA-4. And so it was clear that even in prostate cancer patients who have few mutations, you can see now that there is a T cell response that's generated to these specific neoantigens after anti-CTLA-4 therapy. So why are we not seeing the same clinical responses in patients after we give anti-CTLA-4? And so we thought that maybe the T cells were no longer getting into the tumor microenvironment. Even though that the mutations are present and the T cells can respond, they're not infiltrating into the prostate tumors. And so in another clinical trial with 20 patients who had localized prostate cancer, we were able to give 2 doses of anti-CTLA-4 and have access to the tumor tissues at the time of surgery and ask whether or not their T cells were infiltrating into the tumor microenvironment. And you can see that prostate cancer prior to treatment, in these pre-treatment samples shown here, the prostate cancer has very few infiltrating T cells. However, after treatment with anti-CTLA-4, there's now increase in the CD4, CD8, CD45RO, ICOS positive T cells, clearly indicating that we've converted the cold prostate tumor, which has few T cells, into a hot prostate tumor that has lots of infiltrating T cells. And so the T cells are capable of getting into the prostate tumor microenvironment. So why aren't we getting rejection of these prostate tumors the same way we got rejection of the bladder tumors? And so what we found was that within the tumor microenvironment, there was also a change in inhibitory pathways. And this is really very important to keep in mind because the immune system is very dynamic. As it evolves over time, it will constantly try to control itself. So if you think about it, T cells, when they're turned on, CD3 and CD28 turns the T cell on, CTLA-4 is also upregulated, and that's to control the response. And here, when we're driving the T cells to now be activated in the prostate tumor, there are also mechanisms that are trying to control that. And so what we found with these mechanisms were an increase in the PD-L1 pathway and the VISTA pathway, which are both inhibitory pathways. And just to highlight that point, again, this is an immunohistochemical study of prostate tumors. Pre-treatment, there is no PD-1, there's no PD-L1, and there's no VISTA. In hundreds of prostate tumors that we've looked at, prior to treatment with an immunotherapy agent, those are all negative. It's only after the patients have received treatment with an immunotherapy agent, now you can identify these specific pathways. And this is so important to understand that a single biopsy will never be able to give you a full understanding of how the immune response will behave and how it will evolve within a tumor. You cannot make a decision on a single biopsy. You must see the evolving response over time, and we'll need multiple longitudinal biopsies. And so the post-treatment sample now from these patients, you can see, now has PD-1, PD-L1, and VISTA, indicating that these inhibitory pathways will need to be targeted. So where were VISTA and PD-L1 expressed in the tumors? What we found was that they were expressed on macrophages, these CD68 positive macrophages. And so they were either VISTA positive or PD-L1 positive. Very few of the macrophages had both VISTA and PD-L1. And you can see the VISTA is in white and the PD-L1 is in pink, and CD68 is yellow for the macrophages. So we took the T cells from the patients to ask whether or not these T cells can respond without the VISTA and PD-L1, and then when VISTA and PD-L1 are in the in vitro settings, what happens? And you can see the first bar is showing interferon gamma and TNF-alpha production, and that's when there's no VISTA present, there's just the patient's T cells with anti-CD3. And in vitro, these T cells are quite capable of producing interferon gamma and TNF-alpha. However, if you coat the plates with VISTA or with PD-L1 Ig, you can now see that the interferon gamma and TNF-alpha decreases as a result of these inhibitory receptors. So we worked with Bristol Myers Squibb and we proposed to them that there was now a second inhibitory pathway, which is the PD-L1, PD-1 pathway, and could we do a clinical trial in patients with prostate cancer that combined anti-CTLA-4 with anti-PD-1 to understand whether or not we would get better responses. And so we published this clinical trial already. I will not go into all of the details. I do want to say, though, that in this clinical trial, for the first time now, we were seeing these types of responses, where you can see this patient had a PSA up to 500, and the patient also had metastatic disease on CT scan. And then when we gave the anti-CTLA-4 and anti-PD-1, the PSA then came back down and became undetectable, and the disease on CT scan went away. What was important in this trial was that, one, the combination led to a higher frequency of toxicity. So we could not continue patients on the study. Importantly, the other thing that we noticed was that all of these responses were happening in soft tissue lesions. So if the patient had soft tissue metastases, they responded. But if the patient had bone metastases, those did not respond. And so metastatic castration-resistant prostate cancer predominantly goes to the bones, and therefore the majority of our patients were not responding. But from a biologic standpoint, why are bone metastases not responding to immune checkpoint therapy? What is different in the bone microenvironment compared to a soft tissue microenvironment? And so again, we had the tissues from the patients, either the soft tissue component or the bone lesions from patients that we taken biopsies. And we had the pre and post-treatment samples. And we asked what was happening in the tumor microenvironment in the soft tissue and what was happening in the tumor microenvironment in the bones. And in the soft tissue, what we found was that the T cells became Th1. They were effector T cells that could produce interferon gamma. But in the bone microenvironment, we never saw this Th1 response or the interferon gamma signature. What we found actually was a Th17 signature in the bone. So we wanted to understand the mechanisms that could drive this Th17 response. And so we went back to the lab and we designed a mouse model that was an immunocompetent mouse model that we could then inject the tumors into the bones of mice to mimic the bone metastases or into the subcutaneous areas of mice to mimic the soft tissue metastases. And then we treated the mice with anti-CTLA-4 and anti-PD-1. And you can see now in the red boxes are the anti-CTLA-4, anti-PD-1 treated, and the blue boxes are controls. And in the bone lesions, now you can see the Th1 responses never occurred in the bone lesions. But the Th17 did increase, which mimicked the human setting. But the subcutaneous lesions, which mimic the soft tissue metastases, you could see had an increase in the Th1, but no increase in the Th17, actually a decrease because the T cells are now skewing towards the Th1. So what drives the Th17 in the bone microenvironment? And so for the immunologists in the room, you may know that there are two cytokines, IL-6 and TGF-beta. And when these two cytokines signal at the same time, they will then lead to Th17 development and skew the T cell response away from Th1. And what we found was that IL-6 is very high in the bone microenvironment. And it's very high whether there's a tumor or not. So BM is without the tumor and BMT is with the tumor. So IL-6 is always high in the bones. But TGF-beta is only high when there's a tumor in the bone microenvironment. And this is because we showed in the paper that we published, it's due to the osteoclast in the bone microenvironment. So that is the only time that when IL-6 and TGF-beta will signal together, and therefore T cells will now skew to Th17. So we propose blockade of TGF-beta in combination with anti-CTLA-4 to treat the bone microenvironment. And so the green box is what I want you to pay attention to. The green box is a combination of anti-TGF-beta plus the immune checkpoint therapy, and this is showing the green box is much smaller in tumor volume as a result of the combination treatment. And what happens when we give the combination treatment in the bones is that now, for the first time, we get a Th1 CD4 expansion and CD8 T cell clonal expansion. And then the purple line showing the improvement in survival of the mice as a result of targeting both CTLA-4 and TGF-beta. So I hope you can understand that from the clinical data, we then look to see which patients are responding or not. We generate a hypothesis to understand what could be the mechanism that's allowing for resistance. We test the potential mechanism of a new combination, and now this combination is moving into a new clinical trial for our patients with bone metastases. So I just want to finish up and say that what I've shown you is just a snippet of how we think of moving between the clinic and the lab to make decisions about potential mechanisms of response and resistance that need to be targeted to improve outcomes. There are many more pathways that we need to consider as shown here. And so the work still needs to be ongoing, and there's a lot to do. At MD Anderson, this is really a team effort. It's led by the James P. Allison Institute, which houses the immunotherapy platform. And it's made up of colleagues in surgery, medical oncology, pathology, our translational research team, laboratory scientists, and computational biologists. We have 111 trials that are doing these mechanistic back and forth between the lab and the clinic across 18 departments, and have enrolled over 4,000 patients with longitudinal samples collected from 7,000 blood samples plus more, and over 4,000 fresh solid tumor tissue samples. So in conclusion, immune checkpoint therapy has joined the ranks of radiation, surgery, and chemotherapy. But combinations are the future. Sorry, multiple immune checkpoints exist. They're very dynamic in their expression. Therefore, they have to be evaluated in both thank you, and on treatment human tumor samples. The organ-specific microenvironment needs to be considered. We can't enroll all patients on a clinical trial and think that the patients with bone metastases are going to respond in the same way as a patient with lung metastases. They don't. They have different microenvironments. And then I hope you understand now that these pre-surgical or neoadjuvant and tissue-based trials provide a feasible platform for obtaining approval of therapy in earlier disease setting and to study biologic effects in patients, which provide insights into mechanisms that can be targeted for rational combination strategies. I have many, many people to thank. Their names were on the slide as I went through. I do want to really thank the patients who participate in our clinical trial. They don't always have to, but they still participate. And thank you, and I'll take questions.
So thank you, Dr. Sharma. That was a wonderful, wonderful talk. So I think normally during a narration we don't do questions, but we don't get this opportunity very often. So maybe a couple of questions from the audience.
Sure. Sorry, my throat is acting up.
Yeah, a very nice talk. And I would like to ask, in primary resistance which develops or during the course of the treatment, what happens to these pathways? Do they work cumulatively? Do they work independently?
So during the time on treatment?
Yes.
Yeah, so some patients are going to have resistance mechanisms, but they're not all going to have the same resistance mechanisms, right? So lung cancer patients may have a very different resistance mechanism to prostate cancer patients, for example. So those are the kinds of things that we're finding. And if the metastatic disease is in brain versus liver versus lung versus the bones, they may also have very different resistance mechanisms.
Thank you.
So Dr. Sharma, I have a question. Like you've told us that if you use both the agents, the CTLA-4 inhibitor plus the PD-1 or the PD-L1 inhibitor, then you augment the immune response and the clinical response. So why are there so few indications of the combination, other than melanoma and maybe RCC? You don't have too many approved indications for the combination, but you have a whole bunch of indications for anti-PD-1 or anti-PD-L1. So why is the combination field?
I think
is it only toxicity or is it something more?
No, I think it's definitely toxicity is a key reason, right? The higher the toxicities, then it's not going to be an issue. But anti-CTLA-4, anti-PD-1 is approved in lung, renal cell carcinoma, melanoma, and I think there are a couple of other approvals that were recently announced for some other tumor types as well. But it is broadly used, and honestly, if the patient can tolerate it, if the toxicities are okay, we've seen that that's good, but it does have a higher frequency of toxicity.
So I think one more question. Yes, please.
Yes.
Please go ahead. Yes.
If there's time. Great talk. I had a question about the bone met example. So you gave great data on how TGF-beta, which is local, basically effectively leads to it not responding. Similarly, patients with liver mets also have reduced response rate, but that, to the best of my knowledge, correct me if I'm wrong, seems to be more systemic. So I was wondering if you could speak to the liver mets, where it seems like there might be also local suppression, but more of a systemic effect. And if I understood you correctly, that's not what you see with the bone, where it's a localized effect.
So the bone metastases are really due to the TGF-beta and osteoclast. But the liver, there's been data to show that the liver metastases may be more resistant due to FOXP3 positive Tregs that accumulate in the liver. So that's a systemic way of now those T cells infiltrating into the liver. Yeah.
So I think if you could come to the center of the stage, we would like to honor you.
Oh, thank you. Should I just take my computer back then? Okay. Okay. Oh, thank you.
थोड़ा जोरदार तालियां हो जाए हमारी पद्मिनी शर्मा मैम के लिए और चेयरपर्सन के लिए। थैंक यू सो मच। हाँ, कमरा गूंज उठा बिल्कुल ना। थैंक यू। So for our first keynote session, I would like to call upon the chairperson, Dr. Tapan Saikia sir, medical oncologist from Mumbai, and Dr. Nilakshi Mahanto ma'am, medical oncologist from Guwahati.
A very good evening to all in the audience, and it is indeed my proud privilege to chair this session with Dr. Tapan Saikia sir, my guide and my mentor, and also to introduce before you none other than the next speaker, Dr. Sangeeta Goswami. Dr. Sangeeta Goswami has taken a long trajectory from a small city of Guwahati in the remote northeast corner of the country, India. And I'm proud to say that she was my student at Guwahati Medical College. It is indeed my moment to introduce her to you. And not only that, I was even more privileged in 2019 when I went to MD Anderson Cancer Institute to actually see the place where she works with Professor James Allison and his team, and the incredible work that they have done in the field of immuno-oncology. Today, Dr. Sangeeta Goswami, she is the assistant professor at the Department of Genitourinary Medical Oncology at the Division of Cancer Medicine, University of Texas, MD Anderson Cancer Center, Houston. She is going to talk on targeting epigenetic machinery to improve response to immune checkpoint therapy. Dr. Sangeeta Goswami, the stage is yours.
Okay, it's working now. First of all, thank you so much, Nilakshi ma'am and Topanda, for this kind introduction. And it means a lot to have both of you as a chair of this session. Before I start, I would like to thank Jyoti and Dr. Bishwas for this, and the entire organizing committee for this kind invitation to speak today. And there has been a little bit of a change in my title today. Today I'm going to speak on reverse translation strategies to improve patient-specific response to immune checkpoint therapy. And you heard the basic concepts related to reverse translation from Dr. Pam Sharma's talk. So as we know, based on the pioneering work of Jim, there has been a paradigm shift in how we treat cancer based on his work on how to target CTLA-4. And you will hear more about this historical journey from his talk tomorrow when he, one of the keynote speaker talk that he's gonna give. So eventually, the field, we would see that we can target multiple immune checkpoint therapy, including PD-1, and this, sorry. So this led to an avalanche of FDA approval of immune checkpoint therapies in multiple tumor types, as listed on the right side. But here what I would like to highlight that there are multiple immune checkpoints that exist, and all the FDA approvals that we have so far is only targeting 3 pathways: CTLA-4, PD-1, and most recently LAG-3. So with this, we can say that immune checkpoint therapy has established itself as a fourth pillar of cancer care in the rank of surgery, chemotherapy, radiation therapy, and targeted therapy. And one key distinguishing feature that immune checkpoint therapy has and not others is the durability of response that we see even in the advanced setting. So it has been, immune checkpoint therapy has conferred durable clinical response, including cure, but however, response has been limited to a cohort of patients. So definitely we are here, right, where we are, like we definitely improved the response, so we definitely improved the tail end of the curve, but the aspirational goal is here, the red curve, that where we can maximize the number of patients who would benefit from immune checkpoint therapy and get the durability of response that we see in subsets of patients. So to do that, we have been running hundreds and thousands of clinical trials across different tumor types. But we are not being able to move the needle as we would have liked, because one issue that Pam brought up is that gap between the clinical trials and the understanding of fundamental science, like why some patients are responding while others are not. So we basically have to learn from every patient in order for us to design a patient-specific treatment regimen. And that is the goal of my clinical research as well as my fundamental research that I have been doing in MD Anderson. So one concept that is initially developed by Dr. Pam Sharma, and we have been a big advocate of this, is reverse translation. And what do we mean by that? As Pam alluded, we need to learn from every patient. So it is an iterative cycle where we run small clinical trials which are tissue-rich so that we can understand the pathways of response and resistance that would help us to form the hypothesis. And that can be tested in preclinical model as well as using computational algorithm. So this iterative cycle is really important for us to move forward. And in today's talk, I would like to give two examples of how we have used this concept in order to develop biomarkers as well as rational combination therapy. So before I move forward, that when I said that we need to maximize the number of patients who would benefit from immune checkpoint therapy, we have to take a step back and understand what are the challenges we are currently facing. One of the challenges is the resistance. Either it's a primary resistance, there's certain tumor types like pancreatic cancer, GBM, they do not respond to immune checkpoint therapy. And many a times, in bladder cancer, kidney cancer, where immune checkpoint therapy works, but patients eventually develop resistance, that's adaptive resistance. What are the pathways of this resistance mechanism? And the other big issue, and we have heard a lot of talk, is that we don't have any predictive biomarkers. We are giving anyone who is eligible for this one how we can enrich our patient population for biomarkers. So here, I would like to give my first example how we came up with a strategy to treat adaptive resistance. As Pam alluded, like she ran the first neoadjuvant trial back in 2006, and that shows the safety and efficacy of using anti-CTLA-4 in the neoadjuvant setting. And we collected sample, although I was not a part of her group at that point of time, but I had the access to tissue when I joined her lab as a fellow during my medical oncology fellowship. So using those samples, what I noted that patients who receive anti-CTLA-4, there is a change in the epigenetic landscape of those T cells. And one of the enzymes that was going up was EZH2, and it was true for across different tumor types. Now the question, and we formed the hypothesis that EZH2 most likely doing something in the T cells, but what it might be doing, because when T cells are activated, as we know, the signal one is TCR, signal two is CD28, and when there's a CD28 signaling, the EZH2 increases. And it is an epigenetic enzyme which causes histone methylation, leading to gene repression. So anti-CTLA-4 which increases CD28 signaling, should also increase EZH2 expression. And that is what we saw in our patient samples. Now the question is, what is EZH2 doing in T cells? The reverse translational strategies, then we went back to the lab and we did a series of ex vivo and preclinical experiments where we found actually EZH2 inhibition improves CD8 T cell cytotoxicity, and on the other hand, its inhibition, it suppresses the bad guys, Tregs. So cumulatively, it should help anti-tumor immunity. So EZH2 is basically what it's doing. It's stabilizing the regulatory T cells, and it is inhibiting CD8 T cells. And in the normal homeostatic setting, this is important, right? Because in order to prevent autoimmunity, when a T cell is activated, we should always have a negative or a break to calm it down. And that is what EZH2 is doing. But in the cancer perspective, it is actually reducing the effectivity of anti-tumor immunity. So therefore, we hypothesize that can we inhibit that to improve response to immune checkpoint therapy? And that's what we saw in preclinical murine models, that when I combine anti-CTLA-4 with EZH1/2 inhibitor, there is improved survival and improved immune checkpoint therapy, improve in the tumor response. But as we all say, we are not here to treat mice or cure mice, but we are here to cure human, right? So with that concept, we took it back to the clinic and we designed a small clinical trial in patients with genitourinary malignancies who have progressed with prior anti-PD-1 or anti-PD-L1 therapy. So patients were receiving 2 doses of anti-CTLA-4, 3 mg per kg, 6 weeks apart, and EZH1/2 inhibitor from Daiichi, which is an oral medication, and they were getting on a daily basis. And we had 2 mandatory biopsy, pre-treatment biopsy, and after 2 cycles of anti-CTLA-4, and 1 optional biopsy on progression. So this is one anecdotal evidence of one of my patients who went on this trial. The patient was an RCC patient, heavily pretreated, 4th line of therapy. When patient was started on this one, this is the pre-treatment CT scan, as you could see on the left side, and this is the post-treatment, that the combination of anti-CTLA-4 and EZH1/2 inhibitor did provide good clinical benefit to this patient. And cumulatively, so far we have treated almost 20 patients with metastatic kidney cancer who have progressed with prior anti-PD-1 and TKIs. And this curve, the green bar, shows the responders. And I wanted to highlight these patients went on to this trial and after like 4 or 5 lines of therapy for renal cancer. And this was really encouraging to see this preliminary data that we had a very significant clinical efficacy in these heavily pretreated renal cell carcinoma patients. So as I mentioned, that we also need to learn what has been going on, because it's not that everyone responded, right? So why some patient responded and why some patient didn't respond. And therefore, we did all the correlative analysis at a single cell level so that we can generate those hypotheses and move it forward. So one thing what we found is, interestingly, and we have talked about CD4 and CD8, but now as an immunologist, we know that CD4, CD8 do not just stay in one state. They are highly plastic and there could be different subsets. So when I looked at this data, initially the first instinct was that, okay, non-responders most likely did not have enough T cells infiltrated, and that's why they didn't respond. But surprisingly, if you could see the top panel here, both in the responders and non-responders, the pre-treatment, they all had effector T cells infiltrated there. However, when I compared the pre-treatment responder and non-responder, I saw there's a specific type of CD8 T cell called progenitor exhausted T cells, and they were more enriched. And why are we interested is because now we know, as I mentioned, that T cells do not stay as like CD8 effector. Once, when they get infiltrated in the tumor cells, they are they are affected T cells, but once they keep on fighting, they get exhausted and they cannot fight anymore. And that is when, even if we have a lot of CD8 T cells, they might not work. So, but there is a specific subset, like progenitor exhausted T cells, which can be reinvigorated to respond to immune checkpoint therapy. And that is what was enriched in the responders. And when you give anti-CTLA-4 or immune checkpoint therapy, and we are seeing response to that. So we are kind of in the middle of putting this paper together, manuscript together, and we are also looking at the spatial, and I don't have much time to talk about that, but stay tuned. This is another way of, it's a visually showing that the progenitor cells were higher in the responders pre-treatment, and they kind of moved to an effector state, as you could see here. It's more enriched in the responders than in the non-responders. So overall, from this part of my talk, I wanted to highlight the importance of reverse translation and how we should learn from every patient to understand what are the resistance mechanism and how we design rational combination therapy. So in this part of the story, we made a clinical observation that anti-CTLA-4 treatment increases EZH2 expression on T cells in patients, and that is an adaptive resistance mechanism. So if we target EZH2, we showed that with the preclinical model, and now currently we are validating with our clinical trial. The combination of EZH1 and 2 plus ipilimumab is safe, and we are seeing initial clinical activity. And this is basically summarizing everything that I have said so far. So now I would like to switch my gear and talk a little bit about predictive biomarker and how should we think about enabling patient selection. So here I would like to give an example of bladder cancer, because that is cancer type that I often treat. And what we have seen that in bladder cancer, especially in advanced setting, out of top 5 mutations, the top 3 mutations are epigenetic pathways. So how are they regulating the tumor initiation or progression has been studied in the field, but how are they regulating response to different therapies is not well understood. So we did a retrospective analysis of 2 big clinical trial cohorts, Checkmate 275 for nivolumab and IMvigor 210 with atezo. And what we found that patients harboring these 2 mutations, ARID1A and KDM6A, have an overall survival benefit when compared to patients who did not have these mutations in response to immune checkpoint therapy, as you could see on this slide on the left. And then, and also in the, this is like IMvigor and this is Checkmate. One is for ARID1A and one is for KDM6A. Basically, what we found that patients harboring these 2 mutations are responding to immune checkpoint therapy better than patients who did not have this mutation. So then definitely we can use it as a predictive biomarker, but we know that single biomarkers have often failed to appropriately predict response to immunotherapy. I mean, so far we have relying on PD-L1 expression to give, but we know that PD-L1 is an inducible molecule, right? Then we tumor mutation burden, but we have seen patients even with a high TMB responding to tumor. Same way that immune-derived like CD8, tertiary lymphoid structure, which are good biomarkers, but tumor microenvironment do not work in silo, that we can have only one biomarker to predict a response. So what we hypothesize, maybe like we should look at the both tumor inherent factors and immune factors and combine those biomarkers to have a more predictive capacity. So to do that, as I just mentioned, that PD-L1 was a good start as a biomarker, but it is not had a good predictive capacity because interferon gamma, when there is interferon gamma in the microenvironment, even a PD-L1 negative tumor can become a PD-L1 positive tumor. So not to give someone immune checkpoint therapy because they didn't have a PD-L1 is, I have found in my clinical practice is not the best way to move forward from this biological perspective. And across different tumor types, their predictive capacity hasn't been very reproducible. So what we did, we looked at other immune biomarkers. And one biomarker that we found is CXCL13. It's a chemokine, and it is important in bringing T cells and B cells into microenvironment and forming TLS that Pam alluded to. And what we found that patients with high CXCL13 in the pre-treatment, they respond much better compared to patients who did not have high CXCL13. So next question that we asked, like, can we combine an immune biomarker and a tumor cell-specific biomarker, and what is the predictive capacity? And here you can see on the left, the patients with ARID1A mutations plus CXCL13 high expression have much higher response to immune checkpoint therapy compared to patients without either of the biomarkers or only with the single biomarkers. And we showed that in the IMvigor and also for KDM6A as well, that combination biomarker is a way to go because they have higher predictive capacity compared to single biomarkers. So to test that, we are doing the first trial of predictive, combinatorial predictive biomarker trial in bladder cancer. So patients with documented ARID1A or KDM6A mutations were enrolled in this trial, and they are getting Nivo or Nivo-Rela, and we are testing the response based on the mutation and mutation plus CXCL13, and also performing all the correlative analysis. So now, as I said, that we have a lot of tumor inherent biomarkers that we have discussed about. We also have immune-specific biomarkers that we have been testing in the field. But what we should be thinking is, well, how do we combine these two to improve the predictability or enrich our patient population? And based on our studies, we found that ARID1A or KDM6A plus CXCL13 is a combination. It has a strong predictive capacity. So finally, to conclude my talk, I would like to again highlight is a reverse translation strategy, possibly a path forward for discovery science and bridge the gap of clinical research and fundamental research so that we just don't randomly run clinical trials just because something works in melanoma. It doesn't mean that it's going to work in prostate because it's a very different microenvironment. So how do we run small trials because of financial reasons, but we learn from every patient based on their tissues, based on their pathways, and we can predict that what should be the next step for the patient. And this is an example of one of my patients, is that she had metastatic papillary plasmacytoid bladder cancer, very aggressive one. She had a lot of comorbidities. I could not give her chemo. I did her next-gen sequencing, and I found she has ARID1A mutation. And this was 5 years ago, and I had a heart-to-heart discussion that let's try single-agent pembro. And it's been 5 years or more. There's no evidence of disease with a single-agent pembro because she had ARID1A mutation. And it stemmed from my research. So as a physician scientist, that is definitely a pretty rewarding moment. So that is why I think it's important to understand from every patient what it's saying and to develop rational combination therapies. So finally, I would like to thank everyone mentioned in the slides, my lab people and my mentors, Jim and Pam, GU Medical Oncology, and all my funding sources, but last but not the least, patients. And thank you so much. I would be happy to take any questions if you might have.
Thank you, Sangeeta. Clever concepts, smart hypothesis, serendipity, or reverse serendipity in your case, I should say. And keen eyes, sharp minds, and the great teamwork can lead to a lot for the human beings. Questions, please.
Thank you. Thank you.
Nice talk.
Thank you.
I'd like to ask you, did you see changes in the transcription factors, TOX, and NFAT, when you're looking at the exhausted T cells? Did you see changes in these transcription factors?
Yes. I mean, that is how, in the single cell data, that is how we annotated those cells based on their transcription factor, TOX, TCF, 7 and others, So that is the single cell helped us to delineate those or annotate those cells, and that is when we found that the affected T cell populations were very similar in both responders and non-responders, but it's a progenitor exhausted population who can be reinvigorated was responding better. Most likely, if you do not target them with an immune checkpoint, they might eventually become exhausted as well. But if you target them, and there new literature is coming up in immunology that is a very critical population in response. So that is why just having CD8, when the way we have been thinking, right, that CD8 high, so patients should be responding, might not be the best way to move forward. But yes, to answer your question, we looked at the transcription factors to annotate those cells.
Thank you.
Hello.
Yeah. Oh, hi.
It's a nice talk.
Thank you.
Maybe I have missed one thing, that CXCL13, that is one of the major factor related to tertiary lymphoid structure, TLS. And it is TLS responds good with this anti-PD-L1 therapy. And you mentioned that ARIDA1A mutation and KDM6A mutation. So whether these two factors has role in TLS structure formation?
That's a very good question. So question is it synergistic or additive, right? We have done some preclinical work, but the issue with the preclinical work, that mice really do not develop TLS because it's such a fast effect. ARID1A and KDM6A mutation, I didn't have time to go over all the pathways, they can induce interferon signaling in tumor cells. So even if they do not have a direct impact, but they could have like indirect impact through interferon pathways in kind of forming the TLS. But whether ARID1A directly induces CXCL13? No, they do not. Based on our work, we didn't see that. So it's most likely additive.
Okay, thank you.
Thank you. I really don't know it's a coincidence or not. There are 3 Assamese people, a small community. One is completely uprooted in Mumbai, one is uprooted in the United States, and one remains rooted in our hometown. I think it's nice to meet together here.
Yes,
thank you so
really
much.
Nice to thank you so much. Thank you.
Thank you.
Tapan sir and Nilakshi ma'am, I would like to request you to please come on stage and felicitate. थोड़ा जोरदार तालियां हो जाए, तालियों की गूंजों से अच्छा लगता है. थैंक यू. Almost like tip over. Thank you, ma'am. Thank you, sir. So for our next keynote session, so I would like to invite our chairperson, Dr. Roshni Bhagwat Pandey, ma'am, and Dr. Anupama Borkar, ma'am.
So good evening, everyone. I invite Dr. Betty Kim, Professor, Department of Neurosurgery, Division of Surgery from MD Anderson Cancer Center to deliver her talk on targeting epigenetic machinery to improve response to immune checkpoint inhibitors. Welcome, ma'am.
Thank you for that kind introduction, and thank you so much for the organizing committees of I-OSICON and also Dr. Jyoti Bajpai for the invitation. So I actually am going to talk about immune engineering approaches to cancer therapy today. So you're all probably wondering, what is a neurosurgeon doing at an immunotherapy place? You know, at MD Anderson, I'm a professor, and clinically I work on patients and take care of patients who have primary and metastatic brain tumors. And so that's been sort of a 15-year journey with all the training and taking care of these patients. But as a surgeon, I was very frustrated by the fact that we weren't really changing the survival curve on many of these patients. Now, with the advent of a lot of technologies, we have intraoperative MRI, where an MRI becomes sort of part of the operating room where you operate, and the patients go back and forth from the MRI and ensure that we have maximal safe resection. We also have intraoperative ultrasound. That is probably the only technology that allows us to get real-time feedback of how much we've taken the tumor out. But at the end of the day, the patient always recurs, and they all succumb from these illnesses. And unlike any other parts of the body where I think a surgeon can take out just a little bit more of that tumor to get a better resection, you know, in these areas where we're really flanked between the speech versus the motor, where a millimeter or two of taking out that extra tumor bit may result in the patient waking up with a lot of speech deficit or paralysis of their face and arm. And so we're really limited about what we can do as surgeons. And so I decided as a student that I would sort of look into different areas of research. And at the time I thought that maybe thinking outside of the biological world and maybe looking at biomedical engineering, looking at ways to develop toolkits that allow us to develop better therapies, for example, would enable me to be a better physician and to go back and forth into clinical versus the lab and back and forth. And so this is this is going to be the first introduction to a target compound that we've developed, and hopefully we can translate this to clinical trial. So here we have sort of my lab focus right now, and I'm going to talk about today an innate immune checkpoint enhancer that we've developed through our knowledge of protein engineering as well. And so as we know, there are a lot of old challenges in terms of cancer therapy and how we can sort of beat this disease. Tumors are very heterogeneous, whether it be between patients having the same sort of tumor called glioblastoma, which is unfortunately the most common but the most lethal form of primary brain tumors in the adult. But they also change over time, and so you have to sort of develop therapies that can sort of be agile and sort of develop with the journey of this cancer. But we also have issues of drug delivery. How do we get that target to the brain? And is that even relevant now with the advent of immunotherapy, where you're using your own cells and the immune cells to actually go now attack these tumors? And then, of course, we have issues of resistance mechanisms that develop, and then the toxicity as well. And as Dr. Goswami and Dr. Sharma talked about, with immunotherapy, only 20 to 30%, for example, in solid tumors, these patients respond really well to immunotherapy. And we wonder, what about the other 60 to 70% of patients? Why aren't they responding? And can we actually improve these survival curves? Well, I'd like to introduce to you that one of the first sort of reactions from an immune response is the myeloid compartment, the innate immune system. And we wondered that a cancer cell is usually cleared initially by the innate immune system, the myeloid cells that actually go and gobble them up through the pathways of phagocytosis. And they're regulated differently depending on the situation, depending on the type of tumor, whether you're a solid tumor or a liquid tumor, for example. And fortunately or unfortunately, a lot of these cancer cells have these markers that cloak them from our body's immune system, such as CD47, which is overexpressed in many solid tumors, that prevent our myeloid cells from recognizing them. And so, of course, there have been antibodies that block these markers of don't eat me, that allow myeloid cells to go and sort of clear initially these cancer cells. But unfortunately, as a single agent and in patients, despite multiple clinical trials, really have not been effective. And we wondered, can we use some of the knowledge that we have for myeloid checkpoint blockade that we have and sort of use a way to allow the processes of antigen presentation to ultimately T-cell priming and T-cell activation be a little bit better in these patients. With every pathways that we have, whether it be for immune system or any other pathway, there's always this yin and the yang where there's pro versus anti. And these are some of the markers that our lab is really focusing on. One is the don't eat me sort of pathways, where can we block this pathway? We know about the PD-1, PD-L1 pathway, the CD47, B2M, but we also try to sort of change the balance where can we actually heighten the eat me pathway, such as the calreticulin and the SLAMF7 that we're working on. So here is sort of a complex slide, but I do want to sort of highlight some of the key elements of our technology that will be very relevant. One is that as the cancer cells, as I mentioned, overexpress CD47, that we can develop antibodies to block it, but that blockage allows the cancer cells to enter phagocytes. And what happens is that they're engulfed and they enter these phagolysosomes. And the goal of these phagolysosomes, in general, is to try and digest whatever they're engulfing, whether it be cancer cells, whether it be infection, or any sort of foreign attacks on the body's homeostasis. What happens here is that in the case of cancer cells, as they're being digested, we hope that the tumor DNA that's been digested are somehow leaked into the cytosol, the innate immune sensing pathway, such as the toll-like receptor pathway or the C-gasting pathway will be activated, allowing us to sort of allow these pro-inflammatory cytokines to be released, to allow T cells to be activated, as we are also trying to load the tumor antigens onto the MHC to be recognized by the T cells, and then allowing the adaptive immune cells to be engaged. Well, unfortunately, this entire process overall is very inefficient. You're just hoping that there's a lot of these tumor antigens that are going to be released. And the phagolysosomes are quite efficient in the way that their goal is to degrade as much of this as possible and get rid of it. And so here we have like a schematic here, where if you have a myeloid cell, like a macrophage, near a cancer cell, can we allow increased phagocytosis to occur? And initially we thought that would be sufficient, but it is not. And so if we actually allow more phagocytosis to occur, can we have more tumor antigens to being presented, and then having T cells, there we go. And then allowing T cells to be engaged and kill off the remaining cancer cells, with the subset being transformed into memory T cells. Well, one of the areas that are really hot right now is the antibody drug conjugate technology, or the platform. I'm having trouble with this here. And that is that you have the targeting agent, an antibody, for example, that you link it with a drug, a cytotoxic sort of drug, and the goal is that you would load the target itself, the cancer cell, but allow the drug that you're loading to kill off and be cytotoxic. Unfortunately, there's been a lot of issues in this realm, in the sense that you're not really engaging the immune system. You're really directing your entire therapy on direct cell killing. And so you wonder if there's a different way of thinking about ADCs. And this is sort of our, it is not published yet, but this is the first time that we're presenting this to the live audience. And that is that if we have an agent like CD47 antibody that should allow this process of phagocytosis to occur initially, well, we can possibly then link that with a toxin. And we just serendipitously discovered through literature that a bacteria called Listeria monocytogenes have a toxin called LLO, or Listeria lysine O. And this toxin allows this intracellular bacteria to enter their sort of the host And it hijacks the cytosol by allowing holes to be placed or pores to be placed on the phagolysosome membrane. And the contents of that phagolysosome get leaked out into the cytosol, and that bacteria then is allowed to sort of live inside these hosts. So we wonder whether we can allow LLO to be conjugated to the CD47 antibody, allowing the CD47 to be to allow the phagocytosis to occur, and whatever the tumor cell, when the tumor cells enter the phagocytes, it'll be leaked into the cytosol as it's degrading. And so you'd have increased tumor antigens being exposed to the cytosol. You'd have tumor DNA being exposed to the cytosol. And could that ultimately lead to increased tumor engagement with the T cell? And so here we have a schematic here where the phagolysosome, through the processes of LLO, where the pores are forming, the contents of the phagolysosomes get leaked into the cytosol, where peptides, tumor antigens are basically exposed into the cytosolic machinery, allowing the MHC to load the tumor antigens in a more efficient manner. You have the DNA of the tumor being exposed for innate immune sensors to be activated. And so I just wanted to show you in a sort of a quick schematic here that we were able to link the CD47 antibody to LLO, show that in a reducing condition, which is what you would find in the contents of, within a phagolysosome, which is a low pH environment of approximately 5.5 or so, that it is cleavable. The LLO cleaves off, and once it's cleaved, then the LLO dimerizes and forms these pores on the walls of the phagolysosome, allowing the contents of that phagolysosome to be released into the cytosol. So we see that here as well. And then this is just to show that with the CD47 LLO, that you do have the bone marrow-derived macrophages engulfing the cancer cells. And this SEM image basically showing that when we label CD47 conjugated with LLO with a gold nanoparticle, just to mark them, that we see them sort of being released into the cytosol, these little spots there, the CD47 LLO presumably into the cytosol, compared to the CD47 alone, where they're all sort of stuck in the phagolysosome, for example. And this is just to show that that CD47 LLO is able to get into the cytosol and no longer within the phagolysosome as tracked by the lysotracker system. So then we do a series of sort of experiments to just show that we have heightened antigen presentation. We have heightened increased CD4 and 8 infiltration into the tumor bed, and that the T cells are antigen specific. What I wanted to show you is this exciting sort of data, where we do this experiment where we treat these animals, and we have multiple disease models now, whether it be breast cancer model, pancreatic model, melanoma model. These are syngeneic orthotopically injected tumors. And this is an example of a breast cancer model where we treat one side with the tumor, one side of the tumor with the CD47 LLO. And we basically see that on the contralateral side, the tumor just doesn't grow. So we've essentially made that animal vaccinated, if you will, from growing any more tumors. And we have also models of metastases where the animal has florid, sort of multiple tumors all over the body. And we inject this into the largest tumor that we can access intratumorally, and the other sites essentially shrink. And so it's not a vaccine per se in the classical sense where it's preventing disease, but you'd have to have the disease first to allow your body to create these wide repertoire of T cells that are going to recognize the tumor antigens and be able to eradicate these tumors. This is just to show that we have multiple disease models. It's just fresh out of our lab, so we don't have a lot of the longer survival curve. But you can see that with the green and the green and red curves here, that's a survival curve, definitely is beneficial with currently intratumoral and intraperitoneal injections, and we are formulating a different sort of, like a version 2 of the CD47 LLO in which we hope to inject systemically in a safe manner with reduced toxicity. And this is just to show that when we look at the subset of myeloid cells, we took sort of the markers that are expressed using the markers that are already sort of in published literature, and we categorized them into sort of 10 different categories. And when we looked at all of the gene expression markers, there was a subset, the clustered sort of 9, we're arbitrarily calling them cluster 9 subset, where they're very pro-inflammatory. Right here. I don't know if you can see this here. They're very pro-inflammatory. There we go. Pro-inflammatory in this, and the amount, it goes from 5% to about 30 to 40% with the CD47 LLO treatment, and we essentially dissected this group 9 further. And what we show here is that number 1 is that if you see cluster number 9, the gene expression profiles that we see are all of the pathways that are involved in sort of being the mediators between the phagocyte and the T cells. So you can see here we have chemokine signaling, we have the JAK-STAT signaling pathway, we have T cell receptor signaling pathway, toll-like receptor pathway that are all lighted up in this particular subset. And so we're actually performing a deeper dive into what these macrophages are. But we also show that compared to just CD47 alone, this is basically a graph. All of these lines sort of signifies interactions. And the thicker the line, it means that there's greater amounts of interactions, if you will. But if you see here with the CD47 LLO, which is in this column, that our inflammatory macrophages, the cluster 9, really has a lot of interactions with their T cells. And what's important to note is that actually the T cells then also feed back and has significant interactions with the macrophages themselves. And so these series of experiments told us that macrophages are not just phagocytes and we just stop there. They're not just antigen-presenting cells and we stop there and we just hope that the T cells will somehow come in and sort of do all of its wonderful job. There is this subset of macrophages that we can actually increase with our therapy that can make the entire the tumor environment be a little bit more like pro-inflammatory, allowing the T cells to work a little bit better and engage with them a bit better. And so our summary is that this first sort of antibody drug conjugate, if you will, is designed not to kill the cancer cell directly, which is what's out there, where the cytotoxic drug that's attached to the antibody sort of kills the cancer cell. This product is not cytotoxic, but it actually allows that immune system to become hot, if you will. And we believe that this is sort of a new way to quote-unquote vaccinate the body once the body has this sort of tumor, where the T cells that you're going to be forming with this exposure will allow you to sort of prevent possibly further mets from forming. And because you have this wide array of T cells now, perhaps this development of mutations or decreased expression of certain genes or whatnot, that may not be as relevant or as important. But we're sort of looking into this further. And we hope that this type of a mechanistic first principles design of an immunotherapy product can be sort of the goal of the future. We're looking at first mechanisms, but we're then developing these novel therapy that can possibly open doors to new avenues and new ways that we think about cancer therapy. And perhaps this is the start. We're hoping that this is the start of a bright new future ahead. And so this is my lab. This is Benjamin Schrank, who did majority of the work presented today. He's a physician scientist in training, soon to graduate, and our laboratory here, and of course the generous funding by these institutions and support at MD Anderson as well. Thank you for your time.
Thank you, ma'am, for that work in progress. I wanted to know that anti-CD47 antibody, that is the same one which has been used in myeloid malignancies, right? And which hasn't failed. So this is only used as a vehicle to induce the phagocytosis? And what was the reason to select breast and pancreatic tumors? Is it because they are cold tumors?
Yes, so you're correct. It's a CD47 as a monotherapy. Doesn't really work, right? And it has toxicity effect, especially in the bone marrow. And I think the issue is that it has worked in sort of the preclinical model, but in patients and in clinical trials, you've had to add additional therapies that become even more toxic. And what we're suggesting here is that we've developed this antibody in such a way that the Fc receptor will be silent so that you don't have this ADCC-like reaction, number one. And we're not using it as a pure phagocytosis agent. And at this point, we don't really know what percentage of phagocytosis you'd need to actually get this systemic response. It's something that we need to work on. And in terms of why we chose pancreatic cancer, well, we have, as I mentioned, we have the slew of in vivo sort of models that we're using. And we thought that to get this into patients quickly, you'd have to work on something that's maybe refractory to all therapy. And we thought, can we just work on pancreas? And this is why this is something that we've been working on just in the last 6 months on the pancreatic model, thinking that this would be perhaps our first in human sort of clinical indication for this therapy.
Thank you. Are there any other questions?
Hi, great talk. So I just want to understand, can these antibody drug conjugates could also be used as adapters for, let's say, CAR T therapy to target the solid tumors? What's your take on that?
So the question is can we use these for CAR T therapy?
Yeah. Like in some ways, could we just use them as adapters or even somewhere in the design? Or do you see any immunoengineering approaches where we could complement them and use them in solid tumors?
Yeah, so I guess, so the reason why we were working on the myeloid cells was to try and avoid the ultimate last step, which is really making sure that the T cells are strong, they're not going to get exhausted, they're going to be able to recognize, in a wide range of tumor antigens. And I think you don't need the CAR T cells, actually, if you do this route, because you now have T cells in your body that's going to recognize not just the one antigen, but like thousands of antigens now that's related to that cancer cell. And so I don't think you need to marry this with CAR T cells, because CAR T cells will recognize that one sort of antigen, and I think it's already, your body would have already sort of generated that, if you will. Can you marry this with other immunotherapies? Yes, and I don't show it here, but we've seen significant, even improved survival when we do combinatorial therapy with PD-L1 and PD-1 checkpoint blockade. And there was a rationale behind it, as we saw a lot of interferon gamma production, and so we thought perhaps could that be a resistance mechanism? And there's an increase in PD-1 expression, and so we did a blocking study that showed even improved survival.
Thanks, that's very helpful. Thank you.
You're welcome.
I would like to call upon the chairpersons to kindly felicitate Madam. Madam, please. Thank you, madam. We'll head to the next session. That is a sponsored symposium by Zydus. So I would like to invite Dr. Soumya Surat Panda, Professor and HOD, IMS and SAM Hospital, Bhubaneswar. Please, sir.
Slides. So I thank the organizing team and Zydus for this opportunity. So this is evolving role of liquid biopsy in advanced non-small cell lung cancer. So the evolution of therapy in lung cancer, as we know, it is a heterogeneous disease. So initially we thought that it was non-small cell, small cell, then we went to non-small cell being classified into squamous and adeno and large cell and other histologic variants. Subsequently, as the time went by, we understood that it is not the histopathology, but rather the molecular drivers. So the driver like EGFR, ALK, and ROS1 came into picture. And after that, then the immunotherapy came into picture, where we looked at the PD-L1 based on the immunohistochemistry. So these are the targetable driver mutations in advanced non-squamous, non-small cell lung cancer. So there are multiple mutations out there where we can target them. And why they are so important? Because the outcome is very, very different. So if you are giving somebody with lorlatinib or crizotinib compared to chemotherapy, then you can see there is a difference in survival. And besides the lesser toxicity. So be it osimertinib, be it lorlatinib, we understand that we need to deliver this. What is the problem? The problem is that right now in 2025, even when the patient is coming to us right now, we are giving them a first cycle, mostly a chemotherapy. Why? Because we are not having the report with us at a correct time. So still now we need to rely on the biopsy and again immunohistochemistry, and subsequently we'll send the blocks for a NGS. If it is not there in-house, then you wait for the report. By second cycle or third cycle, you will be able to deliver your targeted therapy if it is there, or sometimes you continue that if the patient is responding and then you switch. So there is a problem that we are not getting the report on time. So first cycle, mostly patients are going to get chemotherapy if they are not willing to wait for the period. So CGP prior to first line treatment for metastatic non-small is associated with almost 4 times longer overall survival. So we know that. So you have to go with the gene sequencing. So be it CGP, be it customized gene profiling, that's a different matter, but you have to do a gene sequencing, and without that, the survival is very, very dismal. So that part we know. Then comes the tissue biopsy problem. So what we want is that lot of material, lot of tissue, but remember we are not dealing with a breast cancer or ovarian cancer, or say for that matter gastric cancer, you do a endoscopy again and again. We are dealing with a CT guide or percutaneous biopsy, and sometimes the nodules are really deep. So there is a problem with the tissue. So we are not able to get sufficient tissue, for which we need to rely on a liquid biopsy sometimes. So when the tissue is insufficient, or say it is sufficient for a histopathology, but then it is depleted by the time you send a gene sequencing, at that point you have to rely on the liquid biopsy. So the problem of insufficient tissue is because we have to still do a biopsy and the immunohistochemistry. So remaining tissue is insufficient for molecular testing and PD-L1. So problem is poorly differentiated carcinoma. So sometimes it is very easy. It is glandular elements, it is adenocarcinoma, and the other part is squamous cell carcinoma when you see the keratin pulse. But many a times it is a poorly differentiated carcinoma, and you are not able to do a directly proceed from a histopathology to a gene sequencing. You still have to do a IHC. So in that case, it eats away around 3-4 days, and again you have to, after that only you will be able to send a gene sequencing. So this was a large study, demonstrated that tissue genotyping alone may not deliver timely results before the first line treatment. So in this study, 323 patients were taken into account. Out of them, 29% underwent a plasma gene sequencing or say a liquid biopsy. The other half, the blue ones, that is that our patients that what we are doing right now. So in that patients, the tissue NGS is completed only in around 60%. Other patients, biopsy is sometimes not feasible or the quantity is not sufficient. So that's a real world problem. So 44% of the patients with tissue attempted were unable to get a tissue CGP results. So evolving role of liquid biopsy for biomarker detection in, so when do we use a liquid biopsy? As I told you, you need to do it parallelly. You can do it parallelly. So you do a tissue biopsy so that at least you will do a IHC of PDL1, for which liquid biopsy still now is not an authenticated way to do that. So you will do a tissue biopsy. Along with that, you can send a liquid biopsy. So that is known as complementary approach. You will send a tissue biopsy as well as a liquid biopsy. Another is plasma first approach. You don't do a tissue biopsy, you send a plasma biopsy, but again there is a problem because you will not be able to do a PDL1 or some marker still for which tissue is required, so say fusions. And then another is sequential approach. You do a tissue biopsy, but parallelly you again, after that, if the tissue is incomplete, then you do a gene sequence, do a plasma biopsy or a liquid biopsy. That is a sequential approach. Tissue biopsy followed by liquid biopsy. Tissue biopsy plus liquid biopsy is complementary approach. So mostly we are doing a sequential approach. We are sending a tissue biopsy. It is insufficient, then we send for a cell-free DNA. So these are diagnostic algorithms for untreated advanced non-small cell lung cancer based on the ISLC. So they tell that there are 3 areas where we are doing a liquid biopsy right now. As I told you, a plasma first approach, you can do it directly. I told you about the complementary approach. You send the tissue biopsy as well as liquid biopsy at the same time upfront, or a sequential approach. You do a tissue biopsy, tissue is insufficient, then you go for a liquid biopsy. So these are three areas where right now currently we are using a liquid biopsy. So this is again the complete study, comparison of matched plasma versus tissue. So this is to find out whether the liquid biopsy that we are sending right now, that is concordant with the tissue biopsy or not. For the EGFR mutations, the ALCROS and the KRAS, it was a concordant study. There is a high level of concordance based on the tissue and liquid biopsy, CGP concordance was there. This is a Nile study, again the biomarker detection. In that particular study, it is not only that the tissue biopsy is showing a particular amount of mutation, the liquid biopsy is picking up extra mutations. So that is again a very good information. So your information might be expanded. Sometimes you have sent a tissue biopsy, but the tissue biopsy comes negative. You have done a liquid biopsy, you will find out a small amount of ROS1 being picked up in the liquid NGS. That can also happen because the tissue that you are sending is always heterogeneous. It doesn't contain the tumor tissue all around. That is a heterogeneous. So that's why in the liquid biopsy or a cell-free DNA, you can get the results more. So that is also there. So upfront cell-free DNA, next generation sequencing, this is again looking at the TAT. So if you do that of course we know that. If you send a cell-free DNA and just right from the upfront, then the median time is 8 days. So the most of the patients by the first cycle will get a targeted therapy correctly. So this is the NCCN 2023 when it realized for the first time that concurrent liquid and tissue testing approach is feasible, and that might be a way to go forward. So this is another testing paradigm. One, send a tissue biopsy, then liquid biopsy, and another liquid biopsy then send a tissue biopsy that I told you. The third is that simultaneously you can send. So initiating tissue and blood CGP at the same time captures all the alterations and 50% faster. That can be done. This was the ASCO presentation, Association of Comprehensive Molecular Genotyping and Overall Survival in Patients with Advanced Non-Squamous Small Cell Lung Cancer. In this particular study, they found out that concurrent tissue and plasma-based NGS was done. So this is a consort diagram that showed that patients underwent both a tissue and a CGP, and in the blue line that indicates the comprehensive testing. You are sending up a tissue and liquid at the parallel time, so the survival is better because you are getting the targeted therapy earlier. So that's why the survival might be better. And performance of concurrent tissue and plasma NGS testing was associated with a higher likelihood of comprehensive molecular. So your testing results are more authentic, and your comprehensive genotyping have improved the overall survival compared to patients with incomplete or no testing. And these results support implementation of concurrent tissue plus plasma NGS approach initially. So that is there. This was the experts roundtable participants where they decided on some recommendations because still now it is not standardized. So recommendation 1 was publication of guidelines ensuring standardization and quality of NGS. Recommendation 2 was enforcement of lab accreditation. So what should be the actual accreditation and the certification guidelines by the public authority bodies. And recommendation 3 is that you have to find out the labs correctly which are sending this or doing this test, because right now there are multiple labs that offer that. But remember, before going ahead with that, you have to be very, very careful. So as a medical oncologist or as an oncologist, if you are sending that, then you have to remember the pitfalls as well. So you have to know that whenever you are sending that, Suppose say in this particular case, the ROS1 was 0.04%. So it comes to the molecular pathologist that whether he should or she should take a call that this is a significant fusion and whether we should start treatment based on this 0.04%, yes or no. So there is a dilemma. It requires a molecular tumor board. You have to discuss case by case, but most likely if ROS1 is there, you might tend to give that patient the benefit of a ROS1 therapy and they might do well. However, these percentages, the cutoffs, and liquid cutoffs versus with tissue cutoffs, so everything needs to be discussed case by case, especially when your liquid biopsy results are showing a low amount of call, so that is 0.04%. Again, what is the significance? So once you do a liquid, you will find out multiple mutations, like TET2. TET2 is a chip, so you must understand that the TET2 is a chip. It doesn't carry any significance or pathogenic driver. You need not target that, so that is also again very, very important. Then what do you mean by elevated tumor fraction? That is not detected. So when you send for a NGS, there are things that you must look into a report, how to interpret that NGS. You must have a good molecular tumor board, discuss these cases, and then take a call when it comes for therapy. This is again MET overexpression. So MET overexpression, we know that the ISC, when it is 3+, we'll call it MET positive, or say MET amplification by FIS, the copy number more than or equal to 5, or a signal ratio of more than or equal to 2. So these are the FIS of 5+. So these are the areas where you'll call a MET overexpression. But when you are doing a NGS, the cutoff is different. So for NGS-based results, a copy number greater than 10 is consistent with high-level MET amplification. So normally we do a ISC, normally we do a FIS, but again NCCN now looks at the MET definition based on the NGS. So you have to be really careful when we are interpreting the reports of a NGS. Before sending the test, just don't send it right-left. You must understand the implications of sending a test, which is especially in our Indian context is costly. So with that, I conclude. Thank you very much.
Thank you, sir. Thank you for the crisp talk. Now we'll quickly head to the next symposium. Now we'll quickly head to the next symposium. That's a sponsored symposium by DRL. I would like to invite a speaker, Dr. Amit Dilip Bhatt. He's a medical oncologist from Pune to give his talk on new treatment advancement in nasopharyngeal carcinoma. Dr. Amit, sir.
Good evening, everyone. Thank you so much for inviting me to speak over here. Thank you, DRL, for the slides. After a gap of almost 5 or 6 years, we finally have a new immunotherapy drug to play with. We have been using immunotherapy since the last almost 8 or 9 years, earlier before that in clinical trials, and now we have a new drug. We're going to talk about the drug in detail. So this is, when we talk about nasopharyngeal cancers, it's a very common disease in East India, where we see a lot of patients, also in South India, and mainly China. Why China? Because this drug is from there. If you look at the incidence of cancers in India, the breast is the topmost among all sexes, followed by the oral cavity. So we see oral cavity almost day in and day out in our OPDs, and mostly we have oral cavity, and nasopharyngeal cancers is a bit lesser in our routine practice. If you look at our data, nasopharyngeal incidence is rare, it's less than 1 in 10,000. But if you look at the Northeast, especially Nagaland and Kohima, it is almost pretty high. The same is the story with the South India. In CMC Vellore also, we used to see a lot of patients. So when we talk about recurrent metastatic, what are the treatment options? When the patient comes upfront, neoadjuvant is one of the key to treatment of nasopharyngeal cancers. Given the stage where it's locally advanced, usually it's a chemotherapy regime. The TPF is the standard one, docetaxel, cisplatin, and 5-FU. But when it comes to the metastatic setting, we are short of options, and we are just try to play with some chemotherapy drugs, hoping that the patient will really get some response. The only approved drug which finds the regimen which finds itself in the NCCN guidelines is cisplatin and gemcitabine. So let us look at this. The CISGEM, it's the trial. It's a 2016 data, long back, almost 10 years now. And it is gemcitabine cisplatin versus the cisplatin 5FU, which is the standard of care in head and neck. And look at the PFS, hardly 7 months versus 5.6 months. So this is not really an impressive achievement when you talk about NPCs. Even the overall survivors, barely patients managed to reach 2 years, even lesser than that. So achievement of a survival of more than a year and a year and a half was dreamed about in NPCs and really not achievable. This gave the need to have new drugs, new options in treating the NPCs. A lot of immunotherapies which we have with us today, like in other trials, immunotherapies have been a really great help in RCCs and lung cancers. They have revolutionized the treatment in RCCs. Almost every cancer for that matter. You have almost 10-50 indications on an average for every immunotherapy. But when it comes to head and neck cancers, there was something lacking. The known immunotherapy drugs which really fared very well in head and neck other cancers did not do much well in head and neck cancers. There were a lot of failed trials and hence the new drug. Why failed trials? Most of the trials were generalized head and neck cancers, not specific for NPC. And that's why we didn't have really great data for NPC. If you look at that, pembrolizumab, which was commonly used, did not significantly improve the overall survival PFS or overall response rate as compared to chemotherapy in the recurrent and metastatic NPCs. So this is where the rationale and the need for the new drug comes. And we are going to talk about the new drug. The new drug is called toripalimab, now introduced and available in India. We call it an immune checkpoint inhibitor with a difference. Why with a difference? We are going to elucidate on that a bit. It is the first and only FDA-approved treatment for recurrent locally advanced and metastatic nasopharyngeal carcinoma. So this is very important. It is the first and only approved drug. Toripalimab, it's a IgG4 kappa monoclonal antibody. It is used as an IB infusion and it's available in 240 mg vials. Why this 240 mg? Because all those of us who have used immunotherapy have that issue of drug wastage. It's 3 mg per kg and it's again fixed dose and a lot of drug is getting wasted. Here one advantage is that the vial comes in a 240 mg vial and the dose is also 240. So there is no drug wastage. We are all using vials multiple use many a times, but this is where we are saving the drug. So this is the approval. FDA approves toripalimab in nasopharyngeal carcinoma. And this is together with the cisplatin gemcitabine arm. So it is chemotherapy plus immunotherapy combination. And it is approved upfront for recurrent or metastatic NPCs. Now, what is the difference? What is the reason of success of toripalimab when other immunotherapies failed? Now, toripalimab blocks PD-L1 and L2 interaction and binds to a different epitope as compared to Nivo and Pembro. And this is pharmacokinetics. The real benefit is that it has high affinity than Pembro and Nivo, and the dissociation is slow. So it binds quickly, binds to more sites, and slower the dissociation, more the response, longer the response. So that is an advantage over the existing immunotherapy drugs, which this new drug gives us. And let us look at the data. The Jupiter 02 randomized clinical trial. This is toripalimab plus chemotherapy. So inclusion criteria was metastatic NPCs and recurrent NPCs, good performance status, and they were treatment-naive diseases. Those were fresh patients. How were they randomized? They were randomized into toripalimab 240 mg fixed dose plus gemcitabine 6 and cisplatin once every 3 weeks for 6 cycles, followed by toripalimab maintenance 240 mg every 3 weeks. The control arm was placebo plus the same chemotherapy arm, followed by placebo maintenance. The primary endpoints were PFS, secondary endpoints were PFS as per investigator, overall response rate, duration of response, disease control rate, overall survival and PFS. So the demographics, the median age was 46 versus 51. PD-L1 status, PD-L1 positive, almost 1/3 of those patients were PD-L1 positive, more than 1%. Recurrent and metastatic were equally distributed in both the arms, almost 50% in both, and with good performance status. And this is one Kaplan-Meier which really says it all. You don't need more evidence than this to understand how good this drug is. If you look at comparisons, the toripalimab and chemotherapy arm, it is 21.4 months median PFS versus 2. So huge difference in PFS itself. The curves separate early on at 5 to 6 months, and then the separation stays. This is all, as we know, with all immunotherapies. So if you get a response, it's a longer response and it plateaus off. So this is what happens with toripalimab, 48% reduction in the risk of progression to that. So this is a very huge advantage in PFS, which we get by combining toripalimab versus cisplatin and gemcitabine. Looking at the subgroup analysis, disease burden, again, both in recurrent and metastatic disease, it showed a good response. EBV copy, I'll come to that in a bit. And the PD-L1 score, both the PD-L1 score, positive or negative, there was kind of response, a little better response for more than one, but irrespective of the PD-L1 status, this immunotherapy can be given. And most of them were squamous cell, non-keratinizing, unclassified, but the response was there throughout in all subgroups. The OS, interim OS, this was the second endpoint. If you look at different years, at 1 year, it is 90% versus 87%. At 2 years, it's 78 versus 65. And at 3 years, it is 64 versus 49. So up till 3 years, that the difference in survival maintains, almost good 3-year data. And again, as with all the immunotherapies, the curves separate early on, and the immunotherapy arm plateaus off after some time, where the overall survival benefit is there. So if you just go to the first slide where we had gemcitabine and the survival was meagerly reaching up till 22 months, here you have additional survival, and even at 3 years, 64% of people are still alive, 37% reduction in the mortality risk. So this is data which we never imagined in NPC, so much of survival and so much improvement in survival. So the complete response rates, again, 1 in 4 patients got complete response, and median duration of the response was 18 months. So compared to when you add this toripalimab to your chemotherapy arm, you go from duration of response of 6 months and triple the duration of response to 18 months. So that is as good as the drug gets. Safety trial, no specific safety adverse events. Similar to all the immunotherapies, we have used this drug, and it's the drug, the immune reactions, what the other drugs have there. The slow dissociation doesn't increase any specific safety measures. Adverse effects, almost the same in both the arms. The typical immunotherapy, the rashes and the autoimmune infections, that is a little higher in the immunotherapy arm, but as expected with all other immunotherapy drugs. So in conclusion, Jupiter 2, it was a phase 3 randomized control trial, addition of toripalimab versus the placebo to the chemotherapy arm of cisplatin and gemcitabine, showed superior PFS and OS. The trial met its primary endpoint, and no new safety signals have emerged. And this obviously cemented place of toripalimab in the NCCN guidelines. If you look at NCCN guidelines a couple of years back, there was no mention, and you had a list of drugs to choose from, because NPC is always it's a difficult disease to treat. And when and look at today's guidelines, 2025, and cisplatin, gemcitabine, and toripalimab, it is category 1 recommendation as of now. The drug really works. The toripalimab subsequent line. So what happens if your patient is already exposed to previous regimens and already previously treated? The Jupiter 02 trial was treatment-naive patients. So in this patient, this is the data Polaris 02 for single agent toripalimab. Again, inclusion criteria, good performance status, all adult patients and previously treated NPCs or intolerant to other types. The dose here, this was the dose finding earlier study. It was a phase 2 trial, 3 mg per kg. The dose approved today is 240 mg flat dose. And this was given once every 3 weeks until disease progression, death or intolerable toxicity or voluntary withdrawal. So this is a drug which we started and continued till progression. Primary endpoint of the IRC was overall response rate. Secondary endpoints were duration of response, disease control rate, PFS, OS, and obviously the safety data. Again, the demographics almost same. Almost everyone had finished at least 1 line of therapy. 50% patients had progressed on 2 lines of therapy. So all those of us who know that an NPC who has progressed on 2 lines of therapy, probably we are going to tell only palliation in our clinic. And here is another option where the patients do well. So overall response rate in this group where patients that progressed on first and second line was almost 20%. Median duration of response was 12.8, and overall survival was 17.4. So again, mind you, where those NPCs who progress on within 2 lines of therapy, your probable survival is less than a couple of months. And again, now we are pushing the survival to 17.4 months, which is really a big achievement. Overall response rates 20.5%. Disease control rates are almost 40%. So this is by both immune resistant and risk criteria. So this was really a word about the EBV DNA. We know EBV is one of the causatives. And the treatment regime also showed multiple fold reduction in the EBV DNA from baseline. And those patients who have reduction in the EBV titer, they showed a difference. So EBV DNA, although it was not the subgroup analysis, EBV DNA titer and reduction in the EBV DNA titer can be used as a surrogate end marker to predict responses in our patients. Again, safety signals, no specific safety signals, the usual side effects, the hepatitis and proteinuria, and no life-threatening side effects. So in conclusion, POLARIS-2, a large prospective clinical study for chemo-refractory recurrent metastatic NPC, progressed on first or second line therapies, and it showed a manageable safety profile. The overall survival was 17.4 months. Response rate and duration of response. Importantly, both these were proved in patients who usually would go to palliation, and there was subsequent substantial benefit for those patients with NPCs. And again, the EBV DNA, it gave us food for thought. Probably EBV titer and monitoring EBV titer might help us understand how to treat and whom to treat the patients better. So again, in NCCN guidelines now, when the patient is progressed, again, toripalimab finds its way as a single agent as well. So upfront, it can be given in combination, the category 1 recommendation with chemotherapy and toripalimab. If the patient has progressed on first or second line therapies, then you can use it as a single agent, toripalimab. Thank you so much.
Thank you, sir. We'll proceed with the last session before heading to dinner. I would like to invite Dr. Lalit Mohan Sharma, sir. He's a medical oncologist from Jaipur, to kindly come and do the moderation for the next panel discussion for head and neck cancer. Please, sir.
So very good evening to all of you. I thank Dr. Ghanshyam Biswas, Professor Dr. Hemant Malhotra, sir, Dr. Jyoti Bajpai. You want to say something, sir? To organize a conference of this stature needs a lot of guts and courage. So thank you, Dr. Ghanshyam Biswas, Dada, for your excellent organizing this team. May I invite my panelist, Dr. Roshni. Dr. Roshni is a very good friend of ours from Tata Memorial Hospital. Currently, she is practicing in Jabalpur. Dr. Priya Nayak. Dr. Priya is medical oncologist from Bhubaneswar. Dr. Ganesh Subudhi, medical oncologist from Bhubaneswar. Dr. Moharana, Department of Medical Oncology, Bhubaneswar. Dr. Anand, please come. Dr. Anand is nuclear medicine expert and PET-CT expert from Mumbai. Dr. Suhashish Mishra, he is from Department of Radiation Oncology. Dr. Satyavrat Das, he is surgeon amongst us from Bhubaneswar. And Dr. Ipshit Das is also from surgical oncology from Bhubaneswar. So we have a very balanced panel of medical oncologists, surgical oncologists, nuclear medicine and radiation medicine experts. So let us begin with the case. We will have 3 cases. First will be early stage, second will be locally advanced, and third will be metastatic disease. So to start with, this is case 1, 42-year-old male, no comorbidities, no other problems. Patient has been detected to have T1 or T2 oropharyngeal lesion. Biopsy is suggestive of moderately differentiated squamous cell carcinoma. PET-CT scan suggestive of no metastasis. So coming to the treatment option in such early stage oropharyngeal cancer. So let me start with Dr. Roshni. Dr. Roshni, do you have any role of medical oncologist here? Can you use the mic? So there is no, in fact, role of medical oncologist. Commonly we send them to surgeon or to radiation because these are highly curable. So may I ask my surgeon colleague, Dr. Das. Dr. Das.
Second option is to take robotic surgeon's help.
Yes, so let the surgeons highlight the robotic aspects. Dr. Satyavrat.
Yeah,
you are the only
actually,
surgeon amongst us.
Yeah, actually in oropharynx the access was very difficult from beginning, but nowadays due to the advancement in technology, now it has been come as a endoscopic laser surgery and also robotic surgery. So nowadays TORS is very feasible. So though it's a very early stage T1 and T2, so we can try because in robotic surgery we can very precisely get the margin clearance. So I think TORS will be better option also for the case.
Dr. Ipsit, do you agree with that? Any of you, Dr. Ipsit? No?
Are there any contraindications to the robotic surgery?
Actually, ma'am, it's a T1, T2. If T1, T2, I think we can go for.
And how do we address like about the post complications? They are high or they are less because this is uncommon, the TORS?
Yeah, in TORS, because the complication rate is less compared to the laser, endoscopic laser surgery.
So any role of radiation? Many times the TORS is not available, the robots are not available, the surgeons are not experienced because most commonly the lower part only being handled by the robots. So Dr. Subhashish, any issue about the radiation?
Definitely. So this is a T1, T2 oropharyngeal lesion. We suppose it's adequately worked up, so no nodal involvement is there. In case of, so after the advancement of TORS particularly, in endoscopic laser surgery has also more failure rate, but TORS had better advantage. But radical RT plus or minus concurrent chemotherapy depending on the T and N stage. So that's a very feasible and ready to go.
Which type of RT you will prefer here?
Radiation, mainly intensity modulated IMRT is the norm to give in case of head and neck malignancy, particularly to spare the organ at risk. So we generally go by IMRT up to 66 to 70 gray, and elective nodal irradiation we go less amount of dose, like around 50 to 54 gray.
In Tata Hospital, Dr. Ashwini is using the brachytherapy, in fact.
Brachytherapy,
any experience of that?
Yes, brachytherapy theoretically it sounds good. Particularly, it's more feasible in case of base of tongue lesions or tongue lesions. But in case of palate or pharyngeal wall lesion, brachytherapy has its own limitation and quite challenging logistically. So when we can achieve almost the same amount of target dose to the dose escalation to the target area with minimizing the side effect part. So so after advent of this IMRT and IGRT technique, precise radiotherapy technique, brachytherapy is not a compulsion and not a ready to go technique for radiotherapy.
But if as a patient I am going with T1 tonsil or oropharynx, my choice will be first will be surgery. You get admission for say couple of days and then discharge. No other long-term complication, and by radiation, of course, the complication will be higher, but with the brachytherapy, the complication, long-term complication probably will be less. Dr. Satyavrat, any input about the robotic surgery in T1, T2 oropharyngeal lesions?
Historically, sir, we are very reluctant for a base of tumor, but with the advent of this robotic surgery, now we are looking into it in a new angle, particularly for stage less than 3 centimeter tumors. The results are equivalent or slightly better.
Any contraindication for the robotic surgery other than the availability, cost and the expertise?
Of course, the first one is availability, the expertise and cost involved with the surgical procedure. And the larger tumors, as well as we do not tend to go for a robotic surgery if there is a particularly the bilateral neck is not a contraindication, but if there is a bilateral nodal disease, and if there is any evidence of retropharyngeal nodes, we tend to avoid robotic surgery. We rather go for a radiation, definite chemoradiation.
Yeah, so can you again highlight the role of bilateral neck dissection? Which cases are there for bilateral neck dissection?
Pardon, sir?
Bilateral neck dissection is needed, like the NCCN is in front of you.
Yes, yes, sir.
The box says that resection of primary plus ipsilateral.
Definitely, sir.
Or
Resection
bilateral.
of the primary via a robotic surgery plus a bilateral low volume disease is very much desirable for a patient point of view. If there is a volume of the disease even if it is bilateral or unilateral, particularly in a base of tongue, the results are not good. So we go for a systemic therapy upfront rather than go for a robotic surgery. So we are not, in this part of the India, we are not very used to robotic surgeries, but as far as the procedure is concerned and what I have consulted with my colleagues over in other parts of India, they usually prefer a very small tumor with low volume of disease, particularly not very high volume disease in bilateral neck.
So I think that is also very pertinent, that if the primary is small, but we find that it will be a bilateral neck dissection, better to give neoadjuvant chemotherapy also to make it more operable. So we should keep all these options open.
Depending on the location also.
Yes.
Say for example, soft palate and midline lesions, bilateral neck had to be adjusted, but particularly T1 lesions where it's lateralized to a particular tonsillar fossa. Say it is well lateralized. There are some studies to only address the ipsilateral neck there, provided in early T stage well lateralized to the tonsillar fossa.
So in fact, if you see the overall outcome between the TORS and the radiation group, the outcome has been better with the patients who have undergone surgery because the long-term complications are less. So again, like we dream and hope that more and more robots will be coming up for the head neck cancer.
This study
add to it, like when we are talking of oropharynx and discussing surgery, I think like beyond T1 disease, we should not go for surgery, rather consider concurrent CTRT, because, and we are talking of, because there will be many occult nodal meds in this kind of patients, and if they comes node positive and after surgery they will be again needing RT. If we have single modality of treatment which can cure the patient, therefore anything beyond T1, we would not consider surgery, and rather go for concurrent CTRT. And I'd like to add chemotherapy as well.
I think that is very valid point, that if you get node positive based on the histopath report, we need to add the radiation. So again, these are the areas by which we learn more and more tumor boards.
Again, sir, one point in the previous slide, the last line, which we can see about highlighting this study particularly, overall, depending on this survival, when we are comparing the TORS and RT, the survival is comparable in all the studies, bearing this only single study. So the fallacy of this study was that the patient selection bias was there, and no proper patient randomization was done depending on the disease type. So
I think very right that this study has been started, the quotation has been from 2002. So over 20 years, there has been a lot of changes in the treatment and biology understanding. So I think this we have discussed. Any role of fractionation or any special radiation technique?
Yes, altered fractionation is a accepted norm, but conventionally no significant superior benefit compared to the conventional fractionation in TCN. So conventional fractionation with a robust IMRT technique is the standard norm which we follow.
Great, thank you. So with this we end up the case 1, early stage cancer. Now case 2, 43-year-old male, T3N2 buccal mucosa lesion, biopsy is commercial carcinoma. Most commonly we get cases like that. PET CT scan, no distant meds. So treatment option for this will be which out of all these 3 options? So let me start with the medical oncologist, Dr. Ganesh.
Let me take this. So this T3N2 disease, and if it is resectable, I would like to go ahead with surgery, and followed by considering the pathology report, we can go ahead with adjuvant CTRT after the surgery.
So Dr. Roshni, commonly surgeons do operate upon, and if you see the report, 33 by 33 node positive and the lesion is like T3, T4. So you feel after the surgery CT plus RT is right approach, or neoadjuvant also plays some role?
See, the clinical examination is very important. So you need to look for skin involvement. You need to look whether your disease is going up to the zygoma. You need to look at the imaging for high intratemporal fossa involvement, and then discuss with the surgeon that what is feasible and what.
Frankly speaking, the unresectability criteria do not involve a skin involvement, or but rather if the disease is involving the pterygoid plate, high ITF involvement, masticator space involvement, internal carotid or carotid encasement is there, or prevertebral fascia is involved. So in that case, we say it is unresectable. So we choose an alternative definite form of therapy, not upfront surgery. Now, if you see there is resectable is quite clear. We will give a quite clinical negative margin after a surgical resection and reconstruction. So borderline is the term that is not there, not very clearly defined, particularly in case of head and neck cancers. So there are certain trials, and based on which we do assume that there is a subset of patient in which we can say it is a borderline tumor, and with discussion in a multidisciplinary tumor board, we probably can give some better treatment except for surgery, and which is to be chosen, taken up first, either surgery or chemoradiation. Chemo first.
Dr. Satyabrat, you also agree, or you have some additional points? Dr. Ipsit.
Yes, sir.
You are Ipsit, no? Yeah. Do you have any additional point?
No, sir. Dr. Satyabrat has told the good points. Actually, there is no defined means borderline or unresectability. Basically, whatever he told, unresectability definition is very quite clear. But for the borderline, it's not that much quite clear. But sometimes we presume that by clinically, Madam said that if it is a zygoma or infratemporal fossa we are suspecting, in that case we can try for the NSCT so that later on we can get the clear margin. But still...
So margin means what, 5 millimeter or 1 centimeter?
At least we require for the 1 centimeter, because pathologically 1 centimeter margin is having the quite good overall survival rate compared to the 5 millimeter. But still 5 millimeter is also acceptable. But 1 centimeter is quite good margin.
Dr. Roshni, you feel any advantage of giving NSCT in locally advanced disease?
The NSCT will improve the margin rate, it will improve the overall response rate, but it might also cause a delay in surgery because you are ending up with complications due to a triplet chemotherapy. There is some role of induction chemotherapy and mandibular preservation as found.
We will discuss later. Dr. Satyabrat, Madam has said, Dr. Roshni has said that it will lead to improvement in the resectability and margin. So when you are operating upon post-NSCT, you are taking up the margin as the baseline or post-NSCT?
Traditionally, we have been taught that the margin originally before any neoadjuvant therapy is the margin to be taken during surgery. But nowadays we are seeing that clinical margin of 1 centimeter, forget about the pre-NSCT margin, is enough. Having said that, I would stress upon that the role of NSCT in particular in head and neck cancer is not very well defined. So far as it is not well defined, I would say that the question of margin is still debatable.
Right. So this was about the unresectable. We will not go into too much detail. Already we have discussed these issues. So next important comes about this was study published by Dr. Patil and Dr. Kumar Prabhash, where they have studied more than 700 patients who were technically unresectable and who took treatment, and these patients were included. And they observed that patients who underwent surgery, like upfront those who were declared unresectable, and post-NACT they were reviewed, and then surgery was done. So those patients who underwent surgery, they did better as compared to patients who did not undergo surgery. So my input will be that whenever we see patients of locally advanced, in one visit if you are declaring unresectable, after NACT we should keep the window open. Some patients do show very good response. So that is important point. Next important about the,
With
if you have
induction
seen,
chemotherapy is if you're giving too many cycles, you'll complete the cisplatin dose. And if you require the cisplatin later during radiation, it
yeah,
creates a problem.
in fact, this is what I wanted to ask you, Roshni. Keep the mic close to you, Dr. Roshni. Locally advanced disease, you had given NACT, surgeon has done surgery. So now, it is not like initially it was N2, N3, now it may be N0 also. So what do you give in adjuvant therapy, especially the chemo partner?
The chemo partner is preferably cisplatin. If one is cisplatin ineligible, so you have the criteria, then you can use carboplatin. And there is also good data from TMH to use biomap, that is nimotuzumab plus cisplatin. But the weekly cisplatin dose was 30 milligram per meter squared with the radiation. The problem,
So I think these are important, that cisplatin may be tough in fact, especially if we are using 3 weekly, 100 mg 300 mg per meter square Q 3 weekly. But weekly platinum low dose along with some other drug like cetuximab or the nimotuzumab, or the other option could be carboplatin. So these are good options. If you see the patients who have undergone NACT and the surgery has shown good pathological response, even if you give these patients adjuvant therapy, the response rate or the overall survival is better as compared to patients who did not receive the adjuvant therapy. So adjuvant therapy is must, especially in the form of radiation along with the chemotherapy. Any inputs from any of you?
Yes, NACT, the role of NACT in case of buccal mucosa is only to make it operable or to functional preserve, which is still debatable. So post NACT, post surgery, adjuvant therapy cannot be omitted depending on the response. It has to be there mandatory as a treatment cascade post operative. So the in case of borderline resectable, particularly the concept of NSCLC arises, and in case of head and neck malignancies, surgery has no substitute. Like if it is surgically ineligible or inoperable, then the survival is definitely, or the outcome is definitely inferior, even though we give radical CTRT in those cases. So surgery, so all the trials which have compared surgery versus no surgery, surgery has given definitely better outcomes compared to non-surgical arms.
Very true. In fact, this study since was published from Tata Hospital, so what they have quoted, Dr. Kumar and team, that patients who were coming to Tata, got neoadjuvant therapy, got the surgery done, and then they went back. They did not come for the adjuvant therapy, or they did not take the adjuvant therapy at their local place. So the outcome was poor amongst those group. I think these are important points we have discussed. Dr. Ipsit, from you, that any complications you face post NACT while operating, do you find any complications after chemotherapy?
Actually, I have not encountered till now any cases post NACT. Whatever cases I have encountered only upfront surgeries. So till now I have not got. So I don't have any experience about any post NACT surgery. I like to ask about this, Dr. Satyavata sir.
Great. So the next important point for discussion is oral cavity cancer. The disease is resectable. Is there any role of neoadjuvant chemotherapy? Dr. Satyavrat, from you.
Sir, for me, in head and neck or oral cancers, the role of NACT does not improve any overall survival. It remains same whether you are operating, giving NACT or not giving NACT. The overall survival will remain same. Of course, there are debates whether we are able to do more of resectable, converting more of tumors to resectable one. The most important aspect is whether we are giving, we are able to do some organ preservation like mandibular preservation. So that is one of the trials that was done in Tata, and they have shown a positive results. We need more trials, such trials, to prove that yes, organ preservation with equivalent survival is a question, is a definitely pertinent question. And now for overall survival, I don't think there is till now.
So in fact,
trials do not suggest
it is not a question of overall survival alone, organ preservation. Do you feel that also is very important? Like this study, Dr. Devendra Chokar et al. Have done from Tata Hospital, yes, and they have seen that the mandibular preservation rate is close to 48%.
One problem with this study is that pitfall is the size of the patient size is very small. So we need a robust data before concluding about this.
But I think, Dr. Satyabrat, we all need to get insight from all these, and we all need to generate more and more data. But
Yes,
this sounds very important, that unnecessarily we are removing the mandible, maybe whatever part you remove, segmental or partial or whatever you name them, but that is important that we are able to preserve the mandible. Nimotuzumab has been one of the partners, radiation along with the nimotuzumab, and supposed to be good drug. Somehow it did not get much popularity, but again Dr. Kumar and team has done great work on that. So last case over last 9 minutes, Dr. Roshni, metastatic neck node with primary unknown. That patient was diagnosed in 2018. MRI finding we can see. FNAC was done, which was metastatic squamous cell carcinoma. There was erosion also of the some bone nodes were there. This was the picture initially. This patient underwent triple endoscopy, which was negative, which was normal. Patient underwent modified neck dissection. Histopath suggestive of the report 1 out of 20 lymph node positive. EBV and P16 were negative. So at this point, we had given cisplatin plus radiotherapy to this patient. This is how the radiation was given. 1 month after completion of the radiation, this patient developed a disease recurrence in the neck node on the same site, and the skin was also infiltrated. And again, FNAC from that was positive for metastatic recurrent disease. So, Dr. Roshni, any clue from your side that what do you do now? 1 month after completion, this patient has recurred.
This is platinum-resistant disease. It is not amenable to any surgical or radiation techniques, right?
So I think that is also very important, that whenever we see a recurrence in head neck, if the disease is localized, we take help from our surgeon colleague. Radiation, of course, has been given and 4 weeks back, so that remains difficult, but at least surgical intervention may be considered again.
Excuse me, sir.
Yes,
sir, this is case 1 further course or case 3? So the primary
no, this is case 3, I'm sorry.
Case 3. So this is new. So that case is basically unknown primary.
Unknown primary,
neck
yes.
dissection is done. Post neck dissection, adjuvant CTRT he has received.
Adjuvant CTRT, and after 4 weeks, this patient had recurred. Again, neck node positive. So at this point, this was the PET-CT scan, and the first recurrence. Again, our surgeon did a good surgery with whatever details I could understand, and then histopath was suggestive of the carcinoma, the node reaching up to the skin. Within 2 months of surgery, this patient again had bilateral neck node with skin and subcutaneous infiltration. There were nodular lesions, and you can see this was on the right side of me. We can see the first and the second one after 2 months of surgery. So you are from medical oncology also?
What are the age of the patient?
72.
And the smoker and non-smoker?
I don't remember that part.
Actually, I'll share my experience only. The mainly typically young patients, smokers, and tumor burden, high tumor burden. Whatever you give the treatment, they do occur. In this patient,
in fact, what you will do at this point, sir?
One of my young patient, the similar scenario, the recurrence came, and I think the skin is involved and lymph node positive.
No, the question is, sir, how will you treat
I think before second surgery, we could have given NSCT. In place of cisplatin, dosi, and 5-FU may have tried. Because
Agreed, sir. That has not been tried, but at this point, what are your treatment options?
Second or recurrence, definitely we have no options. Definitely we have to give the systemic therapy only. And recurrent setting, we can say we can add immunotherapy, and you can do send PD-L1, but we can send immunotherapy, and also you can give cetuximab.
So
both
in fact,
options can be tried.
cetuximab was available at that point. Immunotherapy, of course, was there, but it was not easily available. So to this patient, we gave the cetuximab plus chemotherapy. So I'm sorry, this is case 1 wrongly written. So we had given nab-paclitaxel with cetuximab, and the patient showed good reasonable control, but after 5-6 months again had progression of disease. So at this point, any inputs, what should we give?
You have given paclitaxel and cetuximab.
Yes, sir.
So then the remaining drugs are available, 5-FU and carboplatin also available in place, and you can definitely try immunotherapy.
So we had given immunotherapy to this patient. These are just reference. So started on nivolumab this patient, and this patient was like from a CGHS beneficiary. So the CGHS was kind enough to give the drug. So we completed 6 cycles, and this we can see the difference that after 3 months of nivolumab, this patient showed excellent response. After 6 months, we can see it has further like become normal.
This patient, you
So-
can also offer oral metronomic therapy.
No, at that time we did not give because already chemo was received, and the patient said that I'll take only 1 drug and that injection only. Oral metronomic again was one option at this point, but somehow her daughter also, this patient daughter herself is a doctor. She consulted someone else also in US, and then it was said that oral metronomic we don't give.
Because response rates with single agent nivolumab is also hardly 13%.
Yeah, so in fact, Dr. Roshni, when do you plan to stop? Like this patient had shown excellent response after 6 months. So the CGHS kept asking me that for how long do you continue? So when do we stop the immune checkpoint inhibitor in responding patients?
As per the data, it has to be 2 years.
2 years.
And
Any
then
inputs from any of you?
challenge.
We have actually, after 2 years of any immunotherapy, GI toxicities are higher. One of my patient given pembrolizumab, around 22 cycles, GI
So-
toxicity becomes higher. So we
Agreed,
can
sir.
continue till the toxicity don't bother, but otherwise, and the finance is also not a bother.
So in fact, for this patient, we continued for 2 years, and every 3 months we were writing to CGHS that 3 more months, 3 more months. And then after 2 years, we need to stop. And now the patient is keeping well for last almost 6 years. He met me
When
4
you
days.
have tried cetuximab, and that time also you could have tried low dose OMCT, low dose nivolumab with OMCT.
Yes, sir. In fact, low dose, again, the concept was not available at that time. And the CGHS approval, again, they always ask for the guidelines. So whatever we write, they want some guideline also copy along with that. So they approved this patient for this. So I think we have discussed any inputs or any comments from any of the panelists about
My point is that if young patients and smoker and tumor burden high, in that cases, we should definitely think of any systemic therapy. For example, margin positive and PNI, LVI positive. There also we try RT, but at that time we should add a cisplatin at least concurrent. And whenever there is a chance of adding systemic therapy, you should add it because these patients do bad. And if the tumor, again the recurrence is there, then tumor burden high, the response to the next line of therapy, usually they don't get that much of response.
And I would disagree with him because he's a platinum-resistant relapse. Platinum-resistant relapse doesn't do good with chemotherapy. Option is single-agent immunotherapy. So patient had responded to nivolumab. Why are you asking for chemotherapy?
So I think
platinum-resistant relapse.
looking to
Within
this-
6 months, patient had relapsed.
That basis you can add, but in the upfront setting, I think. If PNI positive, radiation, when we told, usually all radiation oncologists say that you only give radiation. If there is some risk, additional risk, then you add cisplatin in the upfront setting.
I think in this case, the lacking part has been the oral metronomic therapy. Dr. Banawali sir is here, and he has shown excellent response. I also tried to give this patient, but somehow being from a doctor family, well connected, they took opinion from someone else in US, and it was not taken up. So thank you so much, all the panelists. Any comments from the floor, from anyone? Okay, if there are no comments, then I thank all my learned panelists and thank you all. Thank you, Dr. Kancham Biswas, for wonderful time given to us. Thank you.
Thank you, Lalit sir, and thank you so much. So I would admit that so before Banawali sir, before Saike sir, my first oncology guru is actually Lalit Mohan Sharma, because in 2001, so there were no rooms available. So I was just carrying my backpack. So he allowed me, तुम मेरा रूम में रहने का है, but दरवाजे के पास होने का, रात को blood platelet sign करना है. So that is how we entered. So in fact, he is my first guru in oncology. So thank you, Lalit sir. So Odisha being the hub of head and neck, so Rajasthan is no less than in head and neck cancers. So we are almost 20 minutes before schedule, so we have finished on time. And this is the last session of day 1. Thankfully, everybody. And so after this, we can have dinner in the ground floor, so where there was lunch today. And tomorrow morning, 8 a.m., we meet for the meet the professor session, followed by the scientific sessions from 8:45. And I thank my master of ceremony, Dr. Ritu and Dr. Antara. And thank you so much. So everybody please join for the dinner. So tomorrow we have meet the professor. So we have inauguration, we have talk by the Nobel laureate, and at the end we'll have gala dinner, and we have a musical night tomorrow. So there are lots for tomorrow. So have a comfortable sleep, so you can take rest, whoever have come today, until we make in the morning. Thank you.